questions on the dihybrid. So please take note of that we don't ask you everything on the dihybrid. Okay, now the question is saying that they are saying that set Z is this with this. So basically, I'm gonna just draw a line this, you write what's there, and also is this line what's there. So it means that I'm gonna say small f, and then capital H, small h. Yeah. Why didn't I start with the capital H? Because these are two different alleles. Yeah, these alleles, one allele does not interfere the existence of another allele because these are different genes. Yes, Mendel's law of segregation. Mendel's law of, yes, independent assortment, not segregation. Mendel's law of independent assortment. The one allele of one gene does not influence the other allele, meaning that if you have a big head, that is not connected for you. Why do you have small eyes? So the allele, a gene for the big head, does not influence small eyes or the type of eyes you are having. So they are totally different. That's why I didn't start. But if it is the same allele, H and H, then automatically I can say H, capital H. No, I have to start with the capital letter. Yes. Okay, now let's go to our question. The question is saying that give the genotype of parents. How do I identify the genotype? This is a very challenging question to kids. But let me just show you how you can get the genotype of the parent, because they didn't give you anything here. But look at the gametes. Pick a letter which is not repeated, then write it down. The first one is capital F. Capital F. Make sure that you don't repeat the letter before, because there's supposed to be two alleles for each character. But first write the letters which are not. Don't repeat the letter before you complete. It's capital H. Capital H, we have talked about. Capital F. Small f. So I add the small f. But here is small f again. So I've already written it. Then I go to H, capital H. Capital H. I've talked about the capital H, small h. So I've got the genotype because they are supposed to be 4. And then when I come here, is still capital H, capital F. Yes, then small f. And then capital H, and then yes, small h. So yeah. So it means that both of them, they have the same kind of what? They have the same kind of allele. They are heterozygous for their conditions. So all of them, they have the same kind of what? Condition. Then they are saying that number of genotypes that could result in order in offsprings with short fingers and continuous hairline. Yes, number. So in this case, we are going to say number of genotype that would result in offsprings with short. Let me underline this. They are saying short, but short, what did they say? Is short is the capital F, is dominant. So I'm gonna look for anywhere I have capital F. Yes, and then they're saying that fingers, continuous hairline. Continuous hairline, we say that is recessive, meaning that if it's recessive, small h. But it cannot be recessive, cannot show in the genotype unless these two are being shown. Yeah, showing. So how does it show? It's supposed to show by being recessive. So because if I have capital H and then small h, then automatically this individual is not going to be continuous. It's gonna be this Windows what peak. So automatically it's supposed to be small letters, small letters. So I'm going to look for where there is a small letter, and also I'm going to look for where is the what? I'm going to look for where is a capital H. So once I get that, then automatically I'll be able to identify how many individuals are there. So I'm gonna say capital F, small h. This is the first one, the first individual. This is the second individual. Is there anyone else? Yeah, the third individual. Is there anyone else? No. So there are three. So the answer is going to be three individuals. Then they're saying that allele for continuous, allele is, remember, it's caused by recessive allele. So they're not saying alleles, saying allele. They're not saying genotype, because if you say this is the genotype. They're saying allele. Remember, it's caused by recessive allele. You see, this is caused by dominant allele, meaning that these are recessive alleles. So please do not write 2. It's supposed to be 1, because even they're here, they're saying allele. Phenotype of a child who is homozygous recessive for, they're saying phenotype of the child who is homozygous recessive for both characteristics. Is here long finger and continuous hairline. So that is it. So this is a dihybrid cross. So if they ask you which type of cross is this, is a dihybrid cross. Next question. The next question is also about a dihybrid cross. Dihybrid cross. Dihybrid cross. What does it mean? What does it mean? Dihybrid cross. I hope you are seeing clearly. There is variation in characteristics, yes, of fur color and texture in the cat. All right, I'm seeing, and then they are saying that the table below shows the allele that control these two characteristics. These two characteristics. All right. What is next? They're saying that you have these characteristics. The Punnett square below shows the inheritance of alleles in the genetic cross. All right, here's the Punnett square. Name the dominant allele for fur color. Here you come where you see capital letter, it means the dominant allele. So this is B. So the answer here is going to be black. This is black. And then the recessive phenotype for fur texture, recessive, is going to be smooth. So you see how the hybrid is being set. It's not a complicated thing. It's just a normal thing. It's just for you to see that you can answer this. You can answer this. Okay. So they are saying that so we are saying that give the genotype of offspring X genotype. They are saying genotype. So if you are saying genotype, then it means that you're going to come here and here. So it's gonna be small b, capital R. So that is the genotype for that individual. Yes. Then they are saying that the genotype, the phenotype of female parent is female parent. This male and this female. They're saying the phenotype is capital B, capital R. Capital B is black, and the small r, small b is white, and capital R is rough. So it's gonna be white and rough. White genotype of the parent. The genotype of the is gonna be white and rough. But I have skipped this. The genotype of X, yes, we have talked about it. So that is the answer for that. The genotype of parent male. Now you come here, male. They want the genotype. So how do I identify the genotype? How do I identify the genotype of this parent male? So I can only do the genotype of this parent male when sorry. I can only, as I taught you that what you do is you come here, say capital B, yes, you don't repeat it, né? Yes, you don't repeat it, capital B, small b, yes, and then you go capital R, capital B, yes, you don't repeat capital B, but you have a small b here, put a small b, then you capital R, you look for, you have a small r, so small r, so that is going to be the genotype of that individual. Yes. Then they are saying that what is the genotype of the parent male? So you are done with that. State all the possible genotypes. The possible genotypes of what? State all the possible genotypes of offsprings of this genetic cross. All possible. State the phenotype of all offsprings of this genetic cross have in common. They're not just saying offsprings, they're saying what they have in common. Okay, come here and see. This is Z. Okay, capital B. Have a capital B. This one has no capital B. You see? Yeah. This one no. So it can't be capital B. Capital R. This one has a capital R, né? And then also this one has a capital R. So basically, state the physical, the phenotype of that all the offsprings of this genetic cross have in common. So definitely here I'm gonna say capital R, because we say that all of them, they have capital R. It has capital R, it has capital R, capital R. I come back here and I ask myself, what is capital R? Capital R is a rough. So basically, what they have in common is rough. Yes, that is what you can do. That is the dihybrid cross. So we have seen questions of dihybrid cross. DNA profiling. What is DNA profiling? DNA profiling, when you are going for DNA testing, you are going for DNA test, we are just going to do what you call DNA profiling. So we make the profiles of DNA. So how is it identified? You use the, it is shown on the screen as bars. So if the bars are corresponding, then it means that those organisms or those individuals are related to one another. So DNA profiling is a technique used to identify someone's unknown DNA profile using a known DNA profile. For example, you are looking for your father, and then you don't know your father. There are two guys who have come, and these two guys, they are claiming that you are the son or you are the daughter of those guys, but only one guy will be the father of that daughter or that kid. So now I know the DNA profile of you, I test the DNA profile. So your DNA profile becomes the reference point. So that is the known DNA profile. So this known DNA profile, I compare it with these two people whom I don't know their DNA profile. I go and then I test their DNA. If I find that one of them corresponds with my DNA profile, then it means that that is my father. If the two of them, no one corresponds to my DNA profile, then it means that nobody there is the father. So that's how this technique is used. Is a technique we use to identify someone's unknown DNA profile using a known DNA profile. So you are the known DNA profile, the father, the other two guys are the unknown DNA profile. So if they correspond, then we say that you are the father of that kid. So here is an example. Yes, for example, this is a scene, crime scene, someone committed a crime. Therefore, when I go to the crime, I find some material, for example, hair, blood, I collect that sample. When I collect that sample, I take for DNA testing or for DNA profiling. So I get the profile of the DNA. That's why in most cases, when there is a crime scene, we don't allow people to go there. Why? Because you might bypass there, and then when they come to collect the sample, your hair is included there. And then you're gonna be one of the suspects because your DNA, once they test there, they see that you are among the people who were there. That's why if there is a crime scene, don't tend to go there if you are not an investigative person. All right, so you're saying that the crime scene, this is the sample they picked from the crime scene. So here we have the suspect. We have three people whom we are claiming that these are the people who commit the crime. So what they do is to get their DNA profiles, and then I start to correspond with what we found on the crime scene. So if I use a ruler and then I draw here, there is nothing here. I draw this corresponds, that's one. This one corresponds, that's two. There is nothing here, there's nothing. Then this one is three. This one, there's nothing corresponding. And then this one is four. So this one is gone. Then what about this? This one is corresponding. 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 This one is also corresponding. So all of them are corresponding. What about this? Shows that there is some which are corresponding and some are not corresponding. Therefore, we will conclude that the suspect number 2 is the person who did the crime. Why? Because what we collected from the crime scene and his or her DNA profile are exactly the same. Therefore, suspect 2 will be taken for accountability of this. So what are some of the uses of DNA profiling? What are some of the uses of this technique? What are some? Yes. Okay. Number one, it is personal identification. You know about the person someone is missing. So it's very easy to identify the person, the identity. Someone has burned somewhere, so it's easy to identify the identity of that person. Then number two, paternity and maternity. Maybe your mother, you don't know your mother, and there are two ladies who are claiming that you are the mother. They are the mother, so or father. we can use this technique to identify the actual parents of this. How is this one possible for mother? Maybe in the hospital, and they have swapped the kids. You know, when the kids are just born, in most cases they don't tend to resemble their parents, but as they grow on, they turn to resemble their parents. So by that time, still we can do what? We can find the parents there. Then another one is diagnosis of inherited diseases. Diagnosis of the inherited disease. There are some diseases which can be diagnosed using DNA replication to know actually that this is the disease you have using using DNA profiling. So diagnosis of inherited disease, and sometimes we can even find a cure for those diseases. Then another one is criminal identification. Criminal identification, for example, this on the crime scene, they go take the samples which are there, and then they compare to the person they are suspecting, and then we find the person who they did the crime. Then forensics, maybe someone is burned still, maybe in an accident, you can't find anything, so we can still use DNA even if it is 20 years. As long as I get any remain of you, still we can find the DNA from there, and then we can correspond it to. Then they are saying that identifying suitable organ donor it's not easy to find the organ donor. So you have to first see that if the DNA profile of you and the person you are giving the organ is compatible or is almost the same, or otherwise the organ will be rejected by that person's body. So what we do is to first identify to see the correlation between you and the person who is going to receive the organ. And then once these they are compatible, then they transfer the organ from one person to another. Then what are some of the disadvantages of this technique? Number one, it is expensive. Not everybody can achieve or can afford to do this. Not everybody. So it is very expensive. So it requires people who are, yes, well off. Then number two, human errors. Some people imagine someone is drunk, maybe a bear is drunk and then go back and then is trying to analyze. Definitely is going to have some errors. Even if it is not that, still when you're analyzing, you can have some, you are not 100% correct. There are some errors which are developed there. Yes, that's why we increase the sample size, and then also we repeat the experiment so that you increase the reliability, so that you increase the sample size. So it means that you have to be with a lot, and then you have to repeat this technique so that you are sure that what you got is exactly even what another person is gonna get. Then we shall say that that's maybe the human errors have been minimized. Not all hospitals have necessary requirement to carry out this DNA profiling, just like schools. But look at the schools, you'll find out that some schools, they have better equipments, they have almost everything students need to learn. But other schools, they are struggling. Whether they are government schools or where they are private schools, you will find them that they have different categories or different degrees. What about now hospitals? Still, some hospitals are well off. They can have these techniques to be supported. Others, they don't. Others, they have equipments, but they don't have the chemicals they use for this to happen. So you see that some hospitals, they don't have necessary requirements to carry out this DNA profiling. So then it is possible to plant DNA at a crime scene, giving false evidence. Yes, it is possible. If someone wants to frame you, then automatically can be planted, and then it means that this is not a good idea of DNA profiling. So, innocent people or persons' DNA might be at this crime scene, at the scene or crime scene, even though they had nothing to do with the crime. That's why I say that you are just passing, and then your hair drops, and then when you're collecting the sample, then they also take your hair, then you're innocent. But because the DNA profile is showing that it's you who was there by the time the crime was committed, then automatically you're gonna be among the people who are suspects. So that's why we are saying that, yeah, DNA profile has some disadvantages. So let's look at RNA, ribonucleic acid. So once we talk about RNA, then it means that we are going to start what you call protein synthesis. So RNA and DNA are almost the same, but they differ in some few aspects. Why am I saying they're same? Because both of them, they are nucleic acids. There's 3 types of RNA based on the function they do. So there are 3 types of DNA based on the location. But RNA, there are 3, but these ones, they are based on the function they are doing. So number 1, we have messenger RNA. As you hear the word, it is a messenger responsible for carrying genetic information that is transcribed from DNA. So it carries it from DNA and then takes it to cytoplasm or to the ribosome so that protein synthesis can be made. Then number 2, you have ribosomal RNA. That is, it means that it is forming the ribosome. It forms the skeletons of the ribosome. So this one forms the ribosome and produce the proteins based on the information received from the transfer RNA. So the transfer RNA, when it's coming, it comes to the ribosome. So the ribosomal RNA is just the skeleton of ribosome. Then you have the transfer RNA. This one changes, or it has what called anticodons. It code for the specific amino acid. So it transfer the amino acid from the cytoplasm and then brings it to the ribosome so that a protein is what is formed. We will see this when you go into detail of protein synthesis. So what is the role played by RNA in protein synthesis? Number 1, messenger RNA molecules carry the coding sequences for protein synthesis. Number 2, ribosomal RNA forms the core of the cell's ribosome. And then number 3, transfer RNA carry amino acids to the ribosome. That's what I've been trying to explain, that their function determine the type of RNA it is. What they do will determine what that type of RNA is. So there are 3: messenger, ribosomal, and then transfer RNA. So what is the structure of RNA? Structure of RNA is almost like DNA, but just a small difference of uracil instead of thymine, and then a single strand instead of double strand. For it is straight, it's not helical. So saying that a single-stranded molecule consists of nucleotides. We saw that it's also containing nucleotide, phosphate, sugar, and then nitrogenous base. But the phosphate is fine. Sugar is ribose instead of deoxyribose, and nitrogenous bases, it has uracil instead of thymine. Nucleotide is made up of sugar, that is the sugar. Which type of sugar is ribose, and then phosphate and nitrogenous bases. It has a phosphate nitrogenous bases, and then 4 nitrogenous bases of RNA are adenine, yes, adenine, uracil, cytosine, and guanine. So these are the 4 nitrogenous bases regarding to RNA. So this is the basic structure of RNA. So this is the messenger RNA. This is how messenger RNA looks like, and then this is the transfer RNA, how it looks like. Then a ribosomal RNA, it just forms the ribosome, and basically for this level, you don't need to know its structure. Yes. Let's look at protein synthesis in detail. Let's look at protein synthesis in detail. Welcome back to our class. Today we're gonna be looking at the paper 2023 final exam. Yeah, so share the video so that everyone is here. We can discuss this paper. No one should miss out. This paper was really a very easy paper for those people who did the study, and for those people who did use my book for distinction material. Yeah, if you want it, you always get it from the link below. So this paper was a little bit easy, was just a little bit of easy, and we're gonna see how we can really see that those people, they can mark themselves and then they see, and also those who want to prepare for the next exam. All right, let's start without wasting time. M.Saidi, as usual. According to Lamarck's theory, I know most of you don't like Lamarck because of his theory, but me, I like him because he started the story. All right, according to Lamarck's theory of evolution, or for evolution, acquired characteristics are not inherited. Obviously, we say that Lamarck says that acquired characteristics are inherited to the next generation. If you have a person, maybe your father gets an accident or is a bodybuilder, and then you're going to produce kids who are having muscles already. Acquired characteristic is inherited. So here the key point is not. So you have to take note of that. So this one was off. And then they are saying that organisms evolve because they don't because they adapted to their environment. No, here they are saying that organisms evolve because of the adaptation to the environment. How do organisms evolve due to the adaptation of the environment? Is it natural selection? Because adaptation will require that those organisms which are able to adapt will evolve, and those organisms which cannot adapt will die. Yes. And then number 3, they're saying that there is variation among organisms. Lamarck doesn't talk about variation. Yes, so this one is off. Then they're saying environmental change has no influence on the species diversity. Here, they're saying that environmental change has no influence on the diversity of the species. And then here they're telling you that organisms evolve because they adapt to their environment. So how are these two different? How are these two different? Here it means that environmental change have no influence. For example, that if there is scarcity of food, yes, and then the one which is on ground die out. This is opposite of this, né? And they die out, and then there will be no influence on the species, how they diverse they are different from each other. So that is wrong, because species will change based on the what they want, but still based on the environment. If they want to be, for example, they want to stretch their necks, they stretch their necks until they reach, for example, the tortoise. If we are using the giraffe, yeah, you call them giraffe, yes, because that's the best example most of the people are looking at. So the best answer here is gonna be B. And then they're saying that genotype for species characteristics. When you talk about the genotype for species characteristics, what does it mean? They're saying that contain 2 chromosomes. No. Physical appearance, they're saying genotype. Physical appearance is a phenotype. No. Is the composition of a gene pair. Look, they're not saying that genes. They're saying gene pair, meaning that you have, remember that when you talk about an allele, is also a gene, but is half of a gene.
or alternative form in which a gene can exist. For example, tallness is an allele, and also shortness is an allele. For example, capital T, small t, for example. Yes, so this is an allele, this is an allele, but this is a gene, meaning that it's a pair which makes a what? A gene. And then is represented by the genotype of a specific characteristic. Here, characteristic. Here they're saying it is represented by one allele. It's not represented by one allele. Even if it is shortness, it will be represented by the two alleles. Yes, for example, capital R, or small t, they are saying a characteristic. Yes, so when you talk about the characteristic, it means that they are looking for the complete set of that characteristic. So the best answer here becomes C. And then they're saying after discovery of fossil, scientists classify it as after discovery of a fossil, scientists classified it as an African ape because it had, okay, what do African apes have? Large cranium? No. Small cranium? Yes, they have small cranium. Small cranium? Yes. Large cranium? No. So meaning that A and D are out. So these two are out. So let's look at this. Small cranium, non-prognanthus, meaning that they don't have a protruding jaw. Apes have protruding jaws. Yes, prognanthus means protruding jaws. So prognanthus, this is correct, this is wrong. So it means that the best answer is C. They're saying that large canine. This one is correct, this one is correct, but this is wrong. Therefore, B is wiped out. The best answer is C. So C is going to be the most correct answer. Yes, let's go to 1.1.4. The statement below describes the step in the process of cloning. I explained this, the process of cloning yesterday very well, but let's ignore that you are not there. So now let's look at it. Remember, they are saying that embryo, okay, let me just show you here so that number one, you get one cell, you get a cell A and cell B. So this is animal A, animal B. Get a cell, ovum. I'm just summarizing it very fast. Somatic cell. So here is somatic cell, meaning that is 2N. After that, remove the nucleus of the ovum. Yes, throw it away. Take the what? The casing. And then here, remove the nucleus because I want the characteristic of this. Bring the nucleus, throw the casing. So bring the casing here and bring the nucleus, fuse them together with electric shock. Yes, after that, bring these two. Once it is fused together, it looks like a zygote-like structure. Therefore, it becomes, I'm just explaining so that because this is the process they are trying to show you, it undergoes mitosis. Bas. Then let's go back to the question. They're saying that the statement below describe the steps involving in the process of cloning. Embryo in the implanted is implanted into the uterus of an adult female. Embryo is implanted. This must be around this stage 4. Okay, we shall find out. Nucleus from the somatic cell of the donor cell is extracted. This could be 1. Nucleus. Check. Nucleus. They saying the nucleus or the cell? Nucleus from the somatic cell. That's not step 1. You can't extract the cell before you get the cell. You can't extract the nucleus from the cell before you get the cell. So we need to first get the cell. We are looking for these 2 steps of getting these 2 cells. All right, we go back. They're saying that the nucleus from the somatic cell is inserted into the ovum. The nucleus from the ovum of another is removed. The ovum from, okay, now we have to look for it. They're saying that the nucleus from the somatic cell, meaning that, okay, they obtain the cell, yes, but now after obtaining the cell, in this statement, they're not talking about this. So meaning that they're starting from this. They're not talking about this. They're starting from 1, 2. Okay, let's go back. They are saying that the nucleus from the somatic cell of the donor is extracted, meaning that the nucleus is removed, is brought here. So this could be step 1. Okay, we shall see if it is the most correct. Step 1, so this is covered. They are saying that the nucleus from, yes, the nucleus from the somatic cell is inserted into the ovum. The nucleus, this is done. The nucleus is of the somatic cell is inserted into the ovum. Do we have the ovum? The nucleus from the ovum of another individual is removed. This is step 2. Yes, this should be step 2. Because I removed here the nucleus, so I remained with this. So now I inserted this into this. This should be step 3. The nucleus from the ovum of another cell is removed. It's done. The ovum of the new cell is given. The nucleus from the somatic cell is inserted. Now this is step 3. So meaning that now I have 2, 4, 3. Then I'm remaining with this and this. So they are saying that the ovum is now, the ovum with a new nucleus is now given electric shock. Now we are here. We are giving it electric shock, meaning that now this becomes 4. Yes, and then now the embryo is implanted into. So meaning that I'm going to have, yes, it's going to be 2, yes, 4, yes, 3, 4, 1. Meaning that 2, 4, 3, 5, 1. So this is the most correct answer. So the answer is A. Let's go to the next question. Then they are saying that 1.5, shrubs, these are short grasses of the family. This family, they are saying that can be found in Australia, South America, Indochina, and parts of Africa, as shown in the map below. It is hypothesized that all continents were once one large continent called Pangaea, and that they separated due to continental drift. They said, okay, sharp. They're saying that this is evident. This evidence that the family, yes, this family, what about it? All belong to the same species. No, they don't belong to the same species. They don't belong. That's not an evidence to show that they belong to the same species. Number 2, they're saying that they are equal distributed in all continents. Who told you that? They are not equally distributed. Are you trying to mean that what is here and what is in South America and what is in Africa, they are equal? No. Wrong. They're saying become extinct. They are still existing. They're still existing. Arose from a common ancestor. Simple, because at first they were together. Yes, but they went to different environment due to the separation of the continent. And then they developed into different species. That's why we say that they don't belong to the same species. Therefore, D is the most correct answer. 1.6. Yeah, these ones, they take some time, but when you reach there, we're going to be faster. The diagram below shows the DNA profile of a girl that her mother and 4 males. What about the males? Okay, maybe they are claiming. Which male is a girl's biological father? I have to look for all the bands which correspond to the mother, because the mother is known. This goes with the mother. Done. This goes with the mother. This goes with the mother. Now, the remaining bands from the girl must be matching with the guy who is claiming. Okay, let's go. This one matches. Cool. Yes. This one matches. Is there any? Yeah, because this one is matching. So is there any other for the girl? No. So it means that this is number 3 is the biological father. It must All of them must be ticked for the case of the girl, but they must be the remaining one, which did not match with the mother, must match with the father. Hence, number 3 is the most correct answer, which is C. Okay, the following are the characteristics of group of animals. Yes, animals. If you're saying animals, just a moment, guys. Okay. Just a moment. Okay. Animals. They're saying that which characteristics of group of animal able? Which are the characteristics of group of animals able to interbreed, occupy the same habitat, produce infertile, belong to the same species? Which combination correctly represents a population? Which combination represents a population? Yeah, it means that you can't know which group represents a population. Just a moment, guys. Let me increase here so that I can see clearly. Yes. Yes. Which population does the population is being shown by what? Is being shown by the able to interbreed, and they must produce a fertile offspring, occupy the same habitat. Yes, they must be able to interbreed, but interbreeding they must produce the fertile offspring. And the only word which can help us to bring about to produce a fertile offspring is one word. Which word is that? Same species. They belong to the same species. So the answer is B. Same habitat, same species. Yeah. So we go to the answer becomes 1, 2, 4. 1, 2, 4. Where is 1, 2, 4? Is there any 1, 2, 4? 1. 1, 2, 4. Yeah, it's B. Yes, it's B. So yeah, it gives me the correct answer. Let's go 1.8. Which diagram? Let me see. Which diagram? Not which diagram. During anaphase 2 of meiosis, the two chromatids of the chromosomes are pulled apart, each moving towards the opposite poles at a rate of 1 micrometer per second. This question? Okay. I see it as a new content to students. Anyway, but it's just an application question. Okay, they're saying that, yes, two chromatids of the chromosomes are pulled apart, each moving towards opposite poles at a rate, this is the key point, at a rate of 1 micrometer per second. Okay, each second, 1 second, it means a distance moved is 1 micrometer. Let me say 1 like that. The distance in micrometers between the chromatids after 20 seconds. Obviously, if it's 1 second, 1 micrometer, after 20 seconds, they'll have moved 20 micrometers. So that is a direct question. However much is just an application question. So the answer becomes B. Yes. So the answer there becomes B. Yes, let's go to the next question. The next question, they're saying that, which one of the following indicates the type of variation? I gave you examples yesterday. Anything which does not change since you are born is continuous variation. Anything which changes as you are growing up, it's continuous variation. For example, here they give you height. Height changes, therefore is continuous. The way you are born and the way how you are now is not the same way. Height is continuous. So it's where there is this continuous is, I think I have the answer now. Because the rest are wrong for the height. And it can't be correct for the next one. Okay, skin color. I don't think most of us, we are born yellow bones. Check us now. Hey, we are original indigenous. Yeah. Yes, color changes, skin color changes. You check the kind of people. Some people, I want the black one. I want the light skinned one. I don't know what you're gonna use for them for, but it changes among the individual. All right, this is continuous. Check. Earlobes. The way those earlobes are since we are born, unless you get an accident and then they change. But if not, it's the same thing. So correct. So let's not waste time there. Answer is B. Let's go to terminologies. I know sometimes the terminologies, they make these students a little bit of a problem with it, but I don't think these terminologies were that hard. Ne? Let's go. The first terminology, they are saying the type of inheritance which produce intermediate phenotype. Don't forget that we said it in the what? In the distinction material book, our distinction material book. Ne? Yeah, for those people who were not there. But okay, there we go. Intermediate. How do you call this? Incomplete. Incomplete dominance. Intermediate. Yes, we talked about it. So the answer here is incomplete. Complete dominancy. Yes, done. The point at which adjacent chromatids overlap during crossing over. Chiasma. Guys, spelling is mandatory. The process by which information is copied from DNA to RNA. Transcription. Told you. Transcription. Let's go to the next question. What are they saying? They're saying that sugar found in DNA. Sugar. Thymine. They're saying sugar, not base. Gone. Thymine. That's a base. They're saying sugar found in DNA. Ribose is found in RNA. So for the DNA, it's going to be deoxy. That's why he's saying D. DNA, deoxyribose nucleic acid. So the sugar found there is deoxyribose. The bond that occur between nitrogenous bases in DNA molecule. The bond that occur between nitrogenous bases in DNA molecule. So how do you call that bond? That bond is weak hydrogen bond. Yes, you must say weak hydrogen bonds. Most of the people, they wrote peptide bond. Peptide bond. In some cultures, they say that sometimes you can laugh because of sadness. Not because you are happy, but you are sad. The same way someone can cry because of happiness. When I laugh at because someone is because of the sadness. So this is weak hydrogen bond, not peptide bond. Yes, because I highlighted it that these are the mistakes students do. Yes, okay, let's go. Manipulation of organism's gene to obtain the desirable characteristic. How do you call that? Manipulation of a gene to get a desirable characteristic. Whenever you talk about manipulating in science, whenever you say manipulating, what did I say? I said that is engineering. Manipulating. But you are manipulating genes. So, what are you manipulating? Genes. Therefore, genetic. Simple. Genetic engineering. Guys, I told you don't need to cram. You don't need to cram. You just understand life science. Tell the people to join the channel. If you want a distinction, Sanda a Duke. Yeah. Use the distinction material. There is no way how you can fail a paper. Never. Yes. Okay. Unless you don't want to. All right. Oh, unless you found me. Oh, you found the channel late. But if you want a distinction, tell the whole world. Thunder Duke. Okay. They're saying that representation showing the arrangement of diploid set of chromosomes. Hey. Representation. They are looking for how do we call that representation? Which shows the diploid set of chromosomes, meaning that chromosome number 1, chromosome number 2, chromosome number, but they are saying, see, diploid. Like that, we call it a karyotype. Karyotype. Done. Terminologies out of 7, 7. Okay, source of variation. How do you call random? We talked about this yesterday. Random mating, random fertilization. Source of variation for the species. I told you that a man produced over 500 million sperms. That is a rich man. That's why they tell you that you are 1 in a million. It means that your sperm was 1 in a million sperms which were released by your father and 2 there, and then only 1 you fertilize the ovum. So random fertilization is also random mating. You don't just find people come and say that, okay, now I want this one. You just go and then marry. You should just say, I want the small one, I want the big one, I want the light skin, I want the dark skin. You see, you're not doing it randomly. So you are minimizing variation. So variation, it must be random mating. When you are chasing this one and then another one comes into your eyes, you just go with that random mating, like a chicken. Yes. So this is both A and B. The answer is not A and B. The answer is both A and B. I explained it yesterday. Both A and B. Organisms that possess 2 factors which separate so that each gamete contain only one of these factors. Law of dominance. There is no domination here. Law of independent assortment. Organisms possess 2 factors that which separate that each gamete. Law of independent assortment. This is done in dihybrid cross. We call this law of segregation. Law of segregation, not principle of independence. So the answer here is none. Because this is a law of segregation by Mendel. You remember that guy Mendel discovered the law of segregation in he was there in 1865. Yes, Mendel, Georg Mendel, that priest. Yes, Georg Mendel. Yes. His life science is nice. So what you should do is be nice to it. Once you are nice to it, the subject is gonna be nice to you. If you are not nice to it, forget about. Okay, guys, just 1 second so that yes, Mr. Ramaphosa has given back electricity to us, so now we are happy. Yes, it's back. Yeah. All right. So if you see some disconnection a bit, just know that you're just switching the system. But we are continuing. Reproductive isolation. Reproductive isolation. Is my mouse. Okay. Reproductive isolating mechanism in species. You separate organisms through reproductive isolation, breeding in different seasons of the year. Correct. Infertile offspring. This is not correct. This is not correct, Baba. Look, same. This word, these people are confusing us, ne? Breeding in the same season of, no. It's supposed to be breeding at different seasons of the year. Let me show you what these people are trying to trick you up, ne? Let me just show you using the distinction material so that you see exactly how these people are trying to this is a real fake. They are faking us, so that we are off. We are sober. All right. Let me show you what they are showing you. Breeding, yeah, is here. You see, breeding at different times, not breeding at the same. So that answer is wrong. So this answer is wrong. You see, when you're saying the same, the answer is supposed to be breeding. This distinction material, there is no way you can go astray. Yes, so this is wrong. Infertile offspring. Correct. Check here in the book. Check here. Infertile of the way it is without even changing anything. Yes, infertile offspring. So you see, we say that mechanism of reproductive isolation. Now there are some people saying that I wish I knew this booklet. Sorry. Ne? Yeah. But you can refer to someone else so that you can get rewards. All right. So the answer here is B only. Yeah. Let's go to the next question. What does it say? They're saying that this is a phase. Ne? What I've seen in this paper, ne, as I'm going, ne, I don't know that much what is down, but what I've seen it here is they are using small words to take you off. If you don't read these questions very well, they're just using very small words just to take you off. All right, let's go. So we have to be keen, guys, when we are trying to answer. Ne? Yeah. So even may have to read too much so that I don't go astray. I might go astray. I give you a wrong answer. No, man. All right, let's go. They're saying that the diagram below represent a cell in early stage. You see? You see these people? The word early. That word, there is early and late. Late stage, mid stage, early stage. So now they're saying early stage. Okay. So if you look at this, which stage is this? This is not interphase. This is early prophase. Chromosomes which are homologous, they align together. Homologous. Yes, crossing over is not yet there, but they have aligned together. And you see that these dots shows that the nuclear membrane is starting to disappear. The nucleolus is already disappeared. You see? These people. So this is prophase. Prophase. Which prophase? Prophase 1. If you don't say prophase 1 is 0. Number of chromatids. So how many chromosomes do you have? 1, 2, 3, 4, 5, 6. So you have 6 chromosomes, but each chromosome has 2 chromatids. So 6 times 2, which is 12. So the answer here is going to be 12. Number of homologous pairs. They are saying number of homologous pairs. They're not saying number of chromosomes. Pairs. So means that 1 pair, 2 pairs, 3 pairs. 3 pairs. Someone is going to write 6. Ask you, do you have eyes? Yeah. Do you see 6 there? No. I see 3. Why did you write 6? I don't know. Okay, let's go to the next question. Identify what is R. Yeah. R is the nuclear membrane. Nuclear membrane. What is S? S is cell membrane. Membrane. Yeah, you know, I told you last time that you guys, you confuse nuclear membrane and the cell membrane. T. What is T? What is now T? T is nucleolus. Nucleoplasm, not in nucleolus. Nucleoplasm. So, the nuclear, okay, maybe they will slash it with the nucleus. Nucleus, yes. But based on this is nucleoplasm or nucleus. So if you give nucleoplasm, yeah, they might give you. If you give nucleus, they might give you. Based on what they have shown us. Ne? Yeah, don't say that. No, me, I wrote nucleus. Yeah, you might be right. Ne? Yeah, correct. All right, let's go. 2 organelles in animal where meiosis takes place. They're asking your parts. The most important structure on you. Which structure is that? Testes. Testes. That's 1. Oh, if you wrote it like this is also correct. Because this is singular, this is plural. Okay? I'm not saying that you write 2. No, just write 1. Hey, students. That's number 1. Number 2 is the ovary. So they give you a tick here, they give you a tick there. Simple. They're saying that you know, they write these funny words of these plants. Okay. Who knows this? I don't know. Let me just quote. The plant carries edible fruit. Scientists have been studying the inheritance of these 2 genes. One for the stem texture, and then another one is for the fruit shape. Okay, sharp. Let's go. They're saying stem can be okay, smooth. Smooth is represented by a capital letter, and then prinkly means that they have those yes, small letter, and then the shape round is capital letter, elongated is small letter. The type of cross, dihybrid. Dihybrid cross. They're saying state the dominant characteristic in the stem texture. Texture is here. Ne? Smooth. Answer here is smooth. Smooth. Boom. Done. This goal. Recessive. Recessive for what? Fruit shape. Fruit shape, fruit shape, fruit shape, fruit shape. Elongated. Elongated. Let's go. 1.5.3. Give there. The genotype. The genotype. It means that you are supposed to write in terms of gene. Prinkly, stem, and elongated. Prinkly is small error, so I can't be with the heterozygous. Small n, and then elongated. Elongated is small r. Listen carefully. Listen carefully. If you wrote NNRR is wrong, ask me what? Why? Because of this space. I explained it yesterday. Yes, a space must not be there because this is a genotype. These are not spams. It's a genotype. So it's supposed to be 1. Yes. They're saying that phenotype of plant with capital N is what? Because the small n would be masked. Capital N is smooth. And what? Capital R is what? Round. Smooth and round. Sharp. If you have the space there, is cool, but the genotype is supposed to be no space. Yeah, let me know if you have questions. If you have questions, if you have challenges, yes, I'll smash them. Yeah, I'll smash them. Yes. Okay, let's go. Let's continue to the next question. Which question? All right, they're saying that the table below shows the information selected on hominid fossils. Okay. All right, we are hearing. What are they saying? What are they telling us? They're saying that P. What is P now? P. Oh, the common name. Australopithecus sediba carabo. We say this is a transitional fossil. Missing link between Homo habilis and the. Okay. Who discovered the bagger? I don't know why they like carabo too much. They brought it in 2017. Yeah. It was the second or third objective. Yeah. Is read the bagger. Lee. Beggar. Yeah. Like that. This is Sediba, Carabo. Yeah, you can and you can find that someone called Carabo is feeding this Carabo. All right. They're saying that Taum child. Oh, Taum, if you're coming from Northwest. Yes. How do we call? Remember, I told you don't go the paper without knowing the 3 Australopithecus africana species. I said Taum child or Taum child, little foot, Mrs. Place. What are they looking for here? They're saying this is Australopithecus africanus. Cool. Just saying A africanus, they would have given a tick. Yes, because there is no Adipithecus here. So A africanus. But they have already given you the spelling here, so there is no stress there. They're saying that who discovered? Who discovered the town child? Raymond Dat. Yeah, it's Raymond Dat. Raymond. I don't know whether you call it Raymond or Raymond. Raymondi. Dat. Raymond Dat. Check that booklet, the distinction material. Check this distinction material. I organized it nicely for you guys. And even I created a video. Check. Australopithecus, Carabo, read the bagger, Malapa. And then they're looking for the 3 Australopithecus africanus species. Check bone. Raymond Dat. Taum. Sharp. Let's go. They're saying name the fossil. Name the fossil. P. We have said it. P is Carabo. Then they're saying identify species R. We said Australopithecus africanus. Let's go. They're saying that give the name of the scientist who discovered Q. We say that read the bagger. Yes. Well, you can also give that one brief Ilof. Yes, Ilof. Wheezy. Yeah, this one. So it depends on whom did you give. Yes. Then I think S, we say that it's Raymond that. Now, by this time, you have 50 marks out of 150. This one is enough for you to shift from a failure, meaning that you're above 30 now, 30%. However, it's not enough for my students like you. Like you. Guys, don't have too much pressure. Like, now I'm failing. That's life. Still, doesn't mean that if you fail this, then life has stopped. No. All right, let's continue. It just make you to prepare, and I pray for you guys so that you pass this, so that you get enough chelete. All right, let's go. They're saying that X, what is X? That is amino acid. I told you guys that you must always label these things before you start. Amino acid. Simple. Let's go. X, Y. What is Y? Y is anti-codon. Z, messenger RNA. Let's go. X, amino acid. Z, messenger RNA. Give the nitrogenous base of sequence, nitrogenous base sequence of DNA base triplet. Complementary to complementary DNA base triplet, complementary to mid codon of Z. Oh, they're saying DNA, meaning that we have to convert it backward. Let's say this is 3, this is 3, this is 3. So we are looking for this. So, but we are converting it backward. You have A, U, G. So we're going to be, say A goes with T, U goes with A, G goes with C. So this is the answer. That's what they are looking for, because they're saying mid in the middle one and for DNA. Then they're saying that give the nitrogenous base of Y. Where is Y now? Oh, the anticodon. So meaning that they want you to change this into transfer RNA, meaning that A goes with the, remember A transfer is an RNA. RNA doesn't have T. Therefore, we're gonna say U. U goes with A, and then G goes with the C. So the answer is this for this question and this for that question. Yes. Describe the process shown in the diagram above. Hey, what is this? This is translation. I told you guys. How many marks are this? Guys, don't tell me that you disappointed me. Unless you are not trusting me. Don't tell me that you disappointed me. There is someone who told me in the comment, I was so sad, but I didn't stop. He told me that you lost me in the first 5 minutes. I knew that this one is looking for attention. Yeah, because I talk too much. Okay, I talk too much, but I wanted you to understand the concept. Yes. Anyway, what do we mark? We can have different ways how we can mark. But this one you are describing, name and describe. The process is called translation. That's one mark for the word naming. And then according to the messenger RNA, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acid are joined together by the peptide bond to form a required protein. Simple. That's how I have been describing. But still in my book, the distinction material, I showed you exactly what you are supposed to do. Now the mouse is jamming. Okay, doesn't want me to this mouse doesn't want me to give you okay, let me use this. Okay, this mouse doesn't want me to give you answers, ne? Sorry. Yes, born it's there. Each transfer RNA is a marking point. Here is a marking point. This is a marking point. Each transferrin A carries a specific amino acid. We give you a tick there. When anticodon on the messenger matches the codon on transferrin A, we give you a tick. Specific amino acid, we give you a tick. Matches the anticodon. Yes. Then you are saying that this brings the required amino acid. Yes. Where does it go? To the ribosome. The amino acids are joined together or attached by peptide bond. Guys, we call them peptide bonds. We don't call them polypeptide bond. We call them peptide bond. I was very clear when I was explaining this. And then to form a required protein. That simple, short as that. But you can find a person writing a full page. So you're okay. That's short as that. That's what we wanted. Let's go. So this description thing, I'm not going to waste too much time because it is open thing, and you can find them in any book. Yes, you can find them in any book. Yes. Let's go to the next question. What are they saying? Just a moment. Yes. What are they saying? They're saying that the table below shows the codons. I explained this. Guys, take note of this because I explained it and I said this is one of the confusing subtopics. The table below shows the codons of codons that code for some amino acids. Okay, sharp. A mutation caused by DNA triplet to change this. Okay, so it means that we changed this to this. But they are saying this is DNA. But what is here is messenger. Meaning that to read this, I have to change it first. So I'm going to say that, okay, this was C. C goes with G. T goes with A. And then G goes with C. But changed it to C goes with G. T goes with A. And A goes with U. Because this is messenger RNA. Wabona? Yes. Now I come. I go to, you go to what? I go to this table. I look for it. GSC. 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 GSC is aspartic acid. What about this? Does it correspond to a different amino acid or the same amino acid? Let's go. And then you're saying that GAU, aspartic acid again. Wabona? Ntuli. I can call you Ntuli. Yes? Isn't it? I'm just saying Ntuli because Ntuli called me and said that. Thank you very much, sir, for teaching me. Yeah, I think I managed to pass. I pray for you guys, so that you pass. Sometimes when you're marking you're marking your students, and then you get something, you think of something, you end up giving a wrong one correct. You never know. Make sure that you pray until the results come back. Yes. Anyway, so since they code for the same amino acid, yes, so it means that the protein will not change. But these are 4 marks. Where am I going to get the 4 marks? You're gonna say that this change of this, yes, this changed into this. You writing it GSC changed to GAU. Those are marks. Those are 2 marks. This changed to this. Why add? Why this one is a mark and this is a mark? Is because you changed it. You see, you changed this. That's 1 mark, and you changed this. Those are 2 marks. And then this code for the same. This code is for aspartic acid as well as this code for aspartic acid. Meaning that these two, they code for the same amino acid. Is another tick. Those are 3 marks. Then the 3rd mark, the 4th mark will say that this will not change the protein formed, or it will not change the functioning of the protein formed. Simple. Why? Because it's like calling you with the first name, calling you with the second name. You are the same person. Yes. Or sleeping on your bed when you are straight and sleeping on your bed when you have carved somewhere, or does not mean that you're a different person. It's the same person. So they're just saying that, yes, however much the mutation occurred, but the mutation did not change the amino acid. Simple. This question, they have brought it back again. Tabulate two difference. I emphasized this question of mutation, and I told you that I know what students find when it comes to this question of mutation. Anyway, this question. Tabulate two differences between DNA nucleotide and RNA. I explained it before. Yes, those people who were on my channel yesterday. They are they are not asking you the differences between DNA and RNA. No. They are saying DNA nucleotide and RNA nucleotide. Clear? So there are two differences. The first one, DNA nucleotide, has thymine, and the RNA nucleotide has uracil. The second one, DNA is made up of, nucleotide is made up of deoxyribose sugar, and RNA nucleotide is made up of ribose sugar. There is no any other difference. They are looking for the nucleotide. They are not looking for DNA. They are not looking for they are not looking for just those nucleotide for the RNA and for DNA. That's all. So if you wrote a lot of things there, but there are 5 marks. Where is the fourth mark? Fourth mark is for the table. Just drawing a table and say DNA nucleotide, nucleotide. Even if you didn't write anything here, you draw a table, a complete table, you will get a mark. At least you are not getting 0. Yeah, at least. Yes. Let's go. Down syndrome. I talked about it also. Down syndrome. Okay, let's go to Down syndrome. What are they saying about Down syndrome? Okay, let's go. Down syndrome is the result of individual having extra chromosome on chromosome number 21. Okay. The genetic variation that can be caused by Down syndrome are, okay, trisomy 21, all somatic cells are individual, 3 copies of, okay, chromosome number, all. This word is a key point. And then they're saying that mosaic means something which is moving. Yeah, okay. Individuals have only some cells, not all. So this is a key point versus this extra chromosome, which is caused by abnormal process of cell division after. This is after. Is caused by after fertilization, but what occurs during gamete formation. This is during gamete formation. This is after fertilization. You see? Yeah. That's the major difference. The type of mutation lead to the, trisomy 21. The type of mutation that leads. You can say chromosome aberration. Those who wrote chromosome aberration, correct? And then chromosomal mutation. Because these are chromosomes. Chromosomal mutation. Yes. Or aberration, correct. Abnormal process during gamete production that lead to 3 copies of abnormal process that lead, name the abnormal process that lead to the 3 copies of non-disjunction. The type of cell division that occurs after fertilization, it is mitosis. They're saying describe how the process mentioned above, which above B here, non-disjunction lead to trisomy 21 is 5 marks. This is a general question. Yes, go back to my distinction material so that we don't waste too much time there. Yes, I talked about it, but sometimes people they have challenge. But before I left meiosis, I said that please don't go without knowing this process, because this process they will ask you. Here is a question. Non-disjunction, failure of the chromosome to separate is they are looking for this, you see, this page. Each nucleus has, it contains 23 chromosomes after meiosis, but non-dissociation when occurs, okay. One nucleus will contain 22, while the other will contain 24. Yes, either of the resulting gamete will fuse with the normal gamete, but they said, yes, with the normal gamete. They said, yes, they said what will happen. Yes, so the normal gamete is 23, then the resulting, the 20, if the 24 fuses with 23, it's going to be 47. The 22 fuses with the 23, it's going to be 45. Yes, so in this case they are looking for 23 because they're saying trisomy 21. It means that the 21, it has an extra chromosome, not short of a chromosome. Yes, let me explain to you. Yes, and then they are saying that, let me show you what they are trying to say here. Yes, and remember, during non-disjunction, this is a cell, né? Under metaphase. Yes, so this is what happens. So during non-disjunction, the whole set is going to go to one side, so meaning that I'm going to have 2. So the normal one is going to be 2 cells. I'm going to have 2 here and 2 here. But during non-disjunction, the whole of it is pulled to one side. And then this one, yes, it will go to one side. And then this one, another one will go to one side. Meaning that here, yes, here I will have 1, 2, 1, 2, plus this one, 3. And then this side, I'm going to have only 1. So this one has extra chromosome, 2, 3 instead of 2. And then here it has short. So they are looking for this because they are saying, yes, if it occurs on chromosome number 21, it becomes trisomy 21. Tri means 3, 1, 2, 3. You understand? So now when this one fertilizes a normal ovum, then it's going to have, now is the cell is supposed to have 23 chromosome. But this one is not going to have 23 chromosome. It's going to have 24. So if this fertilizes a normal ovum, which is 23, it's going to result in 47 instead of 46. So that's what they are saying here. So go to the distinction material. Yes, get there. So that's what they are trying to explain, or they're asking you here in this question. Let's go to the next question so that, yes, this describe 2 differences between trisomy 21 and the mosaic Down syndrome. Yeah, I think I talked about it. Trisomy 21, all cell, check, all somatic cells in the individual have 3 copies of trisomy of chromosome 21, you see, all. But here, yes, make sure you draw a table, né? I think they don't want a table. They're not saying tabulate. While here in mosaic, the individual has only some cells with extra chromosome 21, but here all the somatic cells. Then number 2, the process, when does it occur? It occurs during gamete formation, while here it occurs during or after fertilization. Those are the 2 differences they are looking for. It's just as something from there which you can use to get the answer. All right, I see ourselves trying to finish soon, eh? Yeah, so we are going to the question number 3. Question number 3. How is the paper? Let me know. How is the paper? Let's go to the next question. Ne? Yeah, I think the paper was simple. Ne? The paper was simple for those who read. Yeah, let me say like that. And I think I should come back and I teach you how to read. Does not stop only here, even when you go to the university. Good. All right. The diagram below shows the inheritance of tithe. Oh, they like this funny name. Anyway, I'm not going to remove my teeth because of these names. Okay. Is a rare disease which lead to this destruction of the neuron. It is inherited as Autosomal. Okay. Autosomal means that it occurs in the body cells, meaning that it's non-sex linked. It's occurring in these other cells, these other chromosomes, not on chromosome number 23. Yes. Okay. TT. So meaning that you must not use X and Y. This is not sex linked. It's autosomal disorder. Anyway, let's go. Describe what is meant by is a disorder which is caused by a gene carried on non-sex chromosomes. Yes, non-sex chromosomes, meaning that from chromosome pair number 1 to chromosome pair number 22, it must not be sex. Yes. How many sons do individual 1 and 2 have? 1 and 2. Yes, sons. Son is a box. It's 1, is 2. But this son is not belonging to these two. You understand? This guy, yes, just married the daughter of these two guys, meaning that the answer is 1. Understand? You see? Yes, this is just an intruder who came to marry the daughter. Yeah, the beautiful daughter of 1 and 2. Yes. So the answer here is 1. Using the individual 3, 4, 5, explain why it can be concluded that this disorder is controlled by a recessive allele. 1 is 3, 4, 7. 3, 4, 7. Okay, let me clear up here so that at least yes. Okay, let's see. 3. Okay, this is the affected individual, né? And these two people who are, if they produce a person who is affected, it means that these two people were heterozygous, meaning that it is capital T, small t, capital T, small t. You see? Since they carried that what? Since they carried that gene, this gene, recessive allele, and then this recessive allele and this recessive allele gave rise to this. Meaning that, what is the question? What are they saying? Which question is this? 2.5.3. They're saying that it means that individual 3 and individual 4, they are both normal individuals. I see. That's the first mark. But they gave rise to individual 7 who is affected with the disease. You have to tell us the disease, né? A female who is affected with the disease. Yes, that's the second mark. This means that individual 3 and 4, they were heterozygous. Meaning that they are heterozygous because they are both normal, but they gave someone who is sick. Meaning that they were heterozygous, they were carriers. That's another mark. And then they pass their recessive allele. 1 recessive allele from 3 and 1 recessive allele from 4 to individual 7. That's another mark. You're able to get all the 5 marks. 1 recessive allele from 3, 1 mark. 1 recessive allele from 4, another mark. Yes, to form this one who is affected with the disease. So, yeah, it is an application question, whereby you have to apply and explain exactly what happened to inform individual 7. Then they are saying that individual 1 and 2 can produce children with 3 possible genotypes. Okay, list those 3. Individual 1 and 2 can produce. Yes, individual 1. 1 and 2 is the same thing. 3 and 4, they are both heterozygous. Okay, they can produce. If they are both heterozygous, it means that, look, it means that this is capital T, small t, heterozygous, capital T, small t. So it means that when they produce, they're going to produce capital T, capital T. This is what they are trying to show you. Capital T, small t. This is capital T, small t. And then small t, small t. You check these two. They are both heterozygous. This is homozygous and this is a homozygous. Let's go. The question. They're saying that list all the possible genotypes that have 25% chance of offspring. Then this is 50 and this is 25. This is 25. Meaning that I'm going to come here and say capital T, small t, small t. A comma. That's all. That's what they're looking for. They give you ticks. In human, hemophilia is caused by a recessive allele of X. Of X. Anyway, it's caused by a recessive allele of X. Let's go. Of X. They're saying that in humans, hemophilia is caused by a recessive allele of X. Okay. This is carried on X chromosome, meaning that this is sex-linked. The moment you say that that's sex-linked, hemophilia, yes, we know is a sex-linked disorder. A woman who is heterozygous for hemophilia marries a man who is heterozygous for hemophilia. That's a woman. It means that it's like this, capital H, small h. Yes, marries a man who is hemophilic. Yes, meaning that the man is definitely is like that. Is use the genetic cross to show the percentage chance of couple having a daughter who is homozygous for normal. Homozygous for normal and is a daughter, meaning that this person is gonna be like this. There's no way. Check, the father is not normal. Then it means that the answer is gonna be 0. 0. Yes, but they are saying use the genetic cross to show the percentage chance. The percentage chance is 0% because there is no way. But they are saying use the genetic cross to show. So you're saying 0% is 1 mark, but you have to show. Don't forget to show all the steps in a genetic cross. Let me see, is it P1? Let me see. A man who is, they didn't talk about anything. So I'm going to come here and say P1, F1. P1, parent, phenotype. You go to the question, you'll get the phenotypes there. I told you that this one is simple, and a man who is heterozygous, a woman who is heterozygous, meaning that this is a normal woman. Yes, marries a man who is hemophilic and a man who is affected. Then you go to genotype. Genotype, I'm just rushing so that a lady was like this because they say that heterozygous, marrying a man who is like this. Yalla, let's go. They're saying like that. Don't forget that these are gametes. I always tell you that they are not at the beach, so they must be circled. If you didn't circle, it's still fine. Don't stress. Give you a tick. These are gametes. Wabona. Yeah, but how do you call this process? Meiosis. You saying P1, F1 will give you a tick. So that's the second tick. 0% is a tick. But it's the second tick. This is a third tick because we got it. So you have 4 marks. Mahala. The question is out of how many? 7. So we are searching for the 3. This is the next tick. It's the next tick. Then after that, you can draw your Punnett square here, or you can just cross. When you cross, you'll find out that this, look critically. This goes with this. So it's going to be X, capital H, X, small h. Then this goes with this, capital H, small, capital H, yes. And then this with this, small h. This with this, small h, and then y. Is there any probability of having a child who is homozygous normal and is a female? No. There is no way. Therefore, this is offsprings. F1 offsprings. And then you remember you have to cross, show cross or the Punnett square. So, and this process is called fertilization. So we give you another mark for this. But what I know that the marks are more than what we stipulate. But you saying that therefore the percentage chance is 0%. This is going to be a compulsory mark because the question is emphasizing that percentage chance. They want a percentage chance. Do you understand? Yes. So it's 0%. Having a child who is normal, homozygous, and is a female. Yeah. Let's go to the next question. I think I'm clear. Yes. Let's go to the next question. These papers are simple if you read. If you don't read, yeah, they become sour. But I don't expect you to. Let's go to the next question. The next question. Question number 3. It means that now you have 100 marks. 100 marks. See now, you are approaching slowly a distinction. You're approaching a distinction. I know some of you just made small mistakes, which can be acceptable, but there are some mistakes which cannot be accepted. So, yeah. Anyway, let's go. Yeah, let's go. This is 2023. Yeah, I know some of you gonna be successful. I pray for you so that you become successful. Anyway, learners conducted an investigation to determine the blood group was the most common in the community. I talked about a pie chart yesterday. Yeah, they conducted information about, 200. It's a big number. It means that the sample size. Take note of that. In each of the 3 types of blood group. Okay. Yalla. Let's go. They're saying that in their community, they did not include the first donor in the investigation. Why? Comparison of results. Okay. Yeah. The pie chart below shows. All right. Let's go. The percentage of each blood group in the sample. I know you read all these questions. You know them. So I'm not going through too much. State the aim of the investigation. Ooh. The aim must start with the word to and is always put in the first statement or second statement. That's why always I tell my students. If you can't find it, then you have to figure it out. Learners conducted an investigation to. That is the aim. From to, the investigation. The aim is to determine which blood group was most common in their community. Yes, in the learners' community. Conduct an investigation. Yes. So the aim is done. Answer the following question. State 3 planning. We always ask you this, but you go back and read the question and see the possibilities for you. What are you going to plan? What are you going to do? So, but I need these people who are going to be giving me blood. So you have to organize them. Yes, get those 200 people. Yes, you are not just going to just get them. Yes, you have to get permission because this is a community thing. Get permission where you're going to do this or the clinic, or try to go inform them so that they know and they accept. You are not going to force them. Get permission so that they can do that. You are not going to just collect the blood just like that. Get the necessary equipment. Planning before you do the activity. Yeah, that's what they are looking at. Yes, something like that. State one way in which learners ensure. Yesterday I told you the word language and I emphasized it. If they ask you, don't ask you, how did this person ensure, how did the person do increase reliability? Or how do you ensure reliability? Now they say that, how did the learner ensured? That is a past tense. Your answer must be in a past tense. The learner ensured reliability by using to. Remember, reliability is to increase the sample size by using a big sample size by using 200 people. 200 people. You see? So, this learner used the 200 people. Sharp. So, that is done. Give the reason why give one reason why they did not include the, yes, we said comparing the result. Calculate the percentage, the number of participants that had blood group B. Show your working. Blood group B. They use 200, but only 15% blood group B. So you just come here and say 15 out of 100 times 200. So the answer you get is the answer, around 30%. Done. They give you your 3 marks. Give you a mark, give you a mark, give you a tick. Done. They're saying that name the blood group that has only recessive allele. Only recessive allele, blood group O. Remember, it is recessive allele. Remember? Yes, I said it yesterday. Results of codominance is AB. I said it yesterday. Remember, it's A and B. Give the possible genotype of the blood group represented by 25%. 25%. Yes, 25% is A. But they are saying the possible genotypes. If you remember, A is either homozygous. Go to the distinction material. You will see what I was saying yesterday. A heterozygous. I explained this yesterday. I explained it very well. Very well. Yes. I hope you guys did not disappoint me. Ne? I don't want the marks to come back and then I'm disappointed. Say, oh, why did I do this for my learners, these learners of mine? Ne? No. I don't want that. Ne? I want everyone to pass. Ne? Yeah. I want everyone. Everybody to pass. Anyway, let's go. Question 3.2. We are almost done. We are almost done. We are almost done. Anyway, question 3.2. These are dogs, man. Do you like dogs? Let me know the question in the comment below. Do you like dogs? Do you have a dog at home? Sometimes they call you a dog. And a dog. Okay. Dogs have been selected and bred over many years to produce approximately 340 different dogs breeds. They have been bred for certain characteristics desirable to humans. Saying that analysis of 786 okay, gray wolves, only direct ancestor, present day dogs bred. Okay, all the dogs belong to, I'm just reading so that I get the idea, ne? But you know, you can read it for yourself. Okay, I've got the idea, ne? Very fast. They're saying that name the process whereby the different breeds of dogs were produced. Artificial selection. Yes, these are the things we are talking about here. Where am I? Yes, artificial selection. Is it? Yes. Where is it? Okay, this is meiosis, ne? Yeah, artificial selection is they are looking for you. These dogs, even this question has these dogs we are talking about. Exactly the same dogs. Don't disappoint me. Don't disappoint me. Don't disappoint me, guys. Dogs, wolves. They're there. Born. Check. They are dogs, the wolves. That's what are the wolves we are talking about. Okay, they're saying that. Name the artist. What did we say? Is what? Artificial selection. Human, they select desirable traits. You see? That's what they said here. Desirable. They're desirable to humans. Yeah. Yes. Distinction material. Get it. Yeah. Get it. I'm talking about those who, your sisters, your brothers. Yes. Anyway, let's go. And saying that, name the process whereby different breed of dogs were produced. Artificial selection. Describe how human carried out the process named above. Meaning that they got the characteristics they wanted. They let that individual which has that characteristic to mate with the other characteristics they want, and then the other one which doesn't have the characteristics were not allowed to give to have sex, meaning that to pass their genes to the next generation. So after many generations, they had to be with that desirable characteristic. That's what they are saying. So if you look at this example, check, it was about milk. Choose or select the cow in your herd, or now they're talking about the dogs to produce which has that characteristic. Yes. Then they are saying that let only those dogs to reproduce, the one which have the specific characteristics, then select the offspring that produce that characteristic most. Yes, let those only those offspring reproduce, allow them to have sex and have offspring like that. Keep repeating this process of selection and then the breeding until you achieve your goal. That's it, to get the desirable dog you want. Explain how it can be confirmed that all the dogs breeds belong to the same species. Same species. It means that they can interbreed to produce a fertile offspring. If they are not producing fertile offspring, then they don't belong to the same species. Yes. They must produce to produce a fertile offspring. Hence, this answer becomes right. Yes. Explain how present-day dogs may have been disadvantaged, yes, in the relation to their common ancestor. This common ancestor wolf, Baba, it could resist diseases. Now, you have to take it to the how do they call it, the veterinary doctor. They were staying in the wild, so these dogs are susceptible to diseases. Less chance of these dogs being adapted to the environment. Yes, these dogs, the one which are new, yes, have less chance. This one, it could adapt, Baba. Yes, meaning that some genes are being lost as you are doing this selection. Yes, so the dogs are being disadvantaged. Yes, let's go to the last question. Describe the process of speciation by geographical geographical barrier. Guys, I told you don't go to the paper. Speciation is here. This is what they're looking exactly. This formation of new species. Exactly. There is a population of a particular species. A population of a particular species becomes split into two by a geographical barrier. As a result of, you don't talk about geographical barrier. Okay, you can say river, mountain, or whatever, but geographical barrier. As a result of two parts of the population cannot interbreed, there is no gene flow, yes, between the two. The natural selection will occur independently, yes, due to different environmental conditions. As a result, the population became genotypical. You start with genotypically, then phenotypically different over time. Even if you remove you bring these two populations, you mix them together, meaning that you remove the barrier and you let them mix and they interbreed, they cannot produce a fertile offspring. They cannot produce a fertile offspring. Therefore, one or both population have become new species. This is exactly without changing any word or what. That's what you're supposed to write. That's what you are supposed to write. Someone is gonna tell me that what if I used a scenario? That's not KO because they didn't give you a scenario. KO is when the examiner sees, just give you a tick, not arguing with him or herself. Yeah, should I give no? Something like that. Don't like it. Just when examiner sees your paper, is marking it as you are. You know, when you're marking someone, you see that someone is getting all the ticks. Yeah. You feel good, Baba. You feel good. You feel good. The same way you feel good when your marks are coming back and you have distinction. How do you feel? You feel like you are not fitting on the when marks are nice. All right, let's go, man. You feel good the same way I'm feeling good when I'm teaching you. Yeah, I feel good, ne? I feel good. All right, let's go. They're saying that. Name the type of diagram represented above. Okay, the diagram will represent the evolution of the family Hominidae. Okay, sharp. They're saying that. Name the diagram. I showed you these things. Phylogenetic tree, and I showed you the questions you're supposed to answer, and I don't think that they skipped those questions here. Let's go. Name the diagram shown above. Phylogenetic tree. Done. Give the letter of organisms, which is the common ancestor for all the hominids. P, because all of them, they come from here. Is P. They're saying that. They're saying that. What is common ancestor? Okay, share most recent common ancestor with the gorilla. Gorilla is here. Gorilla is there, but they share a common ancestor. What shares the common ancestor is here, meaning that is this. But what shared is R, because gorilla and R, they share a common ancestor. So the answer here is gonna be R. Give two organisms that have S as an ancestor. Two organisms that have S. Where's S now? S is this. Is this point. Stop it. Stop it when technology. Okay, this is S. So meaning that you're gonna say this and this. Those are the two individuals. Let's go. Name two organisms that have S as a common ancestor. We have said it. Quadrupedal. These ones. Gorillas. You know them. Is chimpanzee quadrupedal? I think so sometimes. But these ones, they are quadrupedals. Okay? They're saying that describe three anatomical features of the skeleton of a quadrupedal organisms. Skeleton, when you talk about a skeleton, you're gonna talk about the spine, the cranium, because that's a skeleton, the spine, the cranium, and then the pelvic girdle. Yes, I showed you here when talking about. For the case of the skeleton, here, you see, it's a skeleton they're talking about. They're saying about quadrupedal organisms. Foramen magnum is in forward position. Yes, it's a skeleton. Yeah, that. Yes. You talk about the spine is C-shaped, and then long and narrow pelvis. That's what they are looking for. Simple. Simple. As simple as eating ice cream. No bones. Yeah, let's go. Second last. Yeah. Fossil evidence is used to support out of Africa hypothesis. State out of Africa hypothesis. Modern humans originated from Africa and migrated the rest of the world. I told you when I was concluding, don't go to the paper without knowing this. Modern humans originated from Africa and migrated the rest of the world. And I showed you this. That take note of this. Unless you do not want to trust me. The modern humans originated from Africa and migrated the rest of the world. That's what they want. The word modern must be there. Modern. Don't just say humans migrated the rest from Africa. And Homo habilis, Homo naledi, Homo neanderthalensis, all those are humans. But the modern ones, Homo sapiens. Yes, they're looking for. Okay, let's go. Last question. Describe how the fossil evidence is used to support out of Africa hypothesis. How is this fossil evidence? How is this fossil evidence used to show out of Africa hypothesis, guys? How is it used? Describe how this fossil evidence is used to support out of Africa hypothesis. You guys, I told you this and then you didn't write. Okay. You talk about all the Adipithecus, Australopithecus, Homo habilis. You say that only, the word only must be there. Only. Adipithecus, fossils of Adipithecus, fossils of Australopithecus. You can talk about the examples. Fossils of Homo habilis. Only found in Africa. Then when you go to Homo erectus, you talk about the oldest fossil of Homo erectus, distinction material, and the Homo sapien. Homo erectus and Homo sapiens. Here you talk about oldest fossil of Homo erectus and Homo sapiens were found in Africa. Don't say that only found in Africa. Nope. You have to say oldest. The key point there is oldest. That's why I put it in bold italic and I was supposed to underline it so that your eyes can open when you are reading it. I think, guys, the paper was easy. Yeah. Don't forget to subscribe, like, and share. It has been M.Saidi, has been taking you all over this paper. I think, yeah, the paper is a little bit easy. It was a little bit easy. Okay, they had some challenging questions, but it was not that bad. It was not that bad. Yes. Thank you very much for being with us. Yeah. Thank you very much, guys. It has been a wonderful paper. Don't you think so? Let me know in the comment below to know how was this paper to you, based on you. How was the paper? Yeah, let me know in the question in the comment. Thank you for being with me from the beginning until guys, ne? Yeah, I just love you so much. Stay blessed, ne? Stay blessed, ne? Yeah. Maybe I just a small note that even physical science, yeah, even other papers we will be doing discussing them immediately after you have done them. Thank you very much, ne? See you.
A pedigree. Remember, in this pedigree, you have different styles. A question can come when this pedigree is sex linked. It can come when this pedigree is non-sex linked. So you have to take note of the following before you answer any question concerning about a pedigree. It can be sex linked, it can be non-sex linked, it can be caused by a dominant allele, or it can be caused by a recessive allele. So when you are trying to answer these questions, you have to first know this pedigree is caused by what? Is it a recessive allele? Is it a non-recessive allele, meaning that it's a dominant allele? Because if it is a dominant allele, then definitely you have to use the capital letters. If it is a recessive allele, then it means that you're supposed to use small letters. So please take note of that. Then, if it is saying that, if the question comes and is saying that it is sex linked, if it is not saying that sex linked, please don't use X and Y. So we're going to look at these questions. We have different kinds of questions we're going to look at, so that at least you don't suffer with this. Kundai is saying that I received a distinction. Thank you very much for receiving a distinction. And I know that at the end of the year, you will receive distinctions. I always say that distinction times 2. All right. The first question is saying that these are national examination questions, like obtained from the past papers. Please always and always use these past papers so that they can guide you. All right, the first question is saying that moya is a disorder caused by a dominant allele. Moya is a disorder caused by a dominant allele, capital R. You see, the question is stating it that it is caused by a dominant allele. This disorder damages the arteries supplying the blood to the brain. Okay. So that is what is important here in this statement. We don't want to know supplying the blood. We don't want to know the condition. What is important is caused by a dominant allele. This is a key point. As I say that it can be caused by a recessive allele. It can be caused by a dominant allele. It can be sex linked, or it can be a non-sex linked, meaning that it is an autosomal character. So please take note of that. So in this case, first of all, it is caused by a dominant allele, and they are saying that they are not specifying anything here. They are not saying anything here that it is caused by sex linked. However much I've talked about male or female, I should not use X and Y. So let's go. The first question is saying that how many generations are represented in the diagram? When you talk about generations here, You only have to look at this is the first generation, this is the second generation, this is the third generation. So in other words, there are three generations which are indicated in this diagram. So the answer here should be 3. And then they're saying that give the letter, sometimes letters of an affected male. Now, sometimes we give you a key, which means you have to take note of it and then know that whether it is caused by a dominant, sorry, is caused by a dominant allele, recessive allele, but we don't give you a key to show that this is a male, is affected, this is a female, is not affected. So we just give you the question. So please take note of that. So in this case, they gave you a key. So the one who is shaded is affected with the disorder. The one who is not shaded is not affected. So it means that you have to take note of that. So in this case, we are saying that affected male, a box. We say that a box means a male, and then a circle means a female. So if they're saying that an affected male, so here they're saying a box which is not shaded is unaffected. So they are saying that, so I come here and I find out it's only one person who is unaffected. So the answer is saying is supposed to be H. I think that's why it is also 1 mark. Then they are saying that genotype of individual A. Where is genotype of individual A? So because this is someone who is affected, yes, and this is not, they have not said that this is a sex linked. And then number 2, they produced a kid who is a female, and this kid is not affected. So what does it mean? It means that if this kid is not affected, it means that this person is recessive, small r. And then this one is not affected because this is caused by a dominant allele. So it can either be capital R, small r, or capital R, this individual A. So, but because this is small r, yes, and then they produce a kid who is small r, yes, it means that one allele came from the mother. Obvious, mother is like that, as we have seen it here, and one allele came from the father. Therefore, the father can't be this. So the father is like that, meaning that one allele came from the father, one allele came from the mother, and they gave rise to this kid. Hence, A is of this genotype. So it is A is heterozygous. I think I'm clear there. Yes. Then the next question, they are saying that genotype of A, we are done with that. It is capital R, small r. Give the letter or letters. Can you please start? I just logged in. Yes, we are going on. Yes, so if you have just missed it out, yeah, don't forget that we have a lot of questions down there. So try to keep up, and then you'll get there. So we are saying that give the letters of what? Of let me check here. Someone is disturbing me. All right. Give the letters of individual who is not biological uh, uh, kids for A and B. Where is A and B? A and B. A and B. So in this case, this line means they gave rise to these two kids. This person C, and this person F. It shows that they just got married. You see, they just got married. So F and C are not biological children for this. So it means that the answer here is going to be F, yes, this F and C, because this, if this line was like this, then they would have been this the kids of these parents. But because this line is not like that, it means that, what does it mean? It means that they just got married. So this is not a biological child for these two, and also this is not a biological child for these two. So I think I'm clear there. Then they are saying that uh, they're saying that sorry, they're saying that brown enamel. This is another question. It's another question. Brown enamel of teeth is sex. Now here, this question, they have specified that this is a sex-linked. So in the question, if they don't specify that this is a sex-linked disorder, please do not talk about X and Y. However, you see now here, we have not talked about X and Y. So please find out whether it is caused by recessive allele, dominant allele, sex-linked, autosomes, meaning that non-sex linked. So there is another question here. This question is about pedigree again. But this pedigree, what are they saying? One type of defect in human is carried on single allele. Okay. The diagram shows the inheritance of deafness in family. So they are not specific, that they are not saying that this is caused by is sex linked. No, they are not saying that. So if it is not showing you that is caused by sex linked, then automatically I have to keep it the way it is. Yeah, it means that you are not supposed to 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 check to use X and Y. Yes, you are not supposed to use X and Y. Just a moment. Okay, it's locked. All right. They are saying that how many generations? We have seen this question before. How many generations is one generation? This is the first generation. This is the second generation, and this is the third generation. So this pedigree diagram showing 3 generations. Children of Paul. How many children of Paul and Liz? This is Paul, this is Paul, and this is Liz. So the answer is gonna be 1, 2, 3. Yes, this is not a child of Paul and Liz. Why? Because this is just married. This means interbreeding or sex is happening, or marriage or cross, whatever you can describe it. And then yes, so it means that there are 3 individuals. So also it is 3. Then they're saying which phenotype is dominant. Which phenotype is dominant? Based on this, you have deafness, and then a person who can hear, deaf and hearing. You have to, since they didn't show you that this is dominant, this is recessive, you have to look at maybe 2 parents and then their kids, and then you see which one leads to the other. So if you look at this and this, this will not show you because we don't know whether it's heterozygous, or even this one will not show you, but this one can show you. This is showing this person is hearing, this person is hearing. But they produced a kid who is deaf. What does it mean? It means that these 2, they had a recessive allele for deafness. It means that deafness is caused by recessive allele. Therefore, hearing is caused by a dominant allele. So, which phenotype? So the answer here must be hearing. Hearing. Yes. And then use the offsprings of Bob and Anna. Oh, Bob and Anna. So it's the same thing we have used. We use the same example. Why are you saying that hearing is the dominant allele? As I said that Bob and Anna, yes, use Bob and Anna to explain your answer. Bob and Anna, yes, look, we are seeing that Bob and Anna, both of them, if this is Bob and this is Anna or Anne, whatever, yes, if these two are normal and they intermarried or interbreed and they produced this kid, and this boy who is deaf. What does it mean? It means that this person, if it is small r, they are recessive. It means that Bob had is capital R, small r, even this one is capital R, small r, and then this came from the mother, this came from the father, and then they joined, and then they formed this kid who is, let me show you, a kid who is having the disease. It means that this allele, and this allele, they came here. Yes. So it means that, so use Bob and Anna and explain your answer. So you can say that Bob, who is a male and is hearing, yes, married, yes, Anna, who is female and is hearing. Did they say it's caused by sex linked? No. So that's why you're not saying anything with X and Y. So this Bob, who is hearing, meaning that Bob is capital R, small r, is heterozygous. In other words, heterozygous. Don't use small r, because they didn't give you the letters to use. Bob is heterozygous, and also Anna is heterozygous. They passed their recessive allele. Even Anna is like this. They passed their recessive alleles to one kid, and the kid became deaf, which is small r, small r. So that is the reason why Bob and Anna produced a kid who is having this defect of deafness. I think I'm clear. So sometimes they try to change it, tilt it, as long as you know the principle. How do you identify? Please look at the babies. Ne? If you see the babies, you'll see them that if one person is sick or is affected by the condition, it means that the parents are heterozygous. You understand? If the two parents are normal, it means that the two parents are heterozygous. But if all of them are normal, then it automatically shows that the parents are homozygous, dominant. Use letters A to represent a dominant allele and small a recessive allele. Show recessive allele to give all the possible genotypes for hearing individual. Possible. Use capital dominant allele and letter A for recessive allele to give all the possible genotypes for people who are hearing. It's 2 marks. Remember, we said it here. A person who is hearing, yes, and remember, hearing is dominant. If it is capital R, say this, capital A is for hearing, ne? And then small a is for deaf. Yes, it means that a person who is hearing can be homozygous, like this, homozygous dominant, or can be heterozygous. This person is hearing and also this person is hearing. So these are the two options you can give. So we will give you a tick here and also give you a tick there. The last questions. I have this question and then I have another question and another question. So yeah, basically I have 1, 2, 3 questions to smash. Okay, let's smash these questions together. All right. They're saying that this question is now pedigree, but now this pedigree is sex linked. Is sex linked. You remember the question we saw? It was not pedigree, but it was sex linked. But now this one is pedigree and is sex linked. So we are bringing sex linked character in the pedigree. This syndrome, however, I don't know how you're gonna, whether it's hotty or gotty or whatever you read it, because it's an scientific term, but I know that many students read it as hot. Okay, is sex-linked genetic disorder? It is caused by a dominant allele. You see, it's caused by a dominant allele on X chromosome. This shows that this is a sex. I told you that you cannot use X and Y unless it's mentioned in the question. Yes. Or they're gonna ask you, is it sex linked or non-sex linked? The diagram below shows the inheritance of this syndrome in the family. Okay. Name the type of diagram represented above. We have been talking about it. Is a pedigree. Is a pedigree. Pedigree diagram. This is RNA. How many female are in this family? How many female are in this family? Remember, circle means a female. Yes, it's 1, it's 2, it's 3. What about this? This one is not a member. It just came to get married. Get married. Ne? But they're saying how many female? Okay. It's 1, it's 2. Since now they are going down, they're saying females, not kids. Yeah. Okay. It's 1, it's 2, it's 3, it's 4, it's 5, it's 6. Yeah. Yeah. They're not saying kids. They're saying all the females. So there are 6 females. Males in F1 generation. F1 generation. They're saying F1 generation. How many males in the first generation? So remember, you have first generation, second generation, then third generation. So how many generations are these? There are 3. So they're saying how many males in the generation? So it's only 1, the father. Yes. Then they're saying that the next question, they are saying that give Gabby's genotype. Who is Gabby? Ne? Who is Gabby? Gabby's genotype. Where is Gabby now? Oh, Gabby. Is it caused by a dominant allele? Yes. It's caused by a dominant allele. So if it is caused by a dominant allele, so what does it mean? It means that when you are writing it, yes, you're supposed to to shaded means affected with a G, because whoever is where is Gabby? Yes, this person is affected with this disorder and is caused by the. So it means that you have 2 options. Either you have Gabby is a female, so it can be capital G, small g, or it can be capital G, capital G, capital G. Yes. So, but the question is saying they give Gabby's genotype. So, which of the 2 is correct? Now you look at the babies. Is there any baby who is normal without the condition? Yes. Is this. And remember, this is the recessive allele. So it means that this is a male with small g, capital Y. And this is a female, small b, small g, small g. So it means that if this individual, if this individual is this one, if this one is a female, small g, né? And then this one is a male, small g, and then y. So the father is already this one. Yes, the father is already having small g, capital Y. Automatically, how did this individual and this individual get this recessive allele if the mother is like this? Therefore, automatically the mother can't be this, so the mother is that, like that. So it means that this Gabi is, so the answer is gonna be X capital G, X small g. So that it shows that the individual is affected because of this capital G, but has a recessive allele which is being transferred to the offsprings so that the offsprings are free from the condition. And then they're saying that we are done with that. They're saying that Anju and Pilusa have 4 children. Give the phenotype of their sons. They want to know the phenotype of their sons, Anju and Pelusa, because they're saying sons. Okay. The sons. Okay. So now look. Pelusa is a male. Yes. Pelusa is a male. And this Pelusa is what? Is affected. So meaning that is having a capital G and a capital Y. But Anju is a female. Anju is what? Anju is a female, of which is caused by a recessive allele. You see, this is Anju. No. This is Pelusa. Pelusa. And then Anju is a female, meaning that it has no effect. And remember, it is recessive. So if they are looking for the sons, so how are these sons going to be? Automatically the son is going to have this. You know why? It's because the father will always give the come from there, will always give the Y to the son, and then the X is always coming from the mother. So the disease or whatever recessive conditions in males, if it is sex linked, is caused by the female. Why? Because the father is always giving the Y chromosome. So this came from the mother. And then since it is recessive, so the person is unaffected. So the description here, you're gonna say that, and for children, give the phenotype of their. So the answer is gonna be unaffected male, or unaffected, or these guys without Hortis syndrome. Yes. Explain your answer. So is the same thing. This question is similar to the question we tried to attempt before. Explain your answer. Why are you saying that they're going to be like this? They're going to be unaffected. So you can say that Pilusa is affected. Yes, you see that Pilusa, we have seen it, that Pilusa is affected. Yes, is a dominant allele. Yes. Well, Angie is unaffected. You can say unaffected, or you can write this. Yes. Males inherit the Y chromosome, as I've talked about, that males inherit the Y chromosome from Pelusa. You see, these boys, the sons, they inherit the Y chromosome from Perusa, and then they inherited this X with the recessive allele from Anju, and this Anju is already affected. Hence, the individual is going to be like that. So, how do I get the 4 marks? 4 marks by telling us what's happening to Perusa and what's happening to Anju. Perusa is affected, a tick, while Anju is unaffected, yes. So the boys or the males will inherit the Y chromosome from the father. Give you another tick, yes, and then they will inherit the X with the recessive allele from the mother. That's all. Yes. You see, genetics is simple, but if you are not keen, it might crush you down. So you must always think before you try to answer questions concerning about this genetics. Please don't just rush and then you answer these questions. No, the questions you have to read them. You read them. You read them. You read them. You read them. So they're saying that brown enamel of the teeth is sex-linked trait. A dominant allele on X chromosome causes brown teeth in human. So it means that if you have brown teeth, yeah, you know what is happening. It means that you, even if you go teeth whitening, it is brown teeth. Anyway, that's genetics. Let me know which kind of Anyway, brown teeth, we are saying that is caused by a dominant allele, but they have not given us any allele we are supposed to use so far. Explain why more males than females have white teeth. Remember, brown teeth is caused by a dominant allele. You see, it means that if I'm going to use B for brown, yes, and then small b for white, because it is caused by dominant allele, and then it is caused by sex. It's a sex-linked trait. It means that females, the female is going to be like this, yes, or it's going to be like this. So that is a female who is brown teeth, while a male is going to be capital B only. Then still the female can be this white teeth, while male is gonna be that. It means that males have 2 options, while female have 3 options. So, and even if a recessive allele appears on X chromosome of the female, it can be masked by a dominant allele. So it means that this individual is still able to live without that, without the brown teeth. So what can you say? How are you going to answer this? Basically, you know that we say that males have one X chromosome. Yes. Look at this general description. Males have one X chromosome. Yes, you see, one X chromosome. If a recessive allele appears on that X chromosome, the individual is having the condition, while the females have two X, you see. If a recessive allele appears on X, on one X, it can be masked by a dominant allele. The individual is still brown, not what? White. So for the lady to be white teeth or that sex-linked disorder must have two recessive allele, but males only have one recessive allele. So it is very common in males. So based on this question, how can you answer this question? You can say that males only have one X chromosome. That is a tick. Males have only one X chromosome. It's a tick. Or males have Y chromosome, meaning that they don't have X chromosome. Yes. And then what do? They have to inherit one recessive allele for them to be white. You see, they only have to be with one recessive allele for them to be white. But remember, the females have to be with the 2. If they have one, they can be masked by dominant allele, and then the individual is what? The individual is brown. So males have one X chromosome. If a recessive allele is inherited, yes, the individual will have white teeth. You see? While females have two X chromosomes. Look, they have two X chromosomes. Yes? They have to be with the two recessive alleles for them to be white. Can I repeat? Males have one X chromosome. If a recessive allele is inherited, yes, the individual is white teeth. While females have two X chromosome, if a recessive allele is inherited, it is masked by a dominant allele on the other, meaning that they have to inherit two recessive alleles for them to be white. Yeah, please take note of that. We can bring it in different versions. Please don't go off. We can bring it for hemophilia. We can bring it for color blindness. So we can bring it in different versions. Yes. So that is the explanation for those marks, form marks. Then they are saying that a man with brown teeth marries a lady with white teeth. Which kind of teeth do you want? You want brown teeth or you want white teeth? Eh? You want white teeth or brown teeth? Okay. Use the genetic cross to show the possible genotypic ratio of their children. All right. What do you do here? Please, do not forget that this is a genetic cross. You have to follow all the procedures. You have to all the steps because we mark by steps. You start with P1. P1 means parent 1. Parent 1, you have to tell us the phenotype of the parent, of which this phenotype is always given in the question. A man who is brown teeth. So you can say brown teeth guy is crossed with marrying a woman who is white teeth. Then they are saying that white teeth lady. Ne? Yes. They're saying that what happens? Use a genetic cross, use XB. So we go to the genotype, the description of an organism in terms of genes. That's the genotype. The description of an organism in terms of what you see, the physical appearance of an individual is what you call the phenotype. So the problem is not phenotype. The problem is not crossing. The problem is the genotype. Because if you fail the genotype, you are most likely to lose the 3 marks. You can get 3 marks even if you don't know the genetic cross. As I told you that this is obtained from the question, check. You just copy here, paste. You get a full mark there. Then, because it is a man, I'm going to use X and Y. And because this is a female, is X and please, we only use this X and Y because they said it is sex linked. If they have not said it, I repeat it because students make mistake. And when you're marking, you feel like crying and you have no choice. The only choice which is there is to cross. Yes. So X, but they said brown teeth, and brown teeth is caused by dominant allele. So remember, they have only one option. Then they are saying, yes, white teeth. Remember, white teeth can only be like this. So small b, small b. Now we give you another tick. Now don't forget this. You go to meiosis. Meiosis, what does it do? It splits these two and then splits that one also. So I always tell you that please don't leave them naked because they are not at the beach. It's only beach where they are people who are naked. Yes. And someone said that people at the beach are not naked. Okay, walk the way you walk at the beach on the street, and then we see if you are not naked. Okay. Let's continue. Then they're saying that after that you have to do what you call fertilization. Fertilization. So fertilization here, you can use a Punnett square or you can cross. Let me just use a cross. The next time I will use a Punnett square. That it means that B, capital B, small b. This with this, capital B, yes, small b. Then this with this, xy and small b. Then this with that. Then we're going to have small b and y. And then these are called offsprings or the babies. And then this is F1. So when you're marking, how do we mark? We come here, we do this. We give you a tick. P1, F1, we give you a tick for that. You saying meiosis and fertilization, we give you a tick. Something I forgot. What is it? I forgot to say these are gametes. Please don't forget these steps. Please don't forget these steps. Because if you see now, you have 1 mark from the question, 2 marks, me saying meiosis and fertilization, 3 marks, you saying F1, P1. Even if you don't know the genetic cross, still you can get the mark. Think I'm clear? Yes. Let's continue. Let's continue. And then we say that, what was the question saying? Use the genetic cross to show the possible genotypic ratio. They want the genotypic ratio of their children. Now, I need to find out the genotype of this, the phenotype. They said genotypic is phenotypic. I need to find out the phenotype of these individuals. So, phenotype. Then I come here, say, this is a female who is normal. Female. You can't say female carrier. No. Why don't we say female carrier? Because we don't show. We can't see either the person is a carrier or not. So it's going to be 2 female, female, yes, normal. Not normal. They specify that brown teeth. Brown teeth. Yes. Brown teeth. Yeah. Brown teeth. And then 2 male white teeth. 2 male white teeth. Yes. So if I've got those 2, what does it mean? They want the genotypic ratio 2 to 2. But if you leave it like that, we cross. You have to simplify it. Divide by 2, divide by 2, say 1 to 1. So that is the correct answer. Here we give you a tick. Yeah, I think I'm clear there. The question is, if you have any challenge with it, please let me know. Let me know. See if you have any challenge with this question. If you don't have any challenge, then we continue to the next question, because basically we're supposed to look at even a dihybrid cross. Supposed to look at the dihybrid cross. Okay, let's continue. If you have any question, please let me know in the comment. I'll try to answer it immediately. Kajibene United FC. I think now we are together. Yes. Let's go to the next. Yeah, it is a pedigree. Sorry, it's not a pedigree. It's a dihybrid cross. So this today's lesson is a mixture of a dihybrid and a pedigree. I also included one question for blood group. Yes. So within 1 hour, the class will be done. So please take your time and then we try to discuss this question. These what? These questions you'll be able to uh, to 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 understand. All right. Humans, in humans, okay, and short fingers, capital F, and then widow's peak, capital H, are dominant over fingers, long fingers, and continuous hairline. Men, a man and a woman, both are heterozygous. They are heterozygous. The two characteristics, for the two characteristics, they plan on having a child. They're just planning to have a child. Okay, sharp. Let's go. Sex determination is caused by the 23rd pair. So we have chromosome number 1 to chromosome number 23. And these are pairs, meaning that they're equal to 46 chromosomes. But from chromosome number 1, yes, to chromosome number 22, we call them orosomes. From chromosome number 23, or the 23rd pair, we call it sex chromosome or gonosome. So when you're describing sex or sex determination, you're supposed to explain based on the 23rd pair of chromosome. So we can say when they say show using a genetic cross, you're supposed to do all the procedure as I've been explaining to you. That is P1 F1. So if you are not going to forget, it's fine. You can leave the F1 for now until you reach the end. So now it's gonna be P1, which is phenotype. So what are you going to say here on the phenotype? You're gonna say male, then crossing with female. You see? Yes. And then they're saying that genotype. Genotype, you come here, you tell us that this is X, this is Y. Remember, males have have one X chromosome, and then they have one Y chromosome. So, and then this one is going to be XY. So, now what happens is meiosis. Meiosis is going to be XY. It's going to be XX, and these are gametes. So we circle them. Even if you don't circle them, still you get a tick. But when you circle them, it brings about that identity or separation. And then now fertilization occurs. This is X. This is X. This is XY, and then also this is XY. So basically that is it. And then now what happens is this is fertilization, and then these are offsprings. But don't forget that this is F1 offspring. So basically use gender. So it means that you have these are females, and then these are males, meaning that you have 50% chance of producing females and 50% chance of producing males. What is the question? Is genetic process to show the gender in humans and is determined by sex chromosomes of the parents. So in this case, that's what they want. If you check our booklet, the distinction material, and you go to genetics, you'll find out that this book is showing you exactly what I've talked about. This is it. 22 chromosomes are called autosomes, and one pair is called the sex chromosome. Males have XY and female have. This is a description, yes, but what they want is this. If they say describe how sex is determined or done, then you can talk about this. But if they are not talking about that, then automatically you have to use because they said using the genetic cross. So basically they want this, they don't want the writing. Yes, let's go to the next question. Robin saying hi. Okay, yes, hi. All right. They're saying that the next question is what's the man with blood group AB and then a lady who is heterozygous for blood group B plan to have children. Yes, they plan to have children. How many alleles control blood groups? You see this booklet for the distinction material. I've already addressed these questions several times. Different blood groups result in multiple alleles. Blood group, there are 3 alleles, alternative form of a gene. Yes, 3 alleles, meaning that occupying the same locus. However, only 2 alleles can be presented into a single organism. See, so the question, how many alleles? There are 3. Allele for A, allele for B, allele for O. So these are the 3 alleles. So there are 3 alleles. I always tell students that before you go to the paper, you must answer these questions because at least one question will come. How many for blood group if the blood group comes? How many alleles? How many alleles? Yes, we said there are 3. That is allele for A, allele for B, and then allele for O. Please don't modify these alleles. When you say allele O and you put like this, we're gonna cross it. Please don't modify it. And don't say allele for A. We will cross it. This is a Roman figure. This is not I. It's a Roman figure. Allele for B, you write like this. We cross. So make sure that you write capital I and then you say A. You write capital I, you say B. You write small I or small I like that. Please don't modify them. Yes, because if you modify them, you will get 0. You will get what? You will get 0. Then we are saying that they're saying how many alleles? We are done with those alleles. Saying that describe how many. Okay, describe the blood dominance that occurs in inheritance of blood group B. Blood dominance, we have 3 types of dominance. We have complete dominance, codominance, and then incomplete. Incomplete, it means that, let me just show you with a booklet so that we don't waste too much time. Incomplete, look, this is, the moment you see a small letter, it means that that is the complete dominance. One allele is completely dominant over the other. Then we have what you call codominance, which is A and B. Both alleles are shown. And then you have the recessive. Yes. So when you talk about the 3 types of dominancy, this booklet addressed it very well. There are 3. Complete dominancy, yes, incomplete dominancy, and codominance. Complete dominancy, one allele is completely dominant over the other, meaning that you have a capital letter and a small letter somewhere. Incomplete, both letters are dominant, are capital letters. But the difference, even in codominance, both letters are dominant. But the only difference which is there is that in incomplete dominance, one allele, sorry, here you produce an intermediate. For example, if you, for example, here, this is complete, you see, at least you have one allele which is recessive. Incomplete, when you combine the two, yes, like here, when you combine the two, you produce an intermediate. You see, an intermediate color, white and red, you produce an intermediate color which is pink. While in codominance, still the cross is the same, but here you produce a mixture of the two individuals originally. So now when we come back here to the blood group, they're saying blood group B. What do we say? So it's gonna be blood group A, sorry, blood group B. Yes, remember blood group E can be homozygous, dominant, or it can be heterozygous. So because of this small error here, this small error, it shows that that is a complete dominancy for blood group B. But blood group AB is codominance. You see? Yes. So here it is incomplete, so is complete dominance. This allele for B is completely dominant over allele for I. Hence, blood group B, the type of inheritance which occurs there, the type of dominance which is occurring there is complete dominancy. Complete dominancy. Yes. Use the genetic cross to show the possible genotypes and phenotypes of their children. Hey, from where? A man who is blood group A. So you see, you're going to follow the same procedure, and I've shown you how to mark this. You come here and say P1. P1 means parent 1, phenotype, and then you tell us. I told you that you always get this from the question. Is the same man with blood group AB. So I come here and say blood group AB. Blood group, yeah, you write it. Blood group AB, and then crossing with the blood group B. Blood group B. Yes, blood group B. Yes. Then genotype. AB is obvious. There is no issue with that. So you're saying that blood group AB is A, then B, crossing with, here is the issue, is it homozygous or heterozygous? They have to give you a clue which shows that it is homozygous or heterozygous. We go back to the question. Lady who is heterozygous, you see, they said it, they gave a clue that is heterozygous. So I'm gonna come here and I say B, small letter, and then I'm gonna say meiosis. Meiosis is gonna occur, and then I'm gonna have A, and then I'm gonna have B. Please leave space here, and then here also it's gonna be B, and then small letter. And then I always tell you that circle them so that you don't get marata anywhere. So you're saying that this is gametes. So after that, you tell us you go to crossing. Yes, you can use a Punnett square or you can cross direct. A, and then this is B, and then this with this is gonna be A, I, and then this with this is gonna be B, and then this with this is gonna be B, I. So how do you call this? We call this is fertilization. And then how do you call this? We call this offsprings. And then how do you call this? This is F1 generation. So we will come and then start marking. I told you P1, F1, we give you a tick. That's Mahala question. Mahala mark. This is also another Mahala mark. This is also another Mahala mark. So out of 6, at least you have 3 marks Mahala. Mahala. So you see, you can't go with once they bring a genetic cross, any genetic cross, you have 3 marks, mahala. Then they give you a tick there, they give you a tick there, they give you a tick there. What is the question? Yeah, genetic cross show the phenotype and genotype of their children. So the phenotype, this is the blood group. You have phenotype, blood group AB, you have AB. Yes, you have A, you have B, and you have, this is AB, this is A, and then you have 2B, 2B, 1AB, 1A, and then 2B. So, and then the genotype are these ones. You can write them again. So that's how we answer questions concerning about blood group. How many? Let me know. These are the questions. Let me know in the comment below. Now, people who are on live, how many alleles control blood group? Question 1. How many alleles control blood group? Let me know. How many genes control blood group? How many? I want to see the answers. I'm waiting. I want to see the answers. How many alleles control blood group? How many genes control blood group? Those are two questions I wanted to hear from you. How many alleles control blood group? How many genes control blood group? Let's go to the last question as I'm waiting for your answers from your side. Black leopard frog can be spotted. Okay, can be okay, sharp. Spotted frog, which is a runner, and then allowed to interbreed, and then they produce 150 spotted offspring, and 50, meaning that the ratio is 3 to 1. Obviously, if it's 3 to 1, remember when you're talking about this, we are talking about dominant alleles. These are dominant and these are recessive. So it means that these ones are the one which are dominant and these ones are the one which are recessive. So which genotype? They have said it. Spotted frog were allowed to interbreed and they produced 150 spotted and 50 offspring without spots, meaning that this is the one which is spotted. Yeah. So frog, yes. They're saying that which genotype is, so this is the one which is dominant. Explain why. Explain why. Explain your answer. Yes, the answer can be explained based on the ratio you obtained, that you got 3 to 1 ratio, meaning that the dominant ones are more. Remember, when you are crossing them, you have 3 options. It can either be this or it can be this, then you have that. So in most cases, these are like that. Just give me an example. Becomes 3 and then this becomes 1. So if it's numbers, they say this is 150, this is 50, and then 150 and this is 50, divide by 50, divide by 50, you get 3 to 1. That's where I got this ratio from. And then they are saying that a frog that is heterozygous for spotted black was crossed with a frog without spots. Spotted black, spotted. We say the spotted one is the dominant, crossed with the without spots. But they are saying heterozygous. Use D. Expect genotype and phenotype of F1 generation. So you see, the answer is simple, the same way we have been doing. So basically, you do P1 F1, P1 F1, you write the phenotype, you do it for me, get the phenotype from here, then write the genotype, you do it for me. Where do I get the genotype? They said spotted, they said heterozygous. it shows that spotted is, so meaning that if they are saying spotted is going to be heterozygous like that. So if this is like that, and then without spots we said is recessive, so basically is always homozygous. Then meiosis is going to occur, then split them, then you get the gametes, then fertilization. After that, then you get the offsprings here. Yes, so you're gonna do it for me. When I asked the people to tell me about, and then I also tipped them that this is not a frog, and hey, people were chasing you, we are running away from. Okay, yeah, I was just joking. This is a frog, né? A frog, and how do we differentiate between a frog and a toad? Yes, a frog is runner, and then a toad is called a buffo. So a frog is that one which can jump. You even see it small in size. It can jump long distance. And then a toad, just the one which hops. Toads that doesn't jump. The one you usually see is they're just toads. Né? Yeah. And then the other question, as I'm winding up, the other question is how many genes control blood group? Is one gene. And which gene is that? Blood group. And then how many alleles? Alleles are 3. Allele for A, allele for B, and allele for I, for O. And then don't forget to write them correctly. Allele for A, allele for B, and the allele for yes. And then how many genes? Is one gene for blood group, yes. And then how many genotypes? The genotypes are 4. That is blood group A, blood group B, blood group AB, and blood group O. And how many genotypes? These are phenotypes. And how many genotypes? Genotypes are 6. That is blood group A. Remember, blood group A can be homozygous or heterozygous. Blood group B can be homozygous or can be heterozygous. Blood group AB is codominant, blood group AB, and then blood group O, which is recessive, making them 1, 2, 3, 4, 5, 6. Yeah, I think that is it. The next class is going to be genetic engineering. Don't forget to subscribe, like, and share for others for our daily new videos. There are some people who want to join my private classes, as now it is open. Private classes. Please find the link in the description below. Yes, join. You pick join class. Yes, so that you join the class. You get you ask questions anytime. We will try to answer you, but late night we might not answer you, but we will answer you immediately when it is morning. Yes, we'll help you with this life science and then physical science. People were asking me about agriculture. Agriculture is also coming, and also people were asking me about maths. We have not yet. We do live classes, but we have not yet started the private classes. But until the teacher said that okay, it's fine, they can create space for you. So but the live classes are always there. Here, when I talk about the private classes, it means that maybe you have in class and then you have a challenge. We have sometimes Zoom and Microsoft classes. Sometimes we attend from our website. Yes, please don't forget to share, to like, for others so they can also benefit. It is M.Saidi as usual. It has been a wonderful discussion with you. Thank you very much. See you again. God bless you. Let's look at the detailed structure of DNA. In 2020, November, December exam, life science paper 2, they brought this question. It was a 20-mark question. So describe the structure of DNA. This was out of 8 marks. So we say that we know that DNA is made up of nucleotides. Remember the structure of nucleotide? So it's made up of nucleotides. Nucleotides are the monomers, the building blocks of DNA. We see that all individuals have the same type of DNA, but this DNA is different in terms of what they code. That's why we have different characteristics. How is this possible? Yet all the DNA is made up of the same kind of nucleotide. Here is the question. How do buildings differ, yet all of them, they have the same material which build them? Yes, remember DNA is made up of nucleotides. So the arrangement of these nucleotides will bring up the different structure or the different composition of this DNA or the different genes they determine. So it's the same thing. If all buildings are made up of the same type, the same kind of material, maybe bricks, but the way how you set these bricks differ in these different buildings, and then they make them to be different. But if you break them down, you find out that they are made up of concrete or cement or mixed with water, mixed with sandy bricks, iron bars, all those things, you find them in all buildings, but the way how you arrange them make them to be different in shapes. So that's what happens in the DNA. The arrangement of these nucleotides make the DNA to be different from each other. That's why we see that all of us, we are different from each other. Even identical twins, they are different. How? You find one is tall, one is small, one is big, one is more clever, one so like that. So they are different. Even the so-called identical on my side, I don't call them identical because they are not exactly identical. They're just halfway. They are partially identical. Okay, let's continue. So each DNA is made up of nucleotide, is a tick, and then each nucleotide is now tell us what is the nucleotide made up of. Each nucleotide is made up of a phosphate group, a deoxyribose sugar, because it's DNA. If it was RNA, then we talk about ribose sugar, deoxyribose sugar. Be specific. Don't tell us that it's made up of a pentose. Is made up of a sugar. Which kind of sugar? Which kind of pentose? Made up of deoxyribose sugar and nitrogenous bases. Nitrogenous bases, yes. And the nucleotides are joined together by phosphate sugar bond to form a strand. So we have seen that it's made up of nitrogenous bases. So there are four different categories of nitrogenous bases. Mention those nitrogenous bases. There are adenine, thymine, cytosine, and guanine. So if it is RNA, then you talk about adenine, uracil, cytosine, and guanine. So these bases, they are complementary to each other. So in the case of DNA, they're complementary to each other. We saw it when we are talking about the discovery of DNA, whereby adenine goes with thymine and then cytosine goes with guanine. Doesn't matter when you say thymine goes with adenine or guanine goes with the cytosine. It's the same thing as long as you compare adenine with thymine and then cytosine with guanine. Then still you get a tick. The nitrogenous bases are linked together by weak hydrogen bonds to form two strands. So we saw that, yes, these are linked together to form two strands. So if it is a double helix, it means that with two strands. So the two strands, they coil or they twist to form a double helix. All right. They're saying that the two strands, they twist to form a double helix. So basically, that is how you are supposed to describe the structure of DNA, and then you get a mark. So here we give you a mark, here we give you a mark, here we give you a mark. A mark, each different color, you give you a mark because this one has been mentioned here. Therefore, you ignore that. This is a mark. Yeah, identifying them, they are complementary. We also give you a tick there, and then we give you a mark here, weak hydrogen bonds to form two strands. It means that DNA is double-stranded. Yes, is a mark, and then this double helix is also a mark. That's how we can mark you in exam. Make sure that you don't skip these points which are in different color, because those are the marking points we are looking for when we are marking. Sometimes people, they say that they have killed the paper. If the paper has killed them, they come out of the paper and say that yes, they have written a lot, yet there are no points. Some people, they write a lot, many pages, and then after that, they come back with the cartoon marks. They say, why am I getting these small marks? It's not about how much you write, but it's about the quality you have written, meaning that the points you have given are the points we are looking for, the facts. We are looking at the facts we need. There are some facts we are looking for when we are marking. So if you skip those facts, even if you write a cloud of words, still you will get 0 from that. So note out when you are using our resources that these marks we are looking, these are the marks. The colored part is a mark we are looking for. If you don't have these notes, request them from that number below, then you'll be able to get the points. This is the structure we are talking about, the structure of DNA or the molecule of DNA. DNA, we said that it is a double-stranded molecule. Yes, it has a nucleotide. If you look at this point, is a nucleotide. It's a nucleotide. Each nucleotide is made up of a phosphate, sugar, and a nitrogenous base. Yes, these nucleotides are joined together by a sugar-phosphate bond to form a strand. Yes, the two strands, you have 4 bases: adenine, thymine, guanine, and cytosine. Adenine goes with thymine, guanine with the cytosine. The 2 strands are linked together by the weak hydrogen bond. So the double helix, we have not shown it here. So these are the points which you need to know. Sometimes you just bring a structure, we hide this, then ask you what is this, what is this. Sometimes we hide this and ask you what is it? Because this side I have adenine, then this side I will have thymine. Sometimes we don't have anything here, but we bring this and then we put it here. So we ask you, what is this? So automatically this shape will indicate adenine. The shape which shows like this indicate the adenine. Therefore, if this shape is adenine, definitely this will be adenine, and then obviously this side will be thymine. So basically, that's how you can try to identify the structure. We will see more questions when we wrap up with this topic, so that you can know exactly how do we answer those questions, and you'll be able to get marks when you go to the paper. There is no way how you can fail if you are using these resources, because whatever we are talking about is what we said. So DNA, we said that is a double-stranded molecule with a weak hydrogen bond. Yes, and then we said that maybe characteristics of DNA still, you can write them like that. It has what you call a deoxyribose sugar. Yes, it has nitrogenous bases. You tell us the nitrogenous bases. The nitrogenous bases are complementary to each other. Adenine goes with thymine, guanine with cytosine, and then in the actual sense, DNA is double helix. So it's double-stranded, and then this double-stranded, it forms what you call the double helix. So if they bring questions concerning about the difference between DNA and RNA, so it means that you can use this point. It's double-stranded, then the other side automatically it's not double-stranded, it's single-stranded. And then this one is weak hydrogen bonds, because single-stranded has no weak hydrogen bonds. This one has weak hydrogen bonds, no weak hydrogen bonds. If this is deoxyribose sugar, then the other side is going to be, has no, it is deoxyribose sugar, the other side is going to be ribose sugar. Has nitrogenous bases, you tell us the nitrogenous bases. For the case of RNA, this one is going to be uracil. Then complementary bases, because the other one, this one is double-stranded. Yes, this one goes with this. Then it means that RNA has no complementary bases, unless it's complementing with DNA. So we will see that when you are trying to translate it. It is a double molecule, it's a double helix. RNA doesn't form any helix. Yes, because it's a short thing and it's a little bit straight. Yes, so basically that's the structure of DNA. What are some of the functions of DNA? What are some of the functions of DNA? So number one, sections of DNA forming genes carry hereditary information. So we see that DNA is very important in carrying the hereditary information. You look like your father because you obtain DNA from your father and your mother. You'll find out that the head looks like your father, the hands looks like your mother. So it means that these genes are being carried from one generation to another generation. What about when you resemble someone? You go to the village and then you find someone there, you resemble that person. Does it mean that something happened? Write in the comment below and then tell us what is your view concerning about that. Then when I see many comments, I'll come back and then I'll give you the reason why you resemble someone far. Think about it. All right. DNA consists of coded information for proteins. Number one, it carries inherited information. The way how you resemble, you obtain it from your parents. Number two, it consists of coded information for protein synthesis. So most our body is made up of proteins. The biggest percentage is proteins. So hormones, enzymes, which bring about the normal functioning of our body, so is made up of proteins. But it is DNA which codes for those proteins. We are going to look at protein synthesis when we continue. Yes. So, but it's DNA which codes for that. And I explained that in the previous lesson. So in other lessons to come, we will be explaining the protein synthesis. All right. So those are the two functions of DNA in our body. We saw that DNA has complementary base pairs. So let's look at the base calculations. In most cases, some books, they don't have this, but in most cases, we ask you this. So let's look at the base calculations. In most cases, some books, they don't have this, but in most cases, we ask it. So meaning that these complementary bases. So we're going to use the principle of complementarity. Adenine goes with thymine, guanine goes with cytosine, and then we'll be able to calculate anything which is needed. Here is the first question. If adenine is 30%, what is the percentage of cytosine? So we know that adenine does not go with the cytosine. So how are we going to bring about the percentage of cytosine? So in this case, you know that percentage means out of 100. When you say that you have got 30%, it means that you have got 30 out of 100. If they say that you have got 60%, it means that you have got 60 out of 100. So now, if they say that adenine is 30%, it means that if adenine complements with thymine, if the percentage of adenine, remember James Watson and Francis Crick, they say that these are complementary base pairs. How did they find that? They found out that the percentage of adenine is equal to the percentage of thymine. Therefore, those are complementary base pairs. So if adenine is 30%, definitely thymine is also 30%. So the remaining percentage, if you add 30 plus 30, you get 100. The remaining percentage will be for cytosine and guanine. And remember, these two are complementary to each other. Therefore, if I divide by 2, then I get the individual percentage of guanine and cytosine. This is what I'm trying to say. So we are saying that adenine goes with thymine and guanine goes with cytosine. So if this is adenine, if adenine 30, then automatically, if adenine 30, then automatically thymine is also 30. So 30 plus 30, okay, I get 60. So 30 plus 30 is 60. So this 60 is for these two. So the balance from 100, because this is percentage, it's indicated here as percentage, the balance will be for this. Therefore, I'll say 100 minus 60, which is equal to 40. So it means that guanine and the cytosine are equal to 40. So that when I add the 60 plus 40, I get 100, which is the percentage. Therefore, because these two are equal to 40, so I'll say that guanine and cytosine are equal to 40. So if I want to get one individual, because this is equal to this, therefore I divide by 2, then I get 20. Therefore, cytosine is 20%. That's the meaning, or that's how you can calculate it. So even if they manipulate it, you have to use this principle of the complementary base pairs, that adenine, thymine, guanine with cytosine. Let's look at another example. If cytosine is 20, what is the ratio of guanine to what is the ratio of thymine to guanine. This one is not worked out, therefore it means that I'm gonna just talk about it and then see if you can find the correct answer. If cytosine is 20%, what is the ratio of thymine to guanine? They didn't talk about cytosine here in the question. All right. So now what are you going to find? Find out the percentage of each individual base. Remember, cytosine goes with the guanine. We saw it here that guanine goes with cytosine. So if this is 20%, automatically even this one is 20%. Therefore, I'm going to use the same principle, 20 plus 20, because 20%, 20%. 20 plus 20 is 40, but this is percentage. Therefore, I'm going to subtract it from 100. So 100 minus 40 is going to be 60. Therefore, it means that cytosine 20, guanine is 20, therefore it's 20. And then what about now thymine and adenine? Because the balance is 60, therefore it means that adenine and the thymine is equal to 60. If I divide by 2, I get 30. So this is 30, 30, 20, 20. So that if I add, I go back to 100. 20 plus 20 plus 30 plus 30, it gives me 100. So if you add and you don't get the actual total number, then it means that your answer is what? Is wrong. So now, what am I going to do? I'm going to say that if this is 30 and this is 20, because we saw that guanine goes with the cytosine, which is 20 here. Therefore, this is 20 and this is 30. So if I divide it by 10 to get the simplest ratio, I'm gonna get 3 to 1. So your answer must be 3 to 1. And this must be corresponding to this arrangement. Don't say that guanine to thymine, then your answer will be wrong because you have set your own question. Your question is the direction is saying that thymine to, or the arrangement thymine to guanine, not guanine to thymine. So we can find this question in objective or in when you are just writing a small subsection. So please don't forget to know how to calculate this. Then another question, you can also practice it. If DNA has 5,000 bases, only 500 is guanine. What is the percentage of thymine to cytosine? Drop your answer in the description below, and then I'll come back to you when I say that there is big challenge in that. So if not, try to contact that number here so that you can be helped just in case that you don't get it. Or you can try to find your teacher and then you'll be able to help you out. Let's look at DNA replication. What is DNA replication? Soon we'll be having an online discussion with the whole country. I'll be letting you know the dates where we're going to have the online discussion. You ask any question you would like to know, and then I'll be able to answer those questions. Even if you're asking the question ahead, but I'll be specific. I'll give you specific topics that, okay, you can look for questions from this topic to this topic. I'll be able to answer those topics, those questions in real life. Thank you very much.
Please use the link below to get the notes, questions, and other videos. Don't forget to subscribe, like, and share to others for our daily new videos.
Yeah, welcome back, guys. Welcome back. Today you're going to be looking at what we call the process of meiosis. So we saw the introduction of meiosis, and today we will be looking at the detailed process of meiosis, and where most questions come from in examination. Yeah, don't forget that we say that in this video we will be having some gifts for those people who are watching. The process of meiosis. Don't forget that we say that meiosis is the process whereby one cell divides to form 4 daughter cells. These cells are genetically different from each other and from the parent. The process of meiosis, we say that a type of cell division that results in 4 daughter cells, each cell with half the chromosome number to the parent. So meiosis is a continuous process. It's a continuous process, but it's divided into different phases. And events. So the events, for example, The first event is what called the interphase. Interphase is not part of meiosis. You must know that interphase is not part of meiosis. It's just a growth phase, because meiosis is regarded as a reproductive process in the reproductive cells, which are determining the cells which you call the gametes. So it includes DNA replication. That's where DNA replication takes place. Remember, when you're talking about DNA, we asked a question: where does it occur? We said in the nucleus. When does it occur? It occurs before the cell divides, or it occurs during interphase. So when you say before the cell divides, it means that interphase is not regarded as a phase of meiosis. It's just a growth phase. It's like saying that you give birth a baby, and then the baby immediately cannot give birth, joining it. If it is replicated chromosome, it will have two chromatids, and these chromatids must genetic material, so that it can be shared equally by the new cells arising from the cell division. It means that now these cells, the new cells which are going to be formed, they are going to have the content. They are going to have the content. They're going to have the chromosomes. They are going to have other cell organelles equally, shared equally, because each cell must have this content. So if it doesn't have, then it means that it will be empty, and then it won't be able to survive. So it is the function of interphase, the growth phase, to bring about the doubling of these organelles and the doubling of the genetic material, so that each cell, each new cell, has a equal amount of the genetic material and also the cell organelles. If we have said that interphase is not the part of meiosis, then what are the phases of meiosis? Number one, we have what called the prophase. Number two, we have what called the metaphase. Number three, we have what called the anaphase. Number four, we have what the telophase. And then we go to prophase 2, prophase, metaphase 2, anaphase 2, telophase 2. So we are talking about prophase, metaphase, anaphase. What is it? What does it mean? Prophase means preparation. Pro means preparation. P versus P. Then you have metaphase versus middle. M versus M. And then anaphase versus apart. A versus A. And then T means telophase, T versus T. So even in prophase 2, metaphase 2, anaphase 2, and then telophase 2. So we form if you take the first letter, we form what we call IPMAT. This is in different color. It means that this interphase is not part of meiosis. That's why we don't have interphase 1 and interphase 2. We only have interphase. So it means that it's not a part of meiosis. So the process of meiosis begin with prophase, metaphase, anaphase, and telophase, whereby we say that this is preparation, middle, apart, and then they have reached the what? The terminal. What are those things which we are talking in middle, apart, terminal? What are they? We are talking about the chromosome, as you see them here. So this is trying to show you exactly what is happening. So it means that we start with one cell, the one cell divide from 2 cells, and then these 2 cells divide from 4 cells. But actually, what is really happening for this cell to divide is what you're going to look at in the details of each phase. Let's look at meiosis 1. Meiosis is divided into 2 phases, into 2 divisions, not phases. The first meiotic division and the second meiotic division. So in the first meiotic division, it will have prophase 1, metaphase 1, anaphase 1, telophase 1. And then the second meiotic division will have prophase 2, metaphase 2, anaphase 2, and then telophase 2. So the first meiosis, this is called reduction division, whereby when you talk about reduction division, you start with 46 chromosomes. At the end of meiosis 1, you end up with the 23 chromosomes in each cell. Hence, we call it reduction division. This division results in the reduction of the number of chromosomes from 46 to 23 in each cell. So the resultant gametes are haploid. It means that they have one set of chromosomes. From chromosome number 1 to chromosome number 23, they are not paired. It means that they are single, haploid, half. It means that the cell has 23 chromosomes, not 23 pairs of chromosomes. All right. Let's look at the phases of meiosis. The first phase is prophase, which means preparation. The cell is preparing for whatever is going to be doing. So the first thing is chromatin network. We say that chromatin network means that entangled DNA. DNA is in form. We don't have a direction when we look at it. We don't know where it starts or we don't know where it ends. So it is chromatin network. When it condenses, it becomes thick. When it becomes thick, it forms what called a chromosome. So the chromatin network condense. When they condense, it means that now that it has formed a chromosome. Now these chromosomes, the chromosome which look alike, homo, which means the same, logos, which means structure. So it means that chromosomes which look alike, they are going to come together. That's why you're saying that homologous chromosomes are formed. It means that the chromosomes which look alike, they come together. When these homologous chromosomes, the way you see it here, when they come together, they cross over. What do they use to cross? They cross using the chromatids. The one chromatid of one homologous chromosome crosses with another chromatid of another homologous chromosome, and then the process continues. So the spindle thread starts to form. It means that now the spindle fiber, they start to form, and then once they start to form, the nuclear membrane starts to disappear. Let's look at the diagram here. So you see that here, the chromosome, they condense, the homologous chromosome. You see this one is short, this one is long. So this short cannot go with the long one. No, the short goes as with the short, the long one goes with the long one. That's the principle. These chromosomes, they come together, and then the chromatids, they cross each other. When they cross each other, we call it crossing over. That's why I'm saying that crossing over takes place. And then the point where they cross over, we call it chiasma. We're going to look at homologous pairs. Let's look at the process of crossing over. So we have seen that the homologous chromosome, the chromosomes which look alike, they're going to line together. When they line together, one chromatid of each chromosome overlap. So it means that they cross each other. When they overlap, these chromatids, they must overlap based on the homologous pattern. For example, it means that the one chromatid from one homologous chromosome and another chromatid from another homologous chromosome, they cross over, and then when they overlap and they cross over, it means that they cross over at a point called chiasma. And then crossover takes place at a point called chiasma. If it is one, we call it chiasma. If there are many, we call them chiasmata. So when we say chiasma or chiasma, depending on the kind of English you are using, then chiasma or chiasma is a point where these two homologous chromatids cross over during prophase 1. And then now the chromosomes separate. So it means that now they have formed different chromosomes. I always ask this question to different people. Are you sure your father is your father? Why am I asking you that? You don't resemble him. Actually, you don't even resemble your mother. You go somewhere, and then you find out that you resemble someone. Why? And they ask you that, hey, when you resemble someone, I know that. Why? Basically, he might be your father. But because of crossing over, this made to the exchange of the genetic material. If you came from the same father, same mother, you breastfed on the same breast of your mother, why don't you look alike? It means that this crossing over, this process of meiosis helps in bringing about variation among organisms. So it is a very important process to make humans, individuals, different from each other. And then this helps in the survival. If all of us, we are the same, then it means that we won't be able to survive. Imagine everyone is rich. Do you think that there will be someone who is working for another one? Everyone has money. Do you think I would have taught you this? No, because you don't need this. No one would have been watching. You don't need this. You have your money. What else do you want? But because there is difference among these organisms, among us, then it brings about what we call competition. Hence, different people will work differently and in different environmental conditions. So that's the purpose of meiosis, to bring about variation among organisms of the same species. All right, let's continue with the process of crossing over. So these differences is being brought about by crossing over. Imagine a father producing over 100 million sperm for a single ejaculation. And then this sperm, each sperm is different from each other. What made them to be different from each other is the process of crossing over, because it forms different combination of chromosomes. All right, let's look at. So we're saying that you have one homologous chromosome and another homologous chromosome. They come together. Yes, when they come together, they overlap. The homologous chromatid overlap at a point called chiasma. Yes, if you look at ABC and then decide ABC. Yes, when they cross, now this one takes this red and then the red takes the blue. That's why you see it here. The blue takes the red and then the red will take the what? The blue. These chromosomes during crossing over, scientifically we call it a bivalent or bivalent, based on how do you write that? How do you pronounce that? Bivalent or bivalent? And then after that, they separate so that each chromosome has genetically different, each chromosome has different material from one another. Look, if you see that ABC, it started with ABC, but this is a small letter. The small letter was found on this chromosome. And then this one, ABC, but this one is a capital letter. The capital letter came from this blue. If you look at it here, it came from the blue. It means that the blue, the red took the blue, and then the blue took the what? The red. So because of this, it's going to cause different gametes being formed. And then, if you look at it, sometimes we can bring it in the exam like this. Homologous chromosomes, when they cross from top. So when it crosses from top, it means that you have to draw it on top. Don't draw it down. That's one of ways of losing marks if you don't show the variation correctly. So they have the chromatids cross over and then they exchange the material or the information. And then now the chromosomes now have new combination of the genes. Guys, don't forget that I told you that there is a gift in this video. Just click that link below. It will take you to a specific page on our website. And on that page, there is a code for you to obtain that airtime. Enjoy it. When you get it, when you load it on your phone, let us know that you got it. Thank you very much. Let's continue. And then you're saying that the second phase is metaphase 1. Metaphase 1, meta means middle. It means that now the chromosomes are always in homologous pair. Homologous chromosomes randomly align along the equator of the cell in 2 rows. It means that they align along the equator in 2 rows. One from chromosome number 1 to chromosome number 23. Another one is from chromosome number 1 to chromosome number 23. That's what it means. Each chromosome is attached to the spindle thread from each pole. This is what I'm trying to say. So they align in 2 rows. You see, they align in 2 rows, and then these chromosomes are being held by the spindle. See, each chromosome is attached to the spindle thread from each other. They do this arrangement randomly. So the key point is homologous because they're in pair and then each other. You see, this one cannot come here. No, they must be together like this. Homologous chromosomes. Yes, that's the key point. And then they come here randomly. Remember, we have 23 pairs, but we're just showing you 2 pairs. Yes, they are here randomly. So it means that another one can be here. This one can be on top. So this set can be on top. This one can be down. Or this chromosome can be this side. This one can be this side. That's what we call randomly. So when they separate, we're going to form different cells. So each chromosome is attached to the spindle fiber or spindle thread from each pole. That's what we are trying to show you, that these threads are attaching the chromosomes from each pole. So after that, once they are done with this, then they start to move apart, which you call anaphase. So in anaphase, the spindle thread shorten. They try to pull. When they shorten, they pull. It's like when you are a bodybuilder, when you carry something, the muscles shorten. When they shorten, they pull. Yes. So they pull the homologous chromosome to opposite poles. And then the chromosomes, what happens to these chromosomes, the homologous chromosome pair, then they do what? They separate. The chromosomes of the homologous pair, they separate. It means that now they move to opposite poles. This is what is happening. So now they move to opposite poles. And then now, when they move to opposite poles, now they reach the pole. So in telophase, each chromosome reach at the pole of the cell. And then once they reach the pole of the cell, the cytoplasm and the cell membrane divide. When the cytoplasm and the cell membrane divide, that process is called cytokinesis. We call it cytokinesis, division of the cytoplasm. And then the two haploid cells are formed. Two haploid cells are what? Are formed, and then the nucleus or the nucleolus, that dense body in the nucleus, they reform. Then it reforms, and then lastly, the new nuclear membrane forms, and then now the cell appears to be a full cell. So this is what I'm trying to say. Yeah, so now we form two cells. Now these cells, cytokinesis took place, and now the nuclear membrane start to reappear, and then the cell goes back as if it is in prophase. So now when it reaches there, now each cell, each of it will undergo meiosis 2. It means that now this one is going to go under meiosis 2, even this one is going to undergo meiosis 2, so that you form 2 here and 2 here. So when you add them, you form 4 cells. So when you go to meiosis 2, what happens? Now prophase 2, you go to prophase 2. When it's meiosis 2, prophase, it becomes prophase 2. The 2 must be there. If you don't write it there, we won't give you a mark because we don't know which prophase is it. So prophase 2, what happens? The nucleolus, remember we said that it appeared, it reappeared. So now the nucleolus and the nuclear membrane, they disappear again. But now here we don't have crossing over. So the new spindle fibers are formed. Yes, they start to form, and then the chromosome become visible. It means that they condense, and then it becomes visible. And then now, once they become visible, now it's ready to undergo metaphase. They go to the middle of the chromosome, the cell. So now you see it here. Now it is forming like this. And then now they are ready to move this thing to attach themselves to the chromosome. But now we don't have homologous chromosomes. We have chromosomes which are not homologous. It means that crossing over cannot take place because it already took place before. Yes. So now, in metaphase 2, the individual chromosome line up. Here, individual or one chromosome align along the equator singly or individually. So the other side, we say that chromosomes, they align along the equator in homologous pair and randomly. Here, they align along the equator singly or individually. So you have to know the difference. So sometimes you ask you the difference in meiosis 1 and meiosis 2. You can use the process to differentiate between the two. So at the equator of the cell, with the centromere attaching the spindle fiber, or centromere attached to the spindle fiber. And then now, this is the picture. If you rotate it, you will see that these chromosomes are in the middle. You see, they're in the middle. If this is the pole and this is another pole, they are in the middle. So this one becomes the equator. Remember, I said that equator is an imaginary line. It's an imaginary line. It's a line which we just imagine, but the line is not there. It's an imaginary line which divide the cell into 2 equal parts. It's the same thing, equator of the earth. Imaginary line running from east to west and dividing the earth into 2 equal parts. We're just imagining, but the line in actual sense is not there. We just imagine that that's the center of the earth. So we just imagine that's the center of the cell. So in anaphase, once they're in the middle, they have to separate to opposite poles. So in anaphase 2, you have spindle fibers start to form. So the spindle fibers, they contract. When they contract, it means that now they pull the chromosomes apart. But now because they are single chromosomes, they are not homologous. Now it's the chromatids which are going to separate. Remember, chromosome is divided into 2 chromatids. Now the one chromatid is going to go one side and another chromatid is going to go to another side of the cell. So this is what I'm saying, that now the chromatids move to opposite. It was together here. Now this one pulls it this side and this one pulls this side. Check the direction of the it's not facing this way, it's facing that way, because the force of pulling is coming from this side. So that's why it's supposed to be like that. When you're drawing, make sure that you take caution when you're drawing it, so that you draw it properly. And check about the shadings. Also, the shadings are very important when you are doing it. The DORA chromosomes reach the poles once in telophase 2. In telophase 2, the DORA chromosomes reach the poles. When they reach the poles, we don't call them chromatids anymore. We call them chromosomes. Actually, that's what we call single-stranded chromosomes or unreplicated chromosomes. So it's supposed to get time, so that this time allows these chromosomes to grow, so that now it becomes double-stranded again. That's why we need what you call interphase. So new nucleus then forms. It means that the nucleolus also come back, or it means that it reforms. Then the cell membrane of each cell constrict, and then the process of cytokinesis occurs. When the cytoplasm divides, now the process of cytokinesis occurs, and then for haploid, it must be haploid, DORA cells are formed. And then now each DORA cell have the chromosome number to that of the parent. So that's why you're saying that meiosis is the process whereby one cell divide to form 4 DORA cells which are haploid, or which have half the chromosome number to that of the parent. And these chromosomes are genetically different because of the crossing over which took place. So the DORA cells are genetically different, as I've just said. And then these are the cells which will form. We form 4 DORA cells, and then you see, we don't call these ones chromatids. We call them single-stranded chromosomes. Watch out this before you write your SBA. So the importance of meiosis. What are some of the importance of meiosis? Let's look at the importance of meiosis. Shall start from there with the importance of meiosis. M.Saidi was giving me the lesson. Thank you very much.
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. We have 3 types of dominancy: complete dominancy, codominancy, and incomplete dominancy. So let's look at them in detail. When we talk about complete dominancy, when you are dominant, it means that you have powers over another individual. So it's a genetic cross where dominant allele masks, blocks the expression of the recessive allele in a heterozygous condition. So in complete dominancy, you have heterozygous condition. So you have what you call the incomplete. When you talk about incomplete, it means that yes, the dominance is there, but it is incomplete. So it means that one brings 50 and then another one brings also 50. So you're saying that it's a genetic cross where none of the allele is dominant over the other. Neither of the allele is dominant over the other. It means that they have equal powers. The president of South Africa and the president of America, both of them, they have the same powers in their countries. So it means that neither of the president is dominant over the other. When it is the meeting, the president of South Africa will have the same contribution like the president of America. That's the meaning. Not that, no, because America is the superpower, now South Africa, no. Both of them, they are presidents. Neither of the two is, they are dominant, yes, they are dominant, but neither of them is completely dominant over each other. Although your country is big or small, still you have the same power as presidents. For example, you have white and red. When you cross them, they form what called the intermediate. Intermediate means that neither of the two is dominant over the other. When you cross them, they produce the intermediate. It's not white, it's not red, but it is in between there. It produces a new character. So the character which is being produced is new. That's the meaning. Yes. But here you have the one character from the parent. Black and white, you either produce white or you produce black. Here when you cross, you don't produce white, you don't produce black, but you can produce gray, which is in between the two. It's not white, it's not black, but it's in between. That's the meaning, intermediate. And then you have the co-dominant. Co means together. Co means together, means two things are existing at the same time. They have the same power, they have expressed themselves at the same rate. For example, you have white and black. When you cross them, you produce a mixture of white and black. They are core, they are together, they are expressed outside or in the phenotype together. You have the white and the black, they are living together. That's the meaning of co-dominance. It means that both of them are dominant, and their dominance is being expressed in the phenotype at the same time. So here you're saying that a genetic cross in which both alleles are expressed equally in the phenotype. Let's look at these examples. You have complete dominance. Dominance allele completely masks the recessive one. It means that you have here, you have the red and the pink, and then you produce red. It means that you have one character being produced in the offspring, either red or pink. One must be produced. Then in incomplete, you have red and white. You don't produce red, you don't produce white. You produce a new character, which is pink. So neither of these is completely dominant over the other. So they produce an intermediate between red and, it means that this one has brought 50 and this one has brought 50, and then I form an intermediate color. Then if you look at the codominance, it means that the two must be there. If you look at this color and look at this color, the red and blue, when you cross them, you produce a mixture of red and blue. Here you produce a mixture. It means that red is completely dominant and blue is also completely dominant, and they express themselves in the phenotype of the offspring. While in incomplete, this is an intermediate, because this one has brought me 50 and also this one has brought me 50. This one, you don't produce neither of the color from the parent. You produce an intermediate. You produce a new color. And then this one, codominance, together, it means that both of the parents' color is being expressed in the phenotype. I think that's how you can differentiate among the two, among the three. Then let's look at one of the questions involving the dominance. So they are saying that an allele for tallness is dominant over allele for shortness. A man who is homozygous. So I know that homozygous, tall, marries, and then they said, yeah, tallness is dominant over shortness. So it means that this guy is homozygous dominant. So marries a lady who is homozygous short. And then short, they say that is what is recessive, or it is recessive. And then using a genetic cross to determine the possible genotypes and phenotypes of the children. So what am I going to do? P1, parent 1, phenotype. Tall guy marries a short lady. You get it from the question, as I told you. From the question, a man who is homozygous, tall. This word homozygous will help us in the genotype, to identify the genotype. The word homozygous, heterozygous, only is important in showing which kind of genotype are you having. You don't put it in the phenotype, because you can't see that this person is heterozygous or is homozygous using the phenotype. You can't do that. You only know that this person is tall, this person is short. But if you go into genetic composition, you will know that, oh, you are homozygous tall or you are homozygous short. That's why we don't add the word homozygous or heterozygous here. So we are saying that a man who is homozygous, we have seen tall, yes, marries a lady who is homozygous short. So we go to genotype. Genotype. They said homozygous tall. Letters are supposed to be the same. And then you have homozygous short. Letters are supposed to be the same. Why they said it here? Homozygous short. Then you go to meiosis must take place. When meiosis takes place, it divides into two. Even here, it divides into two. What do you form here? You form what's called a gametes. So it means that this T is going to come here, this T is going to come here, this T is going to come here, this T is going to come here. And then fertilization must take place. It means that they must cross. You can use a cross or you can use a Punnett square. Either of the two is correct. And then they cross that, crosses this, and then that crosses that. So this one crosses with these two. That's one is done. Then I go to this. This crosses with that, and then this crosses with that. And then I get the results, which you call the F1, the first period generation. So when I cross, I get this. So it shows that all of them were tall. But which kind of tallness? The tallness, if you are describing in terms of genes, it is heterozygous tall. So, or you can use this kind of option. Don't use two because we want one. So then the genotype, it means that all of them are capital T, small t. Which kind of phenotype is this? Because I have the capital letter T, then it means that this individual is going to be tall. So which kind of phenotype? All of them are tall. I don't say heterozygous tall. I don't see the heterozygosity in the phenotype. I don't see that. So I just have to say they are tall. And then the individual of F1 all displays the dominant characteristic. It means that all of them are what? Are tall. In incomplete, you saw that in incomplete, you have the capital letter and the small letter. You see? You have capital letter and small letter. But in incomplete, neither of the allele is completely dominant over the other. Then it means that we don't have a small letter there. So an allele for red and allele for white is also dominant. When the homozygous red crosses with the homozygous white, it means that both of them are dominant. Using a genetic cross, determine the possible genotype and the phenotype of the flowers produced. I have P1 phenotype red. I get it from the question. Red crosses with the white. They're saying it. Red, white crosses with the red from the question. So I always get that mark from there. You have 3 marks free without even thinking. Then genotype. Now I have to come here and then look for these words. They're saying when a homozygous red, you see, homozygous red, same letters, homozygous white, same letters. Then meiosis takes place. When meiosis takes place, it also splits into gametes. So gametes, when they are formed, fertilization must take place. So you can use that method or you can cross. Either of the two. So you bring these ones, you put them there. You bring these ones, you put them there. Then you cross them. You cross them. Or you can bring these ones here and then bring these W's on top. Either way is correct. And then F1 generation, if you look at them, all of them, they are capital R, small w. Capital R, small w. So the genotype, all of them is capital R, small w. And then the phenotype, which kind of phenotype is this? Capital R, small w. If you go back here, capital R and small w, it's not red, it's not white. So it shows that it is an intermediate of the what? Of the two. So it gives me pink. So if it gives me pink, it shows that this is incomplete dominance, whereby I produce a different color. And then offsprings have an intermediate form of trait of the parent. So that's how I can describe them. So note that in incomplete dominancy, you don't have small letters. Then codominance is the same as incomplete dominance, but the only change which is there is because you produce a mixture of the two colors. So they're saying the cow with white fur, capital W, were crossed with a bull with red fur, capital R, as shown in the diagram. Use the genetic cross to show how the roan was got. So how did we get this? So phenotype, P1 phenotype, I have to look for it. Okay, well, white fur, so I say it's white fur crossed with red fur. So it means that red is crossing with white. Red is crossing the white. Here they didn't talk about homozygous, heterozygous, but the statement, the word roan, shows that it's a mixture. Roan means a mixture of two colors. So it shows that these two were homozygous, so that they gave rise to this roan. So now I have to know that capital R and the capital R, small w, sorry, capital W and capital W. Then when meiosis takes place, I produce gametes, which gametes they undergo fertilization. I give you a tick and this and this. So it means that you have scored a tick there. And then I cross them. You can use the cross or you can use this method. Either way is correct. And then I get the F1. So P1 F1, I give you also a tick. So please don't forget those. Don't forget this. Don't forget this. Don't forget this. 3 marks are there for mahal, for free. So we are saying that the genotype is this. Why? All of them are like this. Capital R, small, capital R, capital W, capital R, capital W, all of them. So it means that this is not red, it's not white. What is it? It's a mixture of red and white, which we call roan. That's the meaning. And this is how they wanted you to do that question. It's not difficult if you look at it. In codominancy, you produce a mixture of the two colors. While in incomplete dominance, yes, you produce an intermediate color. But the crossing is exactly the same. So we are saying that both alleles are equally dominant and are expressed in the phenotype. Okay, this is just trying to do a summary. Proportion and the ratio of the genotypes and the phenotypes. Sometimes they ask you to do the ratio, sometimes they ask you to do ratio. So when you talk about the ratio, it describes the number of times a genotype would appear or the number of times the phenotype would appear. So for example, if you look at this is capital R, capital R, capital R, capital R, capital R, small r, capital R, small r, and then small r, small r. So I just have to say this is 1, and then this is 2, and then this is 1. So I'm going to say 1 to 2 to 1. 1 is for this, and then for 2 for this, and then to 1. That's basically what they want in the ratio. And then if phenotypic ratio, you see how many, if this is, let me see, did they give you, they didn't give you the color. For example, if this is red and then small r is white, then it means that this one's gonna be red. This one's gonna also be red. Why? Because it is having a dominant allele, which is masking this. So it's gonna be red 1, red 2, red 3, and only 1 white. So I'm gonna have 3, 2, 1. That's the meaning here. Actually, it's the same thing I've given you. Red is 3 red and 1 white. So the ratio is gonna be 3 to 1. Note, let students practice different examples with the crosses. So it means that when I finish here, I'm gonna send you some questions for you to practice. So basically, that's it. And then if you look at sex determination, we are saying that
welcome back to our class. Yeah, today we are going to be looking at the process of translation. Here, let's look at the process of translation. Basically here, now the messenger has moved out of the nucleus. It has gone to the cytoplasm. And then it attaches itself on the ribosome. So now we want to bring the amino acid to the ribosome so that a chain is formed. Number one, what you say is each transferrin A carries a specific amino acid. It means that it has 3 bases. These 3 bases, they code for a specific amino acid. These bases are called anticodons. When the anticodons on the transferrin A, yes, those 3 bases happens, matches the codons on the messenger. So the 3 bases on DNA, we call them codes. The 3 bases on the messenger, we call them codons. The 3 bases on the transfer, we call them anticodons. So the DNA code for the codons on the messenger, and the messenger codes for the 3 bases on the transfer, which are the anticodons. And then the anticodons, they code for the specific amino acid. That's why we're saying that each transferrin A code for a specific amino acid. It's like a key. The other one will not enter or to open. When the anticodon on the transferrin A matches the codons on the messenger, so what happens? This transferrin A will bring the required amino acid to the ribosome. So it will bring the required amino acid to the ribosome, and then the amino acids become attached by a peptide bond. We call it a peptide bond, not polypeptide bond. Don't say polypeptide bond, we will cross it. It's called a peptide bond. If there are many, we call them peptide bonds, not polypeptide. When you say that, cross it. So amino acids become attached by peptide bonds, and then to form the required proteins. That's what we want. So this is what exactly what is happening. This is exactly what is happening. Each transferrin A carries a specific amino acid. For example, it carries a specific amino acid, a specific amino acid. This cannot carry another acid, no. And then you're saying that when the anticodon, when these 3 anticodon, yes, match with the codon, yes, match with these codons. For example, is T goes with A, T goes with A, and then A goes with U. When they match like that, yes, now what happens? This transferrin A will bring this amino acid, which these 3 bases, yeah, which code the anticodon, will match this. So it brings this required amino acid to it brings the required amino acid to the ribosome. So now the amino acids are joined together. So it keeps on bringing, and then the amino acids are joined together by these bonds, which you call, you see, these bonds are called the peptide bonds to form a required protein. And then you form this polypeptide chain, but now it grows. Once it grows, it is more than 50 amino acids, then we call it a protein. So these are amino acid. If they ask you what is this, it is called a polypeptide chain. Yes. And then this is the messenger, the 3 bases on each, we call it the codons. And then on the DNA, we call it the codes. And then on transfer, we call it anticodons. And then this molecule where transfer, where messenger lines is up, is called a ribosome.
much for watching. Next time we are going to be looking at the mutations on DNA. And then we're going to see some questions. How do we answer questions concerning about DNA, the structure of DNA, so that you are able to pass this topic. Just 1 week is enough to grasp this topic. So you need to know that this is DNA, this is messenger, this is messenger still, this is transferrin A, this is a polypeptide chain, this is amino acid, and then this is a transferrin A, it is a T, that's why it's called T, and then this is 3 bases, which we call the anticodons. 3 bases on this messenger, we call them codons, and then 3 bases on DNA, we call them codes. Yeah, thank you very much. Thank you very much for coming back to our channel. Today we are going to be looking at DNA replication. So what is DNA replication? How does it occur? Why does it occur? Where does it occur? So that's some of the questions we ask on DNA replication. What is DNA replication? To replicate, it means that it is doubling, to double. So like making a copy. So that is the meaning of replicate. But the old and the new must be exactly the same to bring about the meaning of replication. So what is DNA replication? Is the process by which DNA makes exact copy of itself. So when you are writing a definition and you don't talk about DNA making exact copy, if the word exact is not there, then it means that you won't be able to get a tick. So it must have the meaning of, it must have the word exact. So that's why you say that the old DNA molecule must be exactly the same as the new DNA molecule. Then you're saying that when, now the question is when does it occur? When in the cell cycle it takes place? When does it occur basically? Or it occurs during interphase. Yes, you can say during interphase or before cell division, because interphase occurs before the cell divides. Therefore, it is here you have generalized. If you say that cell division, you have generalized. Then interphase, you are just specific to when exactly does it occur, because we have different phases, but the first phase is interphase. So DNA replication occurs in interphase. So where, now another question is about where does it take place, or where does it occur? Then you are saying that it occurs in the nucleus. So it means that DNA replication occurs in the nucleus. When you're talking about DNA replication in this case, we're talking about DNA replication in eukaryotic cells. We're not talking about DNA replication in eukaryotic cells or in prokaryotic cells, because these don't have nucleus. Then why does it occur? Why should we have DNA replication? Sometimes we ask the significance of DNA replication. What is the significance of DNA replication? That is the same as asking you, why does it occur, or why does it take place? It takes place to double. You want to see the word double? Yes, to double. What is it doubling? The chromosome number. Don't say to increase the chromosome number. Yes, it is correct. But when you are mentioning, we want to see the word doubling, to double the chromosome number, or to double the genetic material, so that now each cell can obtain an exact number of chromosomes. So why does it occur? The answer we need is to double the chromosome number. So if you had 23, if you were haploid, then when you double, then you become diploid, so that now the cell divides. If it is case of meiosis, divides, then goes back to haploid so that you maintain that chromosome number from generation to generation. Yes. So another one is results in the information of identical daughter cells during mitosis. Remember, cells undergoing mitosis are diploid. If they are diploid, it's supposed to double. The genetic material is supposed to double. When doubles, the cell must divide. Once it divides, then it means that each cell must have the same number of chromosomes. So it will be identical to the original cells. So it means that if I have 23 pairs in one cell, then it doubles, it's going to become 46 pairs. So when it divides by mitosis, it's going to go back to 23. So that is the meaning of to bring about to identical daughter cells during mitosis. So in meiosis, it is always haploid. Then when it duplicates, then it becomes diploid, whereby it is unreplicated. Yes, it is single-stranded. Sometimes call it unreplicated. But after replication occurs, then it becomes double-stranded chromosomes. So it becomes double-stranded chromosomes. We will see this when we go to meiosis. So in exam, when they ask you what is the significance of DNA replication, they brought this question in 2020 November, December still. But in most cases, students, when you ask such questions, they always find a challenge to answer this question. Then, how does it occur? In other words, the process of DNA replication. How does it occur? How does DNA replication occur, or the process of DNA replication? So, you have to first be with an imagination in the head so that you can describe the process of DNA replication. You need to first know the imagination of the structure of DNA. Then you have to follow the diagrammatic representations, and then you form the two new DNA which are genetically identical. So using that diagrammatic representation, from there, then you can write the points which are needed. All right. DNA we say that is a double helix. Yes, it's a double helix. It means that is two helical, first helical, and then the second helical. So it has two strands. So since we want to carry out DNA replication, this helical must straighten. It must form two strands. Yes. So it must try to break. When it uncoils, it forms this. But I'll show you how do we write in exam. So it forms two strands. Yes, two strands. After forming two strands, now these bonds are supposed to break. So you can say it unzip, or you can say weak hydrogen bonds break. So you can use any of the two because they mean the same. So that is unwinding. It starts to form these two. So now it means that you have two strands which are open. Yes, it was like this. Then because it was coiled like this, so it uncoils, so it forms these two strands. When these two strands, if you bring it here, yes, it opens. When the weak hydrogen bonds between these bases break, then it exposes these two strands. Then if these two strands, when they uncoil like this, then the free nucleotides of DNA from the nucleoplasm will come and arrange. When they arrange, they are forming now another strand on it. This strand must be complementary to this strand. So I have the new strand and I have original strand. Yes, so it means that what is on this DNA which is forming must be exactly to what is this DNA which is also formed, must be exactly the same to this previous DNA. So basically I have three kinds of DNA here, but one is splitting to form two. So
this is the which is original, so it means that it has this original strand. Still, this is original, it has this original strand, and then it has this new strand. So at the end, I'll have two new DNA. So I have two new DNA. So it's gonna form like this, whereby it has the original, the original strand here. It has the original strand here, and then it has the new strand. So then it means that you form two new DNA. How do you write in exam when you're asked to describe the process of DNA replication? DNA double helix. So you start by writing the DNA double helix. Yes, it unwinds the weak hydrogen bond. When it unwinds, it forms two strands. So now the weak hydrogen bond break. So I said that you can say weak hydrogen bond break, or you can say unzip. So when you mention one of that, you get a tick. To form now two strands, it forms two strands. Yes, because I want to form two new DNA, then each strand will act as a template for formation of a new strand. So template is like a mirror. Yes, so the old strand is acting as the object, and then the mirror form another one, which is inverse you are seeing. So now this old strand will act as a template for formation of a new strand. Even this one will act as a template formation of the new strand. What do they use to form the new strands? They use the free-floating nucleotides of DNA from the nucleoplasm. So it forms two new DNA which are genetically identical, each with one original strand and then one new strand. And then this process is controlled by enzyme. So that's basically what you are supposed to explain when you are in exam. So let's look at it when we are writing. Each different color means a marking point. So you should not forget about it. DNA double helix, we give you a tick, unwind. Yes. Weak hydrogen bonds, they break. Yes. Weak hydrogen bond break, we give you a tick. Or DNA double helix, unwind, and then unzips. Then you also give a tick here and give a tick here. So any of that, you will get a tick. To form two separate strands, you will need to see the word two, and these two strands must be separate. So you say to form two separate strands, or to form two single strands. So you get a tick for that, and then you're saying that each acting as a template. What is each? Each strand you have mentioned here is acting as a template. If you don't say each, you say two strands. The two strands formed are acting as a template. We want to see that word, either each or the two strands. We must see that. If you don't mention that, then you lose a mark for the template. Using, what do they use? Using free, want to see the word free, floating nucleotides, free floating nucleotides from the nucleoplasm. So when they use free floating nucleotides from the nucleoplasm, what happens? They form what called a complementary DNA strand is formed. As I showed you that it has the new strand. So the new strand becomes the complementary strand to the original one. So it forms the complementary strand, whereby adenine pairs with thymine, and then guanine pairs with cytosine. The two new DNA molecules are formed, whereby these two new DNA are genetically identical. So it forms two new DNA, and each DNA is identical to the other. So basically, the two new DNA are exactly the same. That's why I said that DNA replication, we want to see the word to form exact copy, exact. So that if they are genetically identical in terms of genes, then it means that it won't affect the physical appearance of an individual. So the person won't keep on changing and changing and changing. So it will remain the same. That's why it is not affecting the physical appearance. Then you're saying that with one original strand and one new strand. So the DNA, the two genetically identical DNA formed, each is having one original strand, and then each is having one new strand, as I explained before. This process is controlled by enzyme. So basically, that's how you are supposed to write. Here we give you a tick, 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 give you a tick, a tick, a tick, a tick, a tick, and then a tick. Most sometimes we also give you a tick here, but sometimes we don't give, depending on the marking guideline. So that is basically the process of DNA replication, and that's how we are going to ask you. What about DNA profiling?
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Evolution in present time. What happens? Does evolution takes place? We want to see it because we are saying present time. Then it means that evolution is taking place. Let's look at it in detail. Describe how mosquitoes develop resistance to DDT. How did it, like when you apply DDT, it does not affect mosquitoes anymore. Then it means that there is a resistance which has been which has been formed. So we need to know that. So that is the evolution. It means that evolution, remember I told you that evolution is increasing the complexity of an organism. The organism become more complex. If the organism become more complex, it means that what was working on it, it's no longer working on it. It has become more complex. It has modified its structure. Then it means that it cannot It cannot be affected by such. So you're saying that now we have these mosquitoes, yes? If the mosquitoes, you have this kind, you have this kind, you have this kind. So what happens when you apply the first application of DDT? Yeah, you see that all these ones, they die. Yes, you see them that they have died. Only this and this, they have survived. So it means that there is variation among the species. There is variation among the species of mosquitoes. Yes, during the first application of DDT, yes, those which are resistant will survive, while those which are not resistant will die out. Those which are resistant will reproduce in large numbers, and they will pass their genes to the next generation. Yes, when they pass their genes in the next generation, they reproduce in large numbers. And then when you apply again DDT, it means that it cannot affect them. So it means that now the mosquitoes have become resistant to what? To DDT. So first application, there is variation among these mosquitoes, whereby some are resistant and some are non-resistant. But the one which are non-resistant are very many compared to those are resistant. So when you apply many which are non-resistant, they're going to die. You see them, they're fading out. While those which are resistant will survive. Yes, these ones which are resistant will pass their genes to the next generation, will produce in large numbers and pass their genes to the next generation. The second application, yes, of DDT, yes, they will not be able to affect them. Why? Because the one which are resistant have survived. Then it means that the DDT will become ineffective to the mosquito. It happens in when you take drugs and you don't complete the dose. It happens HIV, it happens in bacteria. Yes, it happens in their life. So let's look at it. There's a large number of degree of variation in the mosquito. Yes, we saw it. When the DDT was first used, it killed off the large number of mosquitoes, but some mosquitoes were resistant to the DDT and they survived. Those which are resistant, the black one, you saw it, they were resistant, so they survived. When they survived, they passed. Those that survived were able to reproduce. They reproduce in large numbers, increasing the population of resistant mosquitoes. Yes, continued use of DDT had little effect on the resistant mosquitoes. It means that those which are resistant, now they have conquered the environment. So DDT will not be working on them. So at the end of the day, you'll find out that DDT will become ineffective to mosquitoes, or it becomes effective in killing the mosquitoes. Hence, we are saying that hence the resistant mosquitoes increased, and then non-resistant mosquitoes, they died or they decreased in numbers. So basically That's it. That is the present day evolution. Other examples which can help you, you can do it with your teacher. Number one, this is resistance in bacteria. Yes, development of resistant strain of tuberculosis, that is bacteria, Mycobacterium tuberculosis, due to the mutation. So this the bacteria, it will reproduce in large numbers. As it is producing large numbers, it produces some resistant. When you apply the antibiotics, yes, these ones which are not resistant will die out, and those which you see they have died out, those which are resistant, the two of them, they will survive, and then they will reproduce in large numbers. They will pass their genes to the next generation. After many generations, or after successful application of the antibiotics, yes, the antibiotic won't be able to work on the bacteria, and then it means that the resistant one will survive. That's why when they are giving you medication, sometimes they give you a combination of medication, because they know that there could be some resistant, so they must give you the medication in different versions, so that even the resistant ones, they can be resistant to this drug, but they might not be resistant to another drug. So they give you multiple drugs. But what other people do, they just take one, and then when they feel fine, they stop taking medication. You are just creating monsters in your body. You have to take the drug completely without leaving them, the way how the doctor has told you to do. So even if you're fine, you have to complete the dose, so that you kill this resistant and non-resistant completely out of your body. Then another example is HIV resistance to antiretrovirus, ARV, antiretroviral, and antiretroviral, that is ARV, antiretroviral medications. So you see that if this is the HIV, what happens? You have some resistant and some non-resistant, some resistant and non-resistant. When you apply the first medication, the resistant ones will survive, will keep on surviving, while the non-resistant will die. You see that the red ones are dying. And then after many generations, the non-resistant ones will die out, and then the resistance will survive until the end of the day. You pack. So it means that now that medication of HIV cannot work on you anymore. Yes, it cannot help you out. It means that you have to go back and then change the medication. That's how present day evolution is happening. Present day evolution, you can also have another one in black peppered moth, and then the white peppered moth, industrial mineralism, the black peppered moth, and then the white peppered moth. So you can have also such examples. So basically that's it. And then human evolution is switching in. Then I will see you there when you come to human evolution. Don't forget to subscribe for our new videos.
Let's continue with the question 2.4. It's about genetics. We know that many students find challenges with genetics. Question 2.4 from National Senior Certificate 2022, NCS 2022. The diagram below shows the inheritance of flower color in snapdragon plants. The two alleles controlling the flower color are red and white. It means that because the two alleles are capital letter, this is a capital letter, and also this is a capital letter, it means that both of them are dominant. So if both of them are dominant, here it's showing us that you have a dominant red crossing with white, and it gives you pink. When you have red and you have white, we produce pink. So pink is neither red nor white, but is an intermediate between the two. So the type of dominance which is there, the moment you get an intermediate neither of the parent color produced in the offspring, it means that that kind of dominance is called incomplete. It means that red is neither dominant over white and white is neither dominant over red. An example is different presidents of different countries. When they sit on one table, all of them, they will talk about issues affecting them equally. So it means that neither of the president of country A nor the president of country B is dominant over the other. Why? Both of them or all of them are presidents. So neither red nor white is dominant over the other. So when they combine, they produce an intermediate. So let's go to the questions and we see. They are saying that state the type of dominance shown by the Snapdragon plants. So we have seen that it is incomplete dominance. If it was complete, then we would have got either red or white in the offsprings, or both red. Some of the organisms are red and some of them are white. That is complete. If it was codominancy, we would have got a mixture of red and white. But now we get an intermediate, therefore it is incomplete dominance. Give a reason for your answer. The answer, we have explained it, that you produce an intermediate or an intermediate color was produced. A gardener crossed two pink flowers. Use the genetic cross to show the ratio that is expected of when you cross two pink flowers. So it means that if both of them are dominant, definitely this is going to be capital R, capital W, capital W. Yes, they produce one gamete from this side and one gamete from that side. So this one is going to produce a capital R. This one is going to produce a capital W. So it means that this crosses with this, you produce this. And RW is an intermediate of red and white, which is pink. So it means that when you're talking about the genotype of pink, we have to use this. So what are you supposed to write? Okay. We are saying that now, the first thing you need to do is you say, because it is a second generation we are crossing, therefore it is advisable to use P2. So it's going to be P2. You tell us the P means parent. So P2, parent 2, because we have already parent 1. So now pink and pink, it means that that's P2. Yes, if it's a second generation, we use P2. P2, yes. We can say F2, second Fourier generation. We mark this as ones, like, so give you one tick there. So don't forget, if you write P2 or P1, F1, it must be P2, F2, P1, F1. If you write one, you don't write the other one, then you get it wrong. For example, P2 and don't write F2, you don't get a tick. So now P2, you tell us the phenotype. The phenotype is pink. We cross it with another pink flower. The X must be like this. It must not be like this. No, the X must be like this. Then you go to genotype. What's the genotype? We have seen the genotype here. That is capital W, capital R, or capital R, capital W. Therefore, we're going to have to say here, it is capital R, capital W, cross with capital R, capital W. Now, these must separate under meiosis. So you write the word meiosis. So meiosis, you form the gametes, capital R, capital W. Here, capital R, capital W. So we call these ones gametes. Gametes, don't leave these gametes naked. In most cases, I always tell my students not to leave these gametes naked. They're not at the beach. All right. We circle them. We circle them. We circle them. Even if you don't circle them, still you get a tick. But I prefer to circle them. Why? So that we can differentiate them from the genotype. And always know that there must be a space between these two gametes. Don't combine them, because when you combine them, then it will look like it is a genotype. There must be spaces there. All right. Now these gametes, they're supposed to fertilize each other. Don't use the word fission. We won't give you a tick. Fertilization. Fertilization, when fertilization occurs, you can use a Punnett square, or you can use crosses. Since it is a monohybrid cross, you can use crosses. For example, this goes with that. So I'm going to have capital R, capital R, and now this with that. So it's going to be capital R, capital W. Now this with that is going to be capital R, capital W. Then this with that. So it's going to be capital W, capital W. Yes. So this is what called fertilization. So these are the offsprings of the parents. So F1 offsprings. What are they? They said snapdragon. It means that those are the offsprings or the snapdragons. But what was the question? The question is saying, use the genetic cross to show the ratio of the expected phenotype in the offspring. So we are looking for the ratio of offspring. Okay, let's look at the ratio of the offspring. Now, capital R, it is red. So I have red. How many are they? It's 1. Comma. I have capital R, capital W. It is pink. Capital R, capital W is pink. So I have 2 pink. And then now I have capital W. It's white. I have 1 white. So basically I have 1, 2, 2, 2, 1. So that is it. So now, when you are looking for a mark, when we mark, we are looking for ticks, and then sometimes we put what called a compulsory mark. So in case we come here, we do this, we do, yes, this and that, we give you a tick. Phenotype, we give you a tick, it must have a cross. Genotype, we give you a tick, it must have a cross here. If it doesn't have a cross, the tick might not be given to you. So we come to the gametes, we give you a tick, and then we have for crossing, and you get the offsprings, we give you a tick. So because the question is saying give the ratio, so this one might be a compulsory mark. It might be a compulsory mark. So we give a tick, but we put the word C there, which means that it's compulsory. There is one mark short, which is meiosis and fertilization. Give you a tick. So how many marks are there? It's 1, 2, 3, 4, 5, 6, 7. And then in most cases, we give you 6 marks. And also sometimes cross, we can give you a tick there. So maybe we can say that this question is 6 marks. But how do we count? When you are counting, you first count this. However much you get all of them, like you have all of this. Without this now, for this case, you won't get 6. You have to be with all of them. You have to be with all of them plus this. Or you have to be with this plus another 5 so that we make them to be 6. So that's how we mark the genetic cross. This question, it is 2022 Department of Basic Education, NCS. Another question is about evolution. Evolution. Here they're saying that diagram A and B show the ventral, that is the bottom side of the skull, of the skulls of 2 organisms. The diagrams are not drawn to scale. It means that they are not, that's not their size. So now asking, which diagram represents the skull of bipedal organism? Let me first explain the word skull. Skull. Some people, they confuse the word skull and cranium. Cranium is where the brain goes. The skull involves even the jaws. So when you are meaning where the brain goes, please don't use the word skull, use the word cranium. So skull is the whole thing, the head, including the jaws, the teeth, and everything. But when talk about cranium, please, we are talking about where the brain goes. All right. Now, which diagram represents the skull of bipedal? Bipedal, it means that organism is walking on 2 limbs. If the organism is walking on 2 limbs, it means that what shows we have 3 factors which you can identify whether the organism is walking on 2 limbs or not. The first one is the position of foramen magnum. The second one is the shape of the spine. If it is S shape or C shape. S shape, it means that it allows flexibility for the organism. So therefore, it is bipedal. And then C shape, the flexibility is not there. Therefore, it works on 4 limbs, quadrupedal. So, the pelvic girdle for the case of bipedal must be short and wider to support the upper weight and allow easy movement. The other one, it must be small and narrow. Then foramen magnum for the bipedal organism, it must be in the forward position. That's how we want it, in the forward position. While the foramen magnum, it must be in the backward position for the case of the organism which works on 4 limbs. Why is it supposed to be in forward position? Forward position so that the spine can connect to the skull vertically. And then now the skull is going to face straight in front. Straight in front. Hence, the organism is going to walk on 2 limbs. So if you look at this, it's a little bit in the forward position. Don't say in the center. Center of what? No. Even this side can be center if I count from I measure from this side to this side. So it's going to become the center. So the center, we say in forward position. What this one is a little bit backward. So it is not in the forward position like this. So it is backward. So it means that this is a bipedal organism. This one is the quadrupedal organism. So they're saying, give one visible reason for your answer. So the answer here is B. If the answer is B there, therefore it means that give one reason. It's because the foramen magnum is in the forward position. So it is in the forward position. And then they're saying that tabulate 2 visible differences between the upper jaw. Now here, they're looking only and specifically the upper jaw, these jaws which you see. So don't talk about other parts. The upper jaw in diagrams A and B. When they say the word tabulate, that's a mark. The word tablet, even you can see that is 5 marks. So when you draw a table, please make sure that you draw a correct table so that you get all the marks. So you write here, for example, skull A and then skull B, for example. Make sure that you close the table. You lose marks here. Ne? Don't just draw a T and then you write. No. If you draw a table, make sure that you put the heading. Ne? Please close. Don't just draw it like that. Please close the table. Close the table. It doesn't cost you anything for you to close the table so that you don't lose a mark. Be neat when you are writing. All right, now let's compare. Skull A, skull A. This is skull A, skull B. So number one, they're saying the upper jaw. Let's look at these teeth. This and this. The canine here, they are big, meaning that they are well-developed canines. Here, the canines are less developed, meaning that they are small. Number two, this one has a rectangular palate. Here, it has a U palate. Ne? It's like a C or a U palate. Here, it is rectangular palate. So, those are some of the differences you can talk about because they are talking about the jaws. So the palate here is rectangular palate. Here it is U shape, or the jaw, they are U-shaped, and then here, yes. And then they are saying that so you get 2 marks for the difference and 1 mark for the table. Then they're saying explain the significance of the shape of the spine. I talked about it. That is associated shape of the spine, significance of the shape of the spine that is associated with the skull B. What is skull B? Skull B, we say that it is walking on 2 limbs, and the shape, if you look at it, it is S shape. Because they didn't talk about which shape is it, then mentioning the shape could make you to earn a mark. Therefore, the shape is S shape. Yes, S shape. But what is this S shape for? It allows flexibility of the organism. So it makes the movement a little bit easier. Then, in most cases, things related to the graph, I only point out what is very important and the most likely questions which are likely to come when they bring a graph, so that there is no cramming about the graph. So now, if they ask you to describe, when they say you're supposed to read the whole question. Herbicide is a chemical which kills a plant. Glyphosate is used to control the weeds in maize fields. The herbicide kills the weed, but it cannot kill the maize plant. The herbicide kills the weed, but it can also kill the maize plants. It has been found out that weed developed resistant to glyphosate treatment. Scientists carried out an investigation to determine the development of the glyphosate resistance in the weeds. The weeds were treated with the same concentration of the glyphosate each year from 2009 to 2016, and the percentage of the glyphosate resistant weed in the field was recorded every year. Okay. Results as follows. You see that glyphosate resistance, as year goes on, the resistance increases, but 2015 and 2016, the resistance became constant. So now I'm saying, describe the change in the glyphosate resistance over a period of year. It means that here, whenever they ask you to describe, you tell us what is happening. Because if you draw this on the graph, not a bar graph, just a normal graph, you will see that it will be continuing, and then it forms something like this. So there is an increase from 20 2009, 2015, there is a steady increase, yes, in the resistance of the weed over the herbicide, and then it remained constant between 2015 to 2016. So it remained constant. So when you say description, you tell us what you see. Don't give us the reason why there is that increase. That is description. When they ask you to explain, then you tell us why, you tell us that there is an increase, and then you give us a reason why that increase is there. So that is what we call explanation. But if they ask you to describe in science or in here, you just tell us the shape of the what? Of the graph, or the trend, how it moves. That's what they want. That's why you see that the marks, even they're not that a lot. Then they're saying that calculate the percentage increase in the glyphosate resistant weeds from 2010 to 2015. Show you're working. Here, whenever they ask you to calculate the percentage increase, whenever they ask you to calculate for the percentage increase, this is what you are supposed to do. Percentage increase is equal to the final percentage increase is equal to the final figure, the final value minus the initial value over the initial value times 100. Yeah, that's it. So in this case, they say that from 2010 it is 20, and the 2010 is the initial value, and then to 2015 is 65. So I'm gonna say the percentage increase is gonna be equal to 65 minus 20, yes, divided by because the initial, it is 20, then times 100%. So the answer you get is gonna be the percentage increase. So that's how we calculate the percentage increase. So this is a rough thing how you can answer questions when you really sit in this paper, or any paper you're writing. You can really have no questions which are challenging. You are not going to see any new questions. All the questions you're gonna find in the paper are always the questions we ask you every day. Those are the questions you're gonna find in the paper. So there is no new questions because we don't have any new science which has come in. So the curriculum remains unchanged for a period of years. So no new questions. Don't expect new questions in your exam. So please try and focus on, we have the same format of setting, so we cannot set outside the examination guideline. So scientists isolated the gene from for glyphosate resistance from the weed and used it to genetically modify the maize plant. Okay, explain the economic benefit of making the maize plant resistant to glyphosate. Because the glyphosate, now look, they are saying that herbicide is a herbicide which kills plant. Maize is also a plant, and then we use it to control weed in the maize plantation or the fields. So we have to make sure that this glyphosate cannot kill the maize. So the reason why they make this maize to be resistant to glyphosate, so that when you apply the glyphosate cannot kill the maize what? The maize plant. So that is the idea behind it. So I'm not saying that this is the exact question which is going to come, but when they give you such question and you don't have any idea, you can go back in the passage, most especially these questions which are involving of reading, go back, read again, and then identify. Then they're saying that draw a bar graph. Before I go to the bar graph, can you identify the independent variable here? You can identify the independent variable here. So is the weed dependent on the herbicide, or the herbicide depend on the weed? It means that the weed will depend on the herbicide over a period of years. That's why here you see that over a period of time or years. That's why you see here that glyphosate resistance, the resistance will accumulate based on the period of years, meaning that this herbicide will be resisted based on the period the weed has stayed in contact with the herbicide. Therefore, the resistance depends on the contact time it has stayed with the herbicide. Hence, resistance becomes the dependent, and then the period of the contact becomes the independent. Okay, the bar graph. Now here, when you talk about the bar graph, you need to know the things we are looking for so that when you go to the exam, you don't miss any question concerning about the bar graph, concerning about the graph. The first thing you need to know is the caption. Sometimes some teachers don't explain, they just mark, they put a T, they put a C, they put a S. They don't explain this to their students. But what does it mean? How do you answer this question so that you don't skip any mark? All right. So now they're saying that the bar graph should illustrate the results of investigation in the first 4 years. So, okay. Now, I'm not going to draw the actual graph, but I'm just going to talk about how you can draw this bar graph without stress. So years are always an independent variable, meaning that this is time. Time is always an independent variable. So independent is always on x-axis. Independent is always on the x-axis. And then the dependent one is always on the y-axis. I always tell my students that if you forget which one is on the x-axis, which one is on the y-axis, you just write here the word ID, your ID. Ne? Yes, ID, which means that you know the ID you are using. Independent, dependent. So this one is going to go on the x-axis. Then you here put ID, X, Y. So this one is going to go on x-axis. This one is going to go on the y-axis. So that's how you can remember the x-axis and the y-axis. So you just do this small box. I repeat. Ne? You say ID, X, Y. So meaning that ID, independent variable on x-axis, dependent variable on y-axis. The first column on x-axis, the 2 or 3 or 1 goes to the y-axis. All right. Now, if you are drawing a bar graph, make sure that you draw a straight line. Don't mind about mine. Straight line. This is how we mark. You write a caption. Never forget about the caption. So we give you a tick for the caption. Then after that, you write the X, you write the Y here, you write the X-axis here. But when you're putting the value here, for example, in this case, they are saying yeah, and then the percentage, the glyphosate resistant weed percentage. So it means that the resistance is going to be here. Yes, the resistance is going to be here. And then units, it is in percentage. Then the time is going to be here, which is year. And then the years, they don't put anything. So if you just say time in years or just year, then it's fine. It means that now they're going to give you another tick for labeling the what? Labeling the scale. Ne? Not labeling the scale, labeling the axis. So if you label the axis X, they can give you a tick, or Y, they also can give you a tick, or they can combine the two. Now, you go to, after that, you go to the intervals. Please don't start with a 0 here, yet you want to start. If you come back here on the You'll see that is 10, 20, 32. Okay, this one, it's fine. You can start with a 0. But if it is starting from maybe 100, and then you go to 120, you'll find out that some people, they start here 0, 100, 120, 140, 160. So the difference between 0 and 100 is not the same as from 100 to 110. So if you start from 0, make sure that, and you say a difference of 10, it must be 10, 20, 30, 40, 50, 60. It must be consistent. If you don't do that, you're going to lose a mark. And then now you're going to have to draw your bar graphs. It must be straight. Don't forget that these bars must be straight. It must be straight. It must be straight. Ne? So now the distance between these bars, I'm saying this is a rough distance between this space and this space must be equal. It might not be exactly the same because you don't use a ruler when you're marking. No, but when you look at with your eyes, you see that these distances are the same. So it must be equal. These bars, the size must also be equal. So that we give you, if the scale, the interval is the same, we give you a mark for the scale. Yes. If you have drawn the bar graph, not a histogram whereby the bars are connected to each other, then we give you correct type of the graph. We give you a correct type of the graph. We give you the mark for the caption. Then we give you a mark for labeling the axes. We talked about it. Then we give you a mark, maybe 3 of them. We give you a mark for plotting if you have plotted this correctly. So that's how we mark the bar graph or any other graph of any kind of nature. So basically, that's how we mark. Then the last question is natural selection and also out of Africa hypothesis. Still here, I'm not going to answer the actual, like the way it is here. I'm just giving you the ideas how you can answer these questions without stress. Because when you are bringing the question, we can bring it in a different version. Yes, we can bring it in a different version so that you don't lose those marks. So when you modify, where do you modify and how do you modify it? A population of lizards on the island A were well situated to feed mainly on insects. So it means that these were feeding on insects. Scientists moved the 5 other of the lizards species to the neighboring islands. And they are saying the island A, island B, here they reproduce and a new population formed. They're saying the island B has large supply of plant with the fibrous leaves and fewer insects. It means that now the food has become scarce. So this one is going to become a problem. Yes, exposure to this insect new environment may have caused the lizard to undergo evolution. Okay, they're saying 63 years later, scientists returned to the island B to conduct a further investigation on the lizard population there. They observed that the jaw size of the lizards had increased, and scientists also analyzed that the stomach content of the lizard found that it was mainly plant-based. They confirmed that the 2 populations still belong to the same species. They confirmed that the 2 populations still belong to the same species. Describe how scientists could confirm that describe how scientists could confirm that there was a change in the jaw size between the lizards of island A and the lizards of island B. So the question might not come like this, explaining this, but it can come in a different version. So we shall go and then see exactly where we need to have the answers from. So basically, what the scientists could use to confirm or could confirm that there was a change in the jaw this is an obvious answer. It means that they had to measure the lizards on the island A and the lizards on island B, and they check whether the jaw size is the same, and they can compare the differences if the difference is there. Then explain how the large jaw of the lizards on island B would be structurally suited to eat the tough fibrous. Explain how the large jaw of the lizard on island B would be structurally suited to eat the tough fibrous leaves. Yeah, this could be about the jaws being strong, meaning that the muscles are strong enough, and also the teeth, so that they can have a strong bite, and then they can chew that food. It means that the muscles, to do that, it must be strong, so that the force of grip is high, and then they are able to chew this kind of food. How did the scientists determine that the two populations of the lizards, both on island, still belong to the same species? Don't forget that a population, a species, these are organisms that can interbreed to produce a fertile offspring. So if they have the two populations and they try to allow them interbreed, and they produce a fertile offspring, therefore they can confirm that these are still the same species. But if they cannot interbreed to produce a fertile offspring, then automatically they are not the same species. But if they could interbreed and produce a fertile offspring, therefore it means that they are still of the same species. Explain the possible effect that evolution of the lizard has on the biodiversity. Explain the possible effect that evolution of the lizards has on the biodiversity. Here, it could be, it can increase the biodiversity or it can reduce the biodiversity. So basically, those are the two ways. If it forms new species, then automatically it has increased the biodiversity. If it takes away an existing species, then it forms another species, then it means that it is decreasing the biodiversity. However much it's creating, but it's decreasing because the other one is no longer existing. Or it could be 3 species, and then because now the food has come in a different angle, the 3 species, they modify into 1 species, or they modify and the other ones, they become extinct. They become extinct, meaning that now they have also decreased the biodiversity. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Here, the principle is the same. The principle of natural selection, whatever you explain, use the same principle. The first point you have to talk about is variation. There was variation among, now you have to tell us what they gave you, among the lizards, those with the large jaws and those with the smaller jaws. Because the jaws are used to bite or to eat this tough food, therefore, those lizards with big jaws, they could eat enough food, tough food, therefore they survived and reproduced in large number. Those with the small jaws, they could not eat enough food, therefore they died in large number. And this is called natural selection. Yeah, previously we are giving a tick on natural selection. This is called natural selection, but the chances are high that we no longer giving this marking point of this is called natural selection. But put it there, you never know that maybe we can review about it. This is called natural selection. So those with big jaws, they pass their genes to the next generation because they large number. After many generations, all the lizards have big jaws. There is variation, I'm repeating, there is variation among the lizards. Those with, you tell us the variation. Those lizards with large jaws and those with the small jaws. Those with the large jaws, they could eat the tough food. Yes. and then they survived, they reproduced in large numbers, they passed their genes to the next generation. While those ones with the small jaws, they could not survive, they could not eat enough food, therefore they died in large numbers, and this is called natural selection. Those which survived, they passed their genes to the next generation, and after many generations, all the population of the lizards were made up of large jaws. That's how you can explain using Downey's theory of natural selection to show how this evolution of large jaws came about. Lastly, on this paper, please, you should never go to the paper without knowing these questions. The out of Africa hypothesis, which states that modern humans originated from Africa and migrated to the rest of the world. Don't forget the word modern. Modern. However much they might not give it as a ticket marking point, but in most cases, we criticize when the word modern is not there, because not all humans are modern. Homo habilis were humans. That's why you call it Homo, which means a man. Homo erectus were humans. That's why it's called Homo, is a man. Homo sapiens is a human. That's why you call it Homo, which means a man. So Homo habilis, the upright, the handyman, Homo erectus, the upright walking man, Homo sapien, the wise man, those are, all of them are humans. That's why we are referring to modern humans. So please put the word modern humans, Homo sapiens, which means the wise man. I don't mean the school, I mean the man, you who is listening. Someone's going to ask me, what about us who are ladies? Yeah, is a man in science. All right. They're saying that name the family to which modern humans belong. They belong to a family called Hominidae. Hominidae. So they belong to a family called Hominidae. Yes, that's the family to which it belongs. They're saying that which genetic evidence is used to support the out of Africa hypothesis. We have two genetic evidences, but we only teach one. We have what called mitochondrial DNA and Y chromosome. But now we don't talk about the we don't talk about the Y chromosome. Only now the syllabus or the curriculum is talking about the mitochondrial DNA. So the evidence we give you here is mitochondrial DNA. Describe how fossil evidence is used to support out of Africa hypothesis. I've been telling these kids that these are the same questions we ask you. And if you check the book, our book we use, you can download even yourself. That's exactly how we mark exactly. This is the same way how they ask you this question. This book was released a long time ago before even these papers are being set. All right, let me answer. You have to talk about the oldest fossils which were discovered in Africa. For example, you have to talk about that Adipithecus remedus was discovered in Africa. You have to talk about that Australopithecus. Among Australopithecus species, we have Lucy, that is Afarensis. We have Taum child, that's Africanus. Mrs. Place, Africanus. Little fruit, Africanus. Carabo, Sediba. All those Australopithecus species were discovered in Africa. Homo habilis, also in itself, was discovered in Africa. That is the handyman. Now, when you reach here, you add the word the oldest. The oldest species, the oldest fossil of Homo erectus and Homo sapien were also discovered in Africa. That's what you want. So you may not talk about all the Australopithecus species, all of them. You can just talk about one or two. So Adipithecus discovered in Africa. Australopithecus species discovered in Africa. For example, Taum child, Mrs. Place. Then you'd go to Homo habilis. discovered in Africa. Homo erectus, the oldest Homo erectus and Homo sapien discovered in Africa. Let me show you what I'm trying to say. This is what I'm trying to say. Evidence to support out of Africa hypothesis. There are three: the mitochondrial DNA, the cultural evidence, and the fossil evidence. This is what I'm trying to say. That fossils of Ardipithecus were only found in Africa. Fossils of Australopithecus, e.g. Karabo, Littlefoot, Omchild, Mrs. Place, all were found in Africa. Homo habilis were only found in Africa. Oldest fossil of Homo erectus and Homo sapiens were discovered in Africa. This shows that modern humans originated from Africa and migrated to the rest of the world. You see that even I highlighted the key points which you need to know when you're writing in your exam. Yes. So as I'm winding up, I will recommend you to use this book. Go to our website, download it free of charge. If you want to be printed, that's a different story. But go use these resources. I took time to write this book, and I wrote it with the care so that every child of this country in South Africa must pass. You don't have a right to fail. You have a right to pass. You must demand for your rights. What is your right? The right of passing. Please, never ever demand for the right of failure, because you have the ability for you to pass, not the ability for you to fail. I wish you the best. Make sure that you go through these papers. You will make it. Thank you very much. Don't forget to subscribe, like, and share to others so that they also benefit in this before they write their final exam. Thank you very much. M.Saidi, as usual.
Okay, let's look at variation. So differences among organisms of the same species, we call it variation. So when we talk about variation, we are talking about this organism. What are some of the causes of variation? These causes could be genetic, or it could be environmental. You can be tall not because you are genetically tall. It could be environmental. You can be short not because you are genetically short. It could be environmental. Haven't you ever seen father, mother, both short, but they produce a tall kid? It could be genetical, yes, or it could be environmental. Oh, dark skin, or light skin, they produce a dark skin, and then the father said, this is not my kid. Who told you it's not your kid? It's whose kid? So it's your kid, but some environmental factors could happen, or some genetic factors could also happen. When we talk about a species, it's a group of organisms living together. And these organisms must interbreed to produce a fertile offspring. I've tried to explain this. A group of living organisms consists of similar individuals, meaning the same group, capable of interbreeding. Interbreeding is sex, having sex and produce kids. That's interbreeding. Right? Courtship, you do the courtship and then you produce. I hope everyone is above 18. To produce fertile. Fertile means something which can reproduce again, or viable offsprings. Offsprings are babies. We saw them when you're talking about genetics. And then a population, if you have dogs, a lot of dogs in the same area, specific time, then we say that a population of dogs, population of plants. So when we talk about a population, is a group of organisms of the same species. They must be living together and they must be able to interbreed. So if they can interbreed, then it means that it's just a population of those organisms. The reason why I've talked about the species and population, because variation happens in the species, or it happens in the population. That's why I try to do a recap of population and species. What are some of the causes of variation? Number one, crossing over. You remember we talked about crossing over. When we talk about crossing over, homologous chromosomes come together during prophase. prophase 1, you must be specific, prophase 1, and then the chromatids of the homologous chromosome, they cross over. When they cross over, you'll find out that they produce chromosomes with mixed genetic composition. You find out that this part came from this chromosome, while this part came from this chromosome. So this one came from this, and then this one came from this. So it means that genes have been mixed. That's why you have the father, you have the same mother, but you have different people. They appear, they don't even look alike. Sometimes you go even somewhere and they say, like, hey man, you resemble someone I know there. Does it mean that your father went to that lady and then impregnated her and without knowing? No, it's because you share the same ancestor. Then it means that you still have those genes within us. So crossing over, it helps in when homologous chromosomes from form pairs during prophase 1. I said prophase 1 is very important of meiosis. Crossover can occur as you see it here. Crossover occurs, as I've tried to explain, leads to new combination of genetic material in new cells. So if they separate during anaphase, they will go to different cells. Then it means that these cells, they will have different genetic material compared to the parents. If parents had this, then automatically the offspring will have this. So it means that you will have some characters from the father, some characters from the mother, but not all characters that will be the same, exactly like the father. So crossing over helps in bringing about variation. Number 2, random arrangement of chromosomes. How do chromosomes randomly arrange? If you look at this, you'll find out that you can have this, maybe. Remember, they just do randomly. They are like chicken when they are going to mate. Okay, I'll talk about that. For example, we don't know whether this chromosome is going to be on this side or is going to be on this side. It can be like this color, and this color, or it could be like this. So we don't know which one is going to be, which one is going to be this side, this one is going to be this side. And remember, here we're just showing 4 of them. They are not 4 different positions, as long as they are homologous chromosomes, but they have different positions. So you end up having different combination of, for example, in this case, you're going to have this, you're going to have this. In this case, you're going to have this, you're going to have this, and then you have another combination, you have another combination. So you end up having many combinations. So random arrangement of chromosomes during metaphase result in the gametes with unique. You'll find out that you have unique combinations of what? Of alleles. At the equator during metaphase allows different combinations of chromosomes or chromatids to go into each new cells, resulting in meiosis, making them different. So if you look at this sperm will be different from this sperm, and this sperm will be different from this sperm. All these, if you look at them, all of them, they are different, but it came from only 2 combinations, but you have many combinations. You have many combinations. So because of this, it will result, and you don't know which sperm is going to fertilize the ovum. So because of this, then it helps in bringing about variation. And then another one is random fertilization. Look at this egg, and look at these sperms. Look at these sperms. All these sperms, they are looking for this ovum. And we don't know which one is going to fertilize the ovum. You see, we don't know. So if it is like that, it is by chance, it's random. All these sperms are different, but we don't know which one is going to fertilize this. So if the guy produces like 100 million sperms, and then only one, and each sperm is different from the other, only one will fertilize the ovum. So what does it mean? It means that this time it will be a different sperm fertilizing. Another time it will be a different sperm fertilizing. Obviously, we will have different genetic makeup, and then we will have what you call variation. We will have some differences among us. That's why you have different colors, you have different heights, you have different sizes, you have different length of hair. You have why? Because of the genes are being mixed up during these processes. So you're saying that any sperm stand a chance of fertilizing an egg or cell. You see how fast your sperm was. I told you that you were lucky. How fast your sperm was to fertilize the ovum, so that you at least you can exist. If your sperm did not fertilize an ovum and then another sperm fertilized, you think you would have existed. Someone else would have existed, not you. But your sperm was smart, was faster, was everything was perfect, and then it was able to fertilize the ovum, and then you are existing. So you're saying between different ovum and then different sperm cells formed by meiosis, resulting offsprings that are different from each other. We saw that you have different sperms. Each sperm each cell is different from the other. So it shows that random fertilization can help in bringing about variation. Note, each individual is capable of producing over 8 million sperm. You see? Hey, do you know the meaning of 8 million sperm? If they give you 8 million rand, are you going to be rich or poor? Obviously, you're going to be rich. So now imagine that is sperm. Yeah? 8 million sperm are potential. When you talk about potential, it means that those which can fertilize, because we have other sperms which cannot fertilize. But random chance of any one sperm and egg coming together is a product of these two possibilities. Look, 70 trillion different combinations. That is the chance. You see how it means that you won a barrel of 70 trillion. 70 trillion. Even the budget of South Africa might not be 70 trillion. I don't know. I'm not a politician, but it might not be a 70 trillion. This is a lot of money. It's a lot of money if it's terms of money. But sperms, you have all these chances, but your sperm was able to fertilize. So it means that random fertilization can bring about variation. So it is also a cause of variation. Let's look at random mating. Random mating, you just mate whoever comes to your face. For example, if you are chasing this one and then someone comes, crosses your eyes, then you leave the other and then you go with this one. That is a random mating. Whatever comes to your face is what you mate. I think the people who make a random mating are the so-called, they call them mahoshas. They do random mating. Whoever comes, they go. What they're about is money. So that's what you call random mating. So mating takes place within species randomly. If you look at this, yes, this can mate with this can mate with this can mate with this. There is no restrictions that no, Mina, I want a tall, big, no. With this, no. Whatever comes to your eyes, you just go. That's what you call random mating. Between organisms within a species lead to different set of offsprings from each mating pair. So if I mate this with this, different set. This different set. Mate this with this different set. So random mating also helps in bringing about variation. And then lastly, we have what we call mutation. Mutation means change in the genetic composition of an organism. This could be chromosomal. It could be a chromosome changing, or it could be the whole chromosome changing. Number of sets could change. For example, Down syndrome, you have an extra chromosome, so chromosomal mutation. Or it could be a gene mutation, whereby just a specific part of DNA just chops off or is inserted or is substituted, something like this. If you look at this, this is a substitution. This is a DNA. What happened? This was here. You see it is this color, the same color. But what happened? This cut and then another part joined here. So it's also a mutation. This will result in a different organism. These mutations be beneficial or could be a lethal mutation. So changes the structure of gene or chromosome, and therefore the organism's genotype changes too. The moment you change the genotype of an organism, then automatically you change the composition, the phenotype of an organism. Remember, the genotype the genetic composition, what is inside your DNA, determines what is outside. How beautiful you are is determined by the genetic makeup. Not the makeup. No, not makeup. No. It's the genetics of your what is inside. Your manners, the way how you respond, the more how you think is being determined by this. However, it could also be environmental. It creates organisms with new, different characteristics from one generation to the next generation. So if there is gene mutation, if there is any mutation, if there is any change in the genetic composition of an organism, it will result in different organisms, which we call variation. Because the moment the organism phenotype has changed, then we say that variation has taken place. So mutation could be, number one, harmless. It can result in harmless. It has no problem with you. For example, you have an extra finger here. It's not changing your physical abilities. So it could be harmless. It could be harmful mutation. This one, it can even cause death. Albinism, it is a mutation, but it is harmful. How harmful? Because you cannot sustain yourself in sun. If you are original indigenous, like someone you know, yes, it means that, then it means that, yeah, you are strong. But if you don't have it, then automatically what happens? It means that you have a mutation, and then you won't be able to walk in the sun. And then you have the beneficial mutations. They can make you more advanced. Some of you watched these X-Men movies. Okay, that is a movie. But X-Men stuff, yes. They have beneficial, maybe this one can fly, this one can do this. Those are mutations, but beneficial to them. A mutation can be beneficial, or it could be harmful, or it could be just there without any problem with you. This variation could be a continuous variation, or it could be discontinuous variation. So let's look at continuous variation. Continuous variation, this is the type of variation where there is a range of characters. There is a range of characters. There is intermediate values. So if you look at this graph, you'll find out that you have a number of people, number of characters, and then the extreme ends, you have a number of them still, but the number of individuals which have the intermediate value, they are more than the two extremes. For example, if you look at height in humans or size, you'll find out that people who are too small, they are very few, and people who are too big are also very few. But people who are just in the medium size, there are a lot. So that's the meaning. That's why you see that at each extreme end, the graph will lower down, and then as you continue to the medium size or medium value, then you find out that the number is increasing. So if you skip or you pass the medium size or medium value, then you go back to the number dropping down until you reach 0. So basically that's continuous variation. We see that examples of this you can find out. You have the size, you have the height, you have the skin color. You have some people who are too dark, some people who are light, others they're intermediate. But you find out that those who are too dark and those who are too light, they are few. But those ones who are intermediate, you will find them that they're a little bit many. That's why you see them here, the same story. So this continuous variation can be controlled by environmental factors, or could be controlled by genetic factors. So, but the environmental factors has a big influence on that. However, even genetic factors are also part of it. For example, if you look at size among individuals, if you eat miserably, it means that you're gonna become fat. Therefore, it is environmental. It's not that you are born like that. It's because the way how you behave yourself, then it causes that you gain weight. So such characters which can be controlled by environment or influenced by the environment, then can be categorized as continuous variation. Okay, then you have what you call the discontinuous variation. When we talk about discontinuous variation, this one, the environment has a very small impact on it. It means that it's not being controlled by the environment. So in this case, you have characters that you find in the population has no intermediate values. For example, blood groups, you have blood group A, B, O, O. So you cannot have these blood groups. You cannot have blood group. You cannot be blood group A and you are blood group B. No, you cannot be in between there. No, you are either A, B, or O. You cannot be halfway or halfway A, halfway or halfway B. No. You either A, you are B, you are A, B, you are O. So it means that there is no intermediate in between there. But if you look at height, you'll find out someone is too tall, someone is next to there, another, the next height, like that. But for the case of blood group, it's not like that. Another example is tongue rolling. Yes, you can either roll the tongue if you try to see exactly. Some people, if you roll, you can roll the tongue. Né? If you do like this, you'll find out that some people can do the tongue. They can roll the tongue, but others cannot roll the tongue. You either roll it or you don't roll it. So that is the meaning, that if you are discontinuous, then it means that you cannot, you are either this side or the other side. So these characters are being controlled by the genetic makeup of an individual. So if it is genetic makeup of an individual, then it means that these individuals, they either belong to one group or they belong to another group. The environmental factors cannot influence you. It cannot influence you to be blood. If you're blood group A, you cannot be blood group AB. If you are blood group O, you cannot be blood group AB because of the environmental factors. No, if you are born blood group O, you're gonna die blood group O. So if it is like that, then we say that the one which are being controlled by the genes, then we say that that is discontinuous variation. So if you look at this graph, you'll find out that you have A, there is no one here. There is no one here. You have B, there is no one here. You have AB, there is no one here. So it is discontinuous. It is not continuous like height. If you look at height, if you look at height here, you'll find out that for it is continuing.
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
We are saying that genetics, when we talk about genetics, we are talking about what you call genes. Small sections of DNA, and these sections, they have a certain character they are controlling, is what you call genes. So the study of inheritance, the study of these genes is called genetics. Basically, in this topic, these are the things you need to cover. I'm going to cover the following subtopics in this topic. First one is concepts of inheritance, the terminologies. You need to know the terminologies. Monohybrid crosses, crosses which involve one character. Mono, one character. Sex determination, what is the percentage chance for you producing a girl or a boy? How do you determine the sex of a kid? Sex-linked inheritance, the characters which are linked to the sex. Blood grouping, which type of blood group, and then inheritance of blood group. Dihybrid crosses, when we talk about dihybrid crosses, we mean inheritance of two characters. Di means two. Genetic lineages or pedigree. These ones are tracing characters in your family. In most cases, in this case, you're going to be looking at the disorders which can be inherited by the family. So you have mutation. When you talk about mutation, it means change in a part of DNA. It could be the whole chromosome can change, or it can be just a small section of DNA changing. So we're going to look at the mutations. Genetic engineering. Genetic engineering, it means that you're trying to manipulate. So you try to manipulate with the genetics or with the genes of an organism. Then you have a paternity test. Here you're trying to do the testing of the parents, father, mother. If there is any subtopic you don't understand, then it means that you are still lacking, you are susceptible to lose marks because you don't know which part of genetics they are going to bring. We can bring any part from genetics. So you don't know exactly which part they're going to bring. And if you don't study that part, then it means that these marks which are allocated on genetics, you have lost some marks even before you go to the paper. However, by chance, you can find out that the part you don't know, it has not come. And then you can answer those which you know. But I don't encourage that. So let's look at the introduction. Father of genetics. When we talk about father of genetics, we are talking about Gold Mendel. He did a lot of work on inheritance, and he discovered a law which we call law of inheritance. He concluded a gene come in pairs, like when they are, for example, height is being controlled by two parts of genes. When we talk about genes come in pair, a character like, for example, height, size, skin color, they're being controlled by two kinds of genes. So they come in pairs and are inherited as distinct units. It means that one character is not interfering another character during inheritance. Each character is inherited differently from one from the father and one from the mother. It means that each parent is supposed to give one part of the character. Remember, there are supposed to be two, so it means that the first part comes from the father, the second part comes from the mother. That's what he is saying. So let's look at some of the concepts of inheritance, meaning that the terminologies. So chromatin. When you talk about chromatin, it's a substance within a chromosome containing DNA and a protein. Basically, when you talk about a chromatin, chromatin is DNA. But in this case, this DNA is like threads. Yes, it is in coiled form. When it coils, then it forms what you call a chromatin. Then number two, you have what called chromosome. Now, when the chromatin condenses, when it shortens, when it becomes thick, it forms what you call chromosome. So chromatin and chromosomes, they are the same thing, but in different versions. So you can say water in ice form and in liquid form. So it's the same thing, but it's in different phases. So we are saying that thread-like structures of nucleic acids and are found in the nucleus carrying genetic information. So chromosomes are the thread-like, they are like threads crossing each other. And these chromosomes, they carry what called the genetic material. You look like your father because of the genes your father gave you. Then, if you look at this structure, look at this, you might think that this is noodles. Yeah, you think that these are noodles or spaghetti. Yes, but if you condense it, yes, so if you shorten it, you squeeze it, you condense, it forms what you call chromosomes. So when they cross, they look like this. So when you talk about chromosome and the chromatin network, they are the same, but in different phases. You see, you can see that what is here is almost what is here. Small ingredients just to make them to be in this shape are being added, just to make them to be chromosomes. But what they carry inside, the most important thing, what do they carry, is the same information as chromatin network. But they form chromosomes during cell division. What about genes? Chromatin network, chromosomes, genes. Genes are just small sections on the chromosome. So if you just break down the chromosome or the DNA, you form different portions. Those different portions, they form what called genes. And then you have, we are saying that this is a small portion of DNA coding or controlling a particular character, or a particular protein. So it must be these characteristics you have. You are dark-skinned, you are light-skinned. What causes that? It is genes. And then when we talk about allele, alleles are genes. Alleles form chromosomes. Alleles form chromatin network. But chromatin network, if you look at chromatin network, you look at chromosomes, you look at genes, you look at alleles, they are the same things. All of them is the same thing, but at different scale. I'm gonna give you an example. When you talk about that, where do you stay? You say that I stay in South Africa. Where exactly? Then you tell me that I stay in Northwest. Where exactly in Northwest? You tell me that I stay in Rustenburg. Where exactly in Rustenburg? So you keep on reducing the scale. Chromosome, genes, alleles, they are the same but at different scales. So when you talk about allele, it's an alternative form of a gene. You can be tall or short. So an alternative form is called an allele. So we are saying that alternative form of a gene found at the same locus. Locus means the location of a gene, the place of a gene, the position of a gene is what we call a locus. We're gonna bring these things in terminologies, and then please don't forget to give a correct answer. So here is a chromosome. A chromosome is here, but you have different parts, different locations of these chromosomes. So these parts is what you call the alleles. So these alleles may be for height, for size, for color, for blood group. They are supposed to be found on the same position of the chromosome. This side and this side is the same thing. So we call them alleles. So are alternative forms of gene. Then we talk about a dominant allele. So because you have two forms of alleles, one can be dominant, another can be recessive. And both of them can be dominant, or both of them can be recessive. So when we talk about dominant, it means something which suppresses another thing. Yes. So we say that an allele that is expressed in the phenotype when found in heterozygous state. When we talk about heterozygous, it means in different phases. One is like this, one is like this. So it means that this capital letter suppresses this. That's why I say that a dominant allele is that one which suppresses another allele. So this capital letter will suppress the small letter. Then what you call the recessive allele. This one can only express itself in absence of a dominant allele. So if this one is absent, then automatically this small t will also express itself. So we are saying that an allele that is masked in a phenotype when found in a heterozygous state. So it can only show itself in a homozygous. It is only expressed in a homozygous condition or homozygous state. Here is an example. You have homozygous dominant. It means that, you see, they are the same. Homo, I say that when you talk about homo, means the same. Zygous comes from what? Zygote. So it means that this zygote has the same kind of allele. Heterozygous, it means that this zygote has different kinds of alleles. So you have capital letter and small letter. And then homozygous, still the same type of alleles, but these alleles are recessive. So it means that it is the same kind. Homo, which means the same. So same, but they are supposed to be recessive. Then when you go to phenotype, when you talk about phenotype, it means the physical appearance of an organism. You are expressing an individual in terms of what you see. In most cases, when you bring a question concerning about a cross, they're going to ask you the phenotype. The first question, the first part, you are supposed to write the phenotype. We get these answers from the question. Is it clear? So we are saying that physical appearance of an organism is determined by the genotype. For example, tall or short. You can be tall, you can see that a person is tall. You can be short, you can see that a person is short. You can be light-skinned, a person is light-skinned, you can see them. And then what about genotype? Here you are describing an organism in terms of what? In terms of genes, genotype. The gene type, which kind of gene type do you have? So here is genetic composition of an organism. So you know you are tall, but which kind of tallness? You might be tall, but homozygous tall or heterozygous tall. You have an allele for shortness. You can be short, but you have an allele for tallness. You see two people who are short, they produce a tall kid. Two people who are dark-skinned, they produce a light-skinned kid. Two people who are light-skinned, they produce a dark-skinned kid. And then a guy can say that, yeah, this is not my kid. It's whose kid? All right. So here is another example. When we talk about this, describing this in terms of the genes is what called the genotype. Then also this is the genotype, and also this is what? Genotype. So I will say that this is homozygous dominant, this is heterozygous, and then this is homozygous recessive. So I'm describing these flowers in terms of genetic composition. So then when we talk about homozygous, I've been talking about this. Homo, which means the same zygous from the zygote. Then it means that this zygote has the same kind of genes. Two identical alleles for a particular characteristic. Homo, same, can be homozygous dominant or homozygous recessive. When you talk about hetero, when you say hetero, it means different. So the zygote has different kinds of alleles. For example, capital letter B and then small letter b. So they are different letters or different alleles. If you look at this picture, you'll find out that homozygous, you have this. They are found on the same locus, same location. This one and then this one, and then this one and this, you see. While if you look at heterozygous, same location but different letters. Same location but different letters. So this is homozygous recessive, this is homozygous dominant, and then this is heterozygous. For the case of heterozygous, we don't have recessive and dominant, we just say heterozygous. A monohybrid cross is a cross which involves one character. You are right, it involves one character. But now you said pink and white. It still is fine. The answer was correct. But where is the character there? The character is color. That's the character. But this character has two alternative forms. The blue you talked about and the white you talked about. It's either blue or it's white. So those are the what called the alleles. So the character is the color of that individual you talked about. And then alleles are the different types of colors you have talked about. You understand?
Welcome back to our class. Yeah, today we are going to be looking at the process of protein synthesis. What is protein synthesis? So protein synthesis is divided into two words. Number one, it is the protein and the synthesis. When you talk about synthesis, it means manufacture, production of proteins. Protein synthesis is the process by which proteins are made in the cell of an organism to form enzymes, hormones, and the new structures of the cell. So your body is made up of different structures, and these different structures are made up of protein. So it means that if you don't have protein synthesis in your body, then it means that you can't grow, you can't have these hormones, you can't have these enzymes. So the marking point here in the definition is the word proteins are being made in the cell. This process is divided in two different phases. The first phase is called transcription. And when you talk about transcription, it means formation of messenger RNA. Then number two is translation. Translation is the formation of a polypeptide chain. The difference between polypeptide chain and the protein is almost the same, but it depends on how many amino acids are there. We are going to see the amino acids in detail. So how many amino acids are there? If more than 50 and above amino acids, then we call it a protein. But if it's less than 50 amino acids, we call it a polypeptide chain. This diagram shows the whole phase of protein synthesis. This is the nuclear membrane. These are nuclear pores. So we have gone into the nucleus, and then you have gone into the cytoplasm. So this diagram you see, it's just showing you the nucleus and then the cytoplasm. We know very well that the DNA is found in the nucleus. And then this DNA, because it is a king, is the one which codes for the protein, doesn't move out of the nucleus. It's like a king who doesn't move out of the palace. He has the servants, and the servants, they go take information, what the king has said. So these messengers, the transfers, they act as servants for DNA, because DNA wants to produce a certain protein. So it will send the messenger, and then the messenger takes the information to the cytoplasm, then the transfer RNA will bring the amino acids. So when they bring a diagram like this, you have to know how to label. That's the first point you need to know so that you can pass this question. The first thing is you have to know that this is DNA. When it is coiled, if you see something coiled, is DNA. If it is too coiled, then we call it chromatin network. Then the DNA is done which unwinds, it forms the messenger RNA, and then the process of formation of messenger is what we call transcription. Then this messenger RNA will move out of the nucleus via the nuclear pore, and then it goes to the ribosome. You see, these are the ribosomes. This messenger will align itself on the ribosome, and then the transfer RNA will bring the amino acids to this ribosome, and then a chain of protein, a chain of amino acid is formed. Therefore, this process whereby the messenger brings in and then the transfer RNA brings in the amino acid also, the chain is formed, is called translation. So the major difference between transcription and translation occurs in the nucleus, while translation occurs in the cytoplasm. So you have to know at least the basics of this process. Before we go to the process of transcription, so this is a simple diagrammatic representation of the whole process of protein synthesis. So you expect DNA to unwind, and then when it unwinds, it forms it exposes two strands, but one strand will act as a template for formation of a new strand, and then this new strand is what we call the messenger RNA. This messenger RNA, we saw that it moves out of the nucleus via the nuclear pore, and then it goes to the ribosome. The moment it reaches the ribosome, then it starts the process of translation. Then amino acids are joined together, are brought by the transfer RNA. They are joined together to form a polypeptide chain. So if the number of amino acids become a lot, the more than 50 and above, then it forms what you call a protein. So the protein, it must have is a heavy molecule compared to a polypeptide chain. So now let's look at what happens in transcription. We see in the exam, what do you write when we find such questions, and then what happens in translation. Transcription. Don't forget, this is a record that transcription occurs in the nucleus. You have the DNA, and then you have the DNA to unwind, and then messenger RNA is formed. Then messenger RNA moves out of the nucleus by the nuclear pore, and then it goes to the cytoplasm. Basically, that's what you're going to be explaining. So the first point, what you see in different color, it means a marking point. DNA double helix. You have to tell us the natural shape of DNA. DNA double helix unwinds. So that's the first step. Number two, it will say that when the weak hydrogen bonds break. Here, when you say weak hydrogen bonds break, or you say DNA double helix unwind, then it unzips. It's the same as saying weak hydrogen bonds break. What's the meaning? If you look at your trouser, you try to open it, you zip down. When you zip down, you are breaking the bond which we are holding the zip together. You understand? So when you say when you unzip, it means that you are breaking down the bonds which are there. That's why we say that weak hydrogen bond break. Or you can say DNA double helix unwind, then unzips. So you get the same mark. One strand will act as a template. There is a major difference between DNA replication and transcription. Here, when you're talking about transcription, one strand is the one which acts as a template, while the other side, the two strands will act as a template. So something which look like a mirror, so that there is one strand, so that the new strand is being formed. Then to form the messenger RNA, one strand will act as a template, as you see it here, and then messenger RNA is formed. It uses free-floating nucleotides. When you say this, you are supposed to specify, because when you say free-floating nucleotides, in the nucleus, there is free-floating nucleotides of DNA and free-floating nucleotides of messenger RNA. So you have to be specific when you come to this. You have to say that free messenger RNA nucleotides. Yes, so these free messenger RNA nucleotides which are floating in the nucleoplasm are the one which we use. Yes, so if you just say using nucleotides, we will cross it, because we have different kinds of nucleotides in the nucleoplasm. So you have to be specific that free messenger RNA nucleotides. Then the nucleotides, the messenger RNA is complementary to DNA. So it means that where there is A, you're going to have U, and where there is C, you're going to have G. Remember, the messenger RNA does not have thymine. It has what you call uracil. That's why we say A goes with U and then C goes with G. Then the messenger RNA now has coded message of protein synthesis, which means that now it is ready to move out of the nucleus and then go to the cytoplasm, so that it can take the information DNA wants the protein to be manufactured. Yeah, that's why you're saying that now the messenger has the coded information for protein synthesis. Then we are saying that the messenger RNA moves from the nucleus through the nuclear pore, and then it goes where? To the cytoplasm. Where exactly in the cytoplasm? That is on the ribosome. So it basically, this is the general process of protein synthesis. Basically, that is transcription. So what you see in different color, it means the marking point. DNA double helix unwind, weak hydrogen bonds break, and then one strand will act as a template, and then for formation of messenger using free messenger RNA nucleotide in the nucleoplasm, and then the messenger RNA is complementary to DNA, whereby A A goes with U, and then C goes with G. And this messenger, which is formed, has a coded information for protein synthesis, and then this messenger moves out of the nucleus via the nuclear pore to the ribosome. Basically, that's what we want. 6 marks from here. In most cases, you see it as a short question, which requires you to write these statements the way you see it. Please don't change. If you change, make sure that the different colors don't change, because those are the marking points we are looking for. And then let's look at the process of translation. Here, let's look at the process of translation.
Welcome back to our class. So today we are going to be looking at punctuated equilibrium and gradualism. So we are going to compare the two and see where do they differ. Don't forget to subscribe if it is your first time to visit our channel, and don't forget to like our video. So if you look at these two diagrams, one is showing a punctuated equilibrium and then another one is showing gradualism. Gradualism, it shows that a species change gradually. They change gradually over a great period of time. There is no rush in the changing. They change from this point to this point, but the change is gradual. It is not fast. It's just a gradual change. From this point, you see that this rat is trying to change to another color, and then to a lighter color, and then a very light color or bright color. And then if you look at this, still is the same. It shows that there is a gradual change from this point to this point. It didn't happen just in a single event. It happened over a long period of time, over many generations. That is gradualism. While in punctuated equilibrium, you see that the rat started from here. Yes, it's the same as this. So it keeps the same for a long period of time. But this rat is the same which gave rise to this. Yes, so when it reached here, suddenly, yes, within a short period of time, it changed from this color to this color. So when it was here, it changed from this to this immediately without a long period of time. So in punctuated equilibrium, it shows like maybe a mutation occurred, and then the organism changes rapidly within a short period of time, and then becomes like this from this point to this point immediately, and then it remains like that for a long period of time. Or when it reached here, it changed from this color and then went to this color just within a short period of time. While if you look at this, from this color to this color, it took a long period of time for it to change from this point to this point, and also took a long period of time to change from this to this. So if you compare this route and then you compare from here to here, this one shows that it's just a very short period of time. It happened, it changed within a very short period of time, while this one took a long period of time. The same story here, it shows that this rat just changed within a short period of time. It became like this. If you compare it with this, so it took a long period of time. So gradualism, in actual sense, gradualism require a long period of time for it to happen, while punctuated equilibrium requires a very short period of time for it to happen. And then gradualism, it is gradual, while this one, it has some periods of no change, and then periods of rapid change. Periods of no change, if you look at it, and then periods of rapid change. So it has a period of no change, and then has a period of rapid change. So that is what you call gradualism and punctuated equilibrium. Let's look at it. Saying that it explains this punctuated equilibrium, it explains the speed at which evolution takes place. At which speed is evolution taking place?
So we are saying that evolution involves a long period of time, long periods of time where the species does not change or change gradually through natural selection. This is known as equilibrium. So if you look at it, it is from this point to this point. It's the same period of no change. Period of no change. It is just the way it is, or we can say a period of it's just gradually through natural selection. Gradually, this is the gradually through natural selection. Period of no change. They're talking about this. We are talking from this point to this point, from this point to this point, because these errors are the same. They are the same. They are not changing. But if you look at gradually through natural selection, then this is the period you are talking about, or this is the period you are talking about. So during this period, yes, we call it equilibrium. And then we are saying that this alternates. It has period of no change. It alternates. This alternates with what you call punctuated. That is short periods of time. Short periods of time where rapid changes occur. This is through natural selection, during which new species may form in a short period of what? In a short period of time. So we are seeing that from this point to this point, rapid change happens. Yes, it could be also as a result of what? Natural selection, but species form within a short period of what? Within a short period of time. Basically, that is what we call punctuated equilibrium and gradualism. Gradualism, you find out that organisms, they take a long period of time before they change, while punctuated equilibrium takes a short period of time. There is a long period of no change, and then rapid change. Long period of no change or rapid change. They can ask you a question. Explain how did the tiger came up with stripes using gradualism or using punctuated equilibrium. So in this case, for when you're explaining in terms of punctuated equilibrium, you have to bring the there was a rapid change. You have to include the spots became bigger and then became stripes within a short period of time. So there was just a short period of time, then these tigers with spot became stripes. And then for gradualism, you have to tell us a long period of time. It took a long period of time before these tigers came up with the stripes. Let's look at artificial selection. What is, because we have
Today we are going to be looking at the 3 lines of evidence used for common ancestor of all hominids. The 3 lines of evidence used: number 1, fossil evidence. Fossil evidence, we are going to look at the different fossils. Remember, fossils are remains of living organisms. The organisms which existed, their remains form what called fossils. Number 2, we are going to be looking at the genetic evidence. In this case, we're going to be focusing on the mitochondrial DNA. And then number 3 is cultural evidence. So let's look at the fossil evidence. This one is the one whereby students, they think that is the toughest part of human evolution. But it's not tough. It's only that you just need to understand the concepts, and then you will be able to pass exams concerning about this. I'll show you even how do they ask questions concerning about this fossil evidence. So we are saying that under fossil evidence, you need to understand what is a transitional fossil or what is a transitional species. A transitional species is the one that shows an intermediate characteristics between 2 genera. Remember, when you say genera, we are talking about a genus. If it's one. If there are many, we call them genera, or different species, or between 2 species. Or we can define a transitional fossil or species as is the one with the characteristics common to both ancestor species and the species that follows. That's what I've tried to explain. that at least you have to be with the same characteristics. Look at yourself. You and your father and your grandfather or grandmother. You and your granny could be the different species. For example, if your father has some characteristics from the granny, and he also has some characteristics from you, then we can say that a dad is a transitional fossil or a transitional species. It has the characteristics of the granny and has the characteristics of the son. So it means that is having two characteristics. So a transitional species is the one which has both characteristics of the ancestor and then the characteristics of the following what species. Yes. So here is an example of a transitional fossil. You have this animal and then you have this animal. Yes, they are birds, dinosaurs, whatsoever. They are animals. They are under kingdom Animalia. So you have this animal and then you have this animal. So there is a transitional fossil of species here. How is it a transitional species? If you look at this species, there are some characteristics of this bird or this animal, and then some characteristics of this bird or this animal. So if you look at these feet, you'll find out that they are looking like this ancestor. Yes. If you look at this part of the wing, it looks like the ancestor. If you look at this part, it looks like this one. If you look at this part, it looks like this one. So it has both characteristics of the ancestor and the characteristics of the following. So we call this we call it a transitional species or a transitional fossil. What are some of the important sites in Africa which support this fossil evidence? Yes. Number one, we are saying that the first, we have countries, we have South Africa, we're going to look at Kenya, we're going to look at Tanzania, we look at Ethiopia, we're going to look at Chad. If you look at this map, it's showing you the different countries in their location. So let's look at the evidence, the fossil evidence. Here you need to know at least the scientific name, common name, who discovered, where it was discovered, and at least the age. So in this case, I've not included here because I have already designed a small clip which has all that information. So you have the scientific name. The first fossil you need to understand is Adepithecus ramidus, which we call Ad. Yes, who discovered it is Tim White. Yes, and then where exactly is in Ethiopia. Sometimes this one becomes the common name. Sometimes when you're asking, we might not ask you whether the common name or the scientific name. We just ask you a question which is open, whereby you are supposed to answer depending on the question. If it requires scientific name, answer in the scientific name. If it requires a common name, answer the common name. If it's open, you can choose whatever you want. So we have the Australopithecus afarensis. This one is called Lucy. The person who discovered it is Johansson. Johansson discovered it in Ethiopia. Lucy, those people who watched a movie called Lucy, you can even see how did life try to evolve. We have the three fossils. We call them Australopithecus africanus. The first one is Taum child. Who discovered it? Raymond Dart. Where exactly in Taum? It's called Taum child because it was discovered in Taum. Then we have Mrs. Place. Mrs. Place, who discovered Robert Broom, discovered in Stackfontein. Yes, in Stackfontein caves. Sometimes we call them Stackfontein caves in Taum. And then you have the Littlefoot. Littlefoot was discovered by Carl in Stackfontein. Still, they discovered it in the same place like Mrs. Place. But these they didn't discover them in the same period of time. So sometimes you can ask a question that give two Australopithecus africanus species found in Africa or found in South Africa. Then you can now name Town Child, Mrs. Place, and then Littlefoot. Sometimes you can ask which one of the following is known to Australopithecus africanus species. Then you say that Town Child, Mrs. Place, Littlefoot, and then Karab. Because Karab is also Australopithecus, but doesn't mean that it is Australopithecus africanus. Sometimes you can ask a question, just give three Australopithecus species you know. Here, when you say Australopithecus species, we are not saying that give the Australopithecus africanus. It's just Australopithecus. Then it means that even Karabo can be what? Included. But if they are specific to africanus, then you have to answer only question concerning about Australopithecus africanus. Yes. So we have Australopithecus sediba. This one is called Carabo, who discovered it. Is the burger, and then where exactly in Malapa in corridor of humankind in South Africa. Yes, Carabo has some unique features. Sometimes it's called a transitional fossil, or a missing link between Australopithecus, and Homo habilis. So this is a missing link between Australopithecus africanus and Homo habilis, or a transitional fossil. I explained the transitional fossil, that is a species which has some characters of the ancestor and some character as of the next species of the following species. Australopithecus sediba has some characteristics of Australopithecus africanus and Homo habilis. Therefore, it is regarded as a missing link. Then another one is Homo habilis. Homo habilis means the handyman. Homo means man. Then habilis means handy. So who discovered it is Mary and Lewis in the Old Vai George. They discovered it in Old Vai George, Tanzania. So basically, yes. And then we have Homo erectus. Homo erectus. Erectus, upright. Yes. It means that upright walking man. Yes. Homo means a man. Yes. A man. Yes. Homo habilis. Yes. Man. Yes. So it means that upright walking man. Upright man. Yes. This one is the one who discovered it, Du Bois, in Indonesia. So we have even in other areas, but the oldest was discovered there. Then Homo sapiens is called the wise man. Wise man, yes. I heard of a school called Wise Man. Yes, so maybe we can call it Homo sapiens school. All right, wise man. Yes, who discovered is Tim White, still again. He discovered this in Limpopo, in Western Cape, in KZN. Yes, all those places they discovered those Homo sapiens. Now, as I'm asking you, are you not Homo sapien? How comes that they're telling us that they discovered Homo sapien? And even the one who discovered Homo sapien is a Homo sapien. Does it make sense? The person who discovered Homo sapien is a Homo sapien. So he discovered himself. All right. Here they are trying to mean that the oldest Homo sapien. Yes. Oldest Homo sapien. However, the person who is discovering is Homo sapien, but he discovered the oldest Homo sapien. And where did he discover it from? Discovered it from those areas. Then note, Australopithecus sediba, which we call Carabo, is a transitional fossil. I explained this. That is a transitional fossil between Australopithecus africanus and the Homo habilis, the handyman. Between this and this, between this and this. So this Australopithecus sediba is the one which tries to link between these two. And sometimes we call it a missing link, a missing link, a missing link. Yes, number 2 is genetic evidence. Genetic evidence. This is mitochondrial DNA. Mitochondrial DNA. Mitochondrial DNA. Remember, when you're talking about mitochondria, we say that mitochondria are semi-autonomous. They are self-reliant. They have their own DNA. They are not being controlled fully by the DNA in the nucleus. No, it has its own DNA. However, the mutations which occur in the mitochondrial DNA, they are rare. And if they are there, we can use them to trace our lines of descent. But there is something special with the mitochondrial DNA. Yes, let me give you this example. Remember, when we are talking about fertilization in reproduction, we say that the sperm, when it fuses with the ovum, only the head of the sperm fuses with the ovum. The tail does not enter into the ovum. And remember, the middle piece and the tail, the middle piece is the one which has the mitochondria. So it's only the head. So the middle piece and the tail, they don't enter into the egg. What happens then if it is like that? Then it means that the egg will only have the mitochondria of the egg or of the ovum. So all of us, we have the mitochondria from the mothers, from our maternal parents. So it means that if there is a mutation in the DNA of the mitochondria, it means that only that of the lady will be passed to the next generation. So if we try to trace that DNA, we can see that, okay, it came up like this until we find the where it originated from. So I want to trace where the mitochondria from this. So I have the person here. I go like this. Father, mother, yes. In this case, I don't go. Why? Because it won't give me. I'll only go with the lady. Yes, you see. Now, when I reach here, the father does not give me the DNA. No. So I'll go with the female. I continue. When I reach here, then I continue with this. It means that the father doesn't give me. So this is where the mutation maybe happened. Or I can continue until I find the origin. So we are saying that mitochondrial DNA or mitochondrial DNA is inherited only from the maternal line, the mothers. And then you're saying that analysis of mutation on this mitochondrial DNA, yes, shows that the oldest female ancestor were located in Africa. The scientists say that Eve, Muslims call her Hawa. Yes, Eve originated from Africa. So Adam and Eve, even for guys, is East Africa. So it means that we are the owners of this world, because Hawa or Eve and Adam originated from Africa. That's what they say, scientists. So we are saying that, and then all humans descended from her. That is the mitochondria of Eve. That's the one I've been talking about. Yes. So basically, that's how you can explain. Whenever you see a tick, it means that, guys, don't miss out that point. Don't go to exam without knowing these evidences. I repeat, don't go to the exam without knowing these evidences, because if you don't know them, then it means that you won't be able to get the marks. All right, let's continue. Then you are saying that cultural evidence tool making. This one, using stone tools with cutting and sewing, improved the human nutrition. Like if you can cut the meat, so it improves the nutrition, so that you can kill the animal, so that you can obtain nutrients from this animal. Development of stone tools became more sophisticated as hominins evolved. As these hominins are evolving, yes, so the tools became more advanced. It's like a phone. 2000, the phone was just like that, but as you improve, you go on going towards 2020, it becomes more sophisticated. So that's what happened during those ages. And then you're saying that tool required planning and thought. So it means that also the brain was also developing, because they require planning and thought. You don't just come from nowhere and then you develop a tool. No, you can see them as they are just light, you can design them, but if you were the one by that time, you wouldn't have done that. A generation which will come, they will laugh at us, the kind of phones, the kind of computers we are using. Yes, they will laugh at us. You understand? Yes, so it means that technology or development of brain keeps on evolving and evolving. So here is the tool they started with. This check the kind of tool they started with, and then they started to modify it, and then they modified it up to this level. It shows that there is development in thinking. So this shows that from here to this point, it shows that there is an increase in the brain. You can see that the kind of tools they are using here, even the brain size here, you see, is just very small. The kind of tools that are here, these ones are more advanced than this. Even the brain size bigger than this one. This is Homo habilis, this is Homo erectus, and then the kind of tools now we are using are these ones. This is the Homo sapiens. Yes. So by then we used to have axe from stones and pangas from the stones, knives from stones, but now we just have to modify them. Even now we're using the stones. Yes, but they are just modified to suit the requirements. Yes, basically those are the evidences out of Africa hypothesis, and evidence for African origin of modern humans. Our brain, if you look at in the picture, you see that it is bigger compared to this. What is inside here is what makes the human to be reasoning more than any other animal. Let's draw a line here. You see the line here, and then draw a lower line. You'll find out that all this is human brain. But for this one is very small. You see now here, all this is human brain. But this one is very small brain. So it means that human have a bigger brain compared to other primates or other or apes. So we have a bigger brain. So this one is large, the other one is small. So we can say that cranium is a part of a skull which encloses the brain. So you have been told, you know about the cranium is this part which encloses this bone which encloses the what? The brain. So humans have a larger brain, more brain cells. It means that we can reason. So that makes it, yes, to reason, to have a greater intelligence. So they have more brain cells to the body size, This is an example of the elephant I gave you, that although the elephant has a bigger brain compared to us, but we have a bigger brain compared to the body size. So which suggests that we are of a great intelligence. And then we are seeing here that human skull, if you look at it in detail, yes, these are the things we are going to ask you on the human skull. So they're going to ask you, we have a bigger brain, a large brain, small brain. The face is flat here. The face here is sloping. You see them? The face is sloping. Here, the canines are reduced. We don't have well-developed canines. These ones, they have a well-developed canine. You can see them here. And then if you look at the foramen magnum is that hole, is that hole, this hole, which allow this spinal cord to enter. So if you look at here, it is forward. It's forward to the side of the mouth. Yeah, it's forward. So we say that foramen magnum is forward. That's how we want it. If you change it, that is in the center, no, we won't give you a tick. We want you to say that foramen magnum is forward, or it is in the forward position. And then here, foramen magnum is you see, is running backward. So it's running backward. So how do you write it? You say that foramen magnum is backwards. That's what we want. You change the statement, we give you 0. Yes. So basically, that's it. This one helps us, the foramen magnum, this hole, it helps us to know whether the individual is walking on 2 limbs or is walking on 4 limbs. If he's walking on 2 limbs, we say this bipedal. So we say that that's bipedalism. And then this one has a U shape. Yes, if you look at it, it is a U shape, while this one is rectangular shape. If you draw a line here, you see it is rectangular. Rectangle, is a rectangular shape. This one is a U shape. Sometimes they call it C shape. Yes. So basically, you need to know those things. If you look at this part, this part is almost flat, while this one is protruding out. So we will say that it has a well-developed broad ridge. This is the broad ridge they are talking about. Broad ridge are well-developed here. Here we do not have. So basically, you need to know those differences. So in the exam, we will not bring it like this. This is so beautiful. Now in the exam, we'll bring it like this. This one will look like this, while this one will look like this. And then this one will look like this. So you see that the teeth are out here. So when you're bringing an exam, we don't bring you like this. No. We bring you like, so we bring you like this. And then this one will look like this. That's how, then you ask you, we won't even label. We'll say that give the differences between skull A and skull B. And then from there, we will know that this one bridge will develop, this one will develop canines. We'll talk about that. So I will show you how you can answer this question. And then you're saying, what are the differences between the apes and humans? So I'm going to repeat, but now here we are talking about other things we didn't talk about. Large cranium. The cranium here is large, while this one, the cranium is small. We saw that the skull of where the brain goes, for the case of humans is large. This one is smaller. And then this one, the face is flat. Yes, the face is flat. While this one, the face is sloping. This one, the brow ridge are not pronounced. I showed you on the face that the brow ridge are not pronounced. While these ones, the brow ridge are pronounced, or they are well developed. Canines here, we saw that they are reduced, so they are small. While these ones, canines are large, or the canines are well developed. And then these ones are protruding. The jaws, if you look at the jaws, let me go back. If you look at these jaws, the jaws are protruding out. Even you can see it here. But here the jaws are not protruding out. Yes, you see that the jaws are not protruding out. But these ones, they protrude out. So we are saying that jaws are less protruding. They are less protruding. We can say that is less prognanthus. Less prognanthus. And then this one, more protruding. We can say that it is more prognanthus. That's how we can talk about. Then you're saying that this one, foramen magnum, is forward. I repeat, foramen magnum is forward, while this one, foramen magnum, is backward. So we say that this one helps us in determining whether the animal is walking on 2 limbs or is walking on 4 limbs. And then we are saying that more curved spine, we are going to look at this one, more curved spine, that is S shape, while this one, the less curved spine, we call it C shape spine. I'm going to explain this one in detail. Yes, and then short and wider, that is the pelvis. The pelvis is short and wider, while this one, the pelvis is long and narrow. I'm going to explain this and this more. And then we are saying that small and semi-circular pallets, while this one is long and rectangular pallets. So these ones are small. For the case of humans, if you look at your jaw, yes, they are small and circular. You see, I showed you that it looks like C. It's a C shape, while this one is rectangular. If you look at it, we saw that it's like a rectangle, yes? While this one is a C, or it looks like a U, depending on the direction where you are facing. Yes. So it's like 6 and 9, almost the same, depending on the direction. So it could be C or it could be U shape. So what is bipedalism? Bipedalism is walking on 2 limbs. So if you walk on 2 limbs, it means that
welcome back to our session, which is guiding you for paper 2. We'll be looking at questions, most likely examinable questions, which are going to come in your paper 2, both in prelims and final exams. Don't forget that in most cases, we don't set new questions, but we use the questions which are prevailing, and then we modify them. So I'll be showing you how to answer these questions, and I'll show you different questions. Some of them I'll be just speaking about them, and then how do you answer those questions. So keep on so that you don't miss out anything, and don't forget to subscribe, like, and share to others so that you can also benefit. M.Saidi as usual. Let's start with the question 3. The diagram represents the portion of
nucleic acids. Before I start this, don't forget that we have specific topics which we are supposed to look at in paper 2. The first topic is DNA code of life. The second topic is meiosis. The third topic is genetics. The fourth topic is evolution, but both evolution in natural selection and then human evolution. So it means that these are the topics which are being set in paper 2. So when you are reading, you must focus on these 4 topics or 5 topics. So we're going to look at all questions and then the most likely examinable questions or questions you're most likely to find in your paper. If they bring a diagram like this, you have to find out which diagram is representing. So this diagram represents DNA. Why DNA? Because of this, the weak hydrogen bonds. And then it's two-stranded. What are some of the questions which are most likely to be asked about the structure of DNA? Apart from labeling the structure of DNA, they can ask you to describe the structure. Apart from labeling it, describe the structure of DNA, and they put there 6 marks, whereby you have to tell us DNA is made up of nucleotides. Each nucleotide is made up of a phosphate, a nitrogenous base, a phosphate, and a nitrogenous base, a sugar, and a phosphate. It forms 2 strands. Each strand forms 2 strands. Each strand is joined together. So you describe the characteristics of DNA. Basically, what DNA has. It forms 2 strands which twist around to form a double helix. So you can get the 6 marks from there. So in this case, let's label it and then find out what is a V. What is V? V is the nucleotide, because a nucleotide is a nitrogenous base, a sugar, and a phosphate. So this is a sugar, and then nitrogenous base, and then a phosphate. So this circle or box is showing you a nucleotide. Then Z is weak hydrogen bond. Use the word weak. Weak. We want to see the word weak. Then hydrogen bonds. Weak hydrogen bond. And then this is a nitrogenous base. Nitrogenous base. And then also this is a phosphate. Sorry, a nitrogenous a sugar molecule, which is, in this case, it is a ribose, deoxyribose sugar. And then Y is a phosphate group, as we saw it here. So now, since you know that this is a structure and you know how to label it, now let's talk about name the nucleic acid which is being represented. So we have seen it that this is DNA. And they're saying name 2 places in animal cell where you can find this DNA. Where can you find this DNA in animal? You can find in animal in 2 places. That is in the nucleus, and then in mitochondrion. If it's one nuclei, if it's one mitochondrion, if there are many mitochondria, if there are many nuclei, if it's one, it's called nucleus. So it should not confuse you. They are saying name portion V. They are looking for portion V. What is portion V? We say that these are the building blocks. This is the building block of DNA. So it is the nucleo. Molecule Y. Where is Y? Molecule Y is a phosphate. We have seen it that it is a phosphate group. Then they are saying that bond Z. What is bond Z? Bond Z is weak hydrogen bonds. So I think we have seen the answer here. Weak hydrogen bond, we have seen here, phosphate group, nitrogenous base, we have seen it, and then portion W, which is the nucleic acid, sorry, which is the nucleotide. What is the natural shape? When they say natural shape, many students, they say helical, double helical. No, the only answer we accept in exam is double helix. Nothing else. You write something different from that, we won't give you a tick. Name the process during which this molecule makes exact copy of itself. How does DNA make exact copy of itself? How does it replicate? So the answer is DNA replication. So that is the process by which DNA makes exact copy of itself. Why does it occur? Some questions can come here. Why does DNA replication occurs? It occurs to increase or to double the chromosome number, to double the chromosome number. Where does it occur? When you talk about where does it occur, then you talk about the nucleus. And then when does it occur? It occurs during interphase. Yes. And then why does it occur? Why does it occur? We say that to double the chromosome number, to double the chromosome number. And then lastly, how does it occur? That is the process of DNA replication. If they bring a question like this, how do you answer? This is a DNA molecule. So if you look at it here, you see it is a DNA molecule. And why is it DNA molecule? Because of the thymine, which is indicated in the nitrogenous base. And then also the weak hydrogen bonds, 2 strands, so it identifies it to be a DNA molecule. And it cannot be RNA, because even here they say that this is what? This is a DNA replication. Yeah, so question them. Molecule W, where is molecule W? We have seen it, molecule W is here. It is a nucleotide, correct. Molecule U, where is molecule U? The whole molecule is DNA, correct. Then they are saying that Z is weak hydrogen bonds, correct. And then what is V? How do we know the nitrogenous base V? Because nitrogenous base is compatible to this or complementary to this, therefore, and this is given it here, which is thymine, therefore V is equal to A, which is adenine. And then they are saying that name the part of molecule W is W, X, and Y. X is the phosphate group, correct, and then Y is the oxyribose sugar. Why don't I say sugar? You have to be specific because this is DNA, so it is the oxyribose sugar. Where in the cell does this process take place? Where do we find? We have answered this question that this process occurs in the nucleus. And then they're saying that name the phase of cell cycle where replication takes place. We have already answered it, and then we say that interphase. So at least when they bring such questions, you must be able to answer accordingly. So first try to answer what is being labeled. The moment you know what the picture depicts or what a picture represents, at least you are able to bring your mind and then you concentrate in that area. I'm not going to spend too much time here, only maybe if I see a very difficult question there, then that's when I can try to answer related to this, because I wanted to go to transcription and the translation. But molecule C. Molecule C is phosphate, and then this is a nitrogenous base, and then this is phosphate, sugar, deoxyribosyl sugar, and then nitrogenous base. It forms what you call a nucleotide. And then weak hydrogen bonds, and then this is a sugar, this is a complementary base. So this is a complementary base, and then here, yes, they said it's weak hydrogen bond, and then you have this, how do you call that? That is not a strand. We call it a backbone. And then the whole strand must include with the nitrogenous base and this. So the difference between a whole strand and a backbone is the backbone does not involve the nitrogenous bases. If you are asked, what is this bond? This is a phosphate sugar bond, but it is a covalent bond. If you refer it to physical science. Okay. Two scientists who discovered, James Watson and Francis Crick, name one organelle where DNA is found in the nucleus and the mitochondria. Describe how mutation of DNA may change the structure of the protein. The moment DNA molecule changes, is going to change the sequence of the bases on messenger. And messenger RNA is going to change the sequence of the amino acids in which they are being brought to form a protein. So if a different protein is formed, if the amino acid leads to a different sequence, then a different protein is going to be changed and or is going to be formed. Then amino acid leads to the same sequence as previous one. Then the protein is not going to, a new protein is not going to be formed. Table 2 different structure difference. Now, if the major point here is structure, the word is structure between the monomer, and they are saying about the monomer of DNA and RNA. So here they are talking about the monomer, and if you look at the monomer, they are looking at the nucleotide of DNA and the nucleotide of RNA. So you only look at the structure which is the nucleotide, of which the nucleotide is made up of a phosphate, a sugar, and then the nitrogenous base. So there are only two differences here you can look at. You can look at the sugar. DNA nucleotide has deoxyribose, deoxyribose sugar, while RNA has ribose sugar. And then DNA has thymine, while RNA has uracil. So after that, you draw a table. It must be a complete table. Yes, it must be a complete table. And then you put DNA, and then you put RNA at least. Then you get a tick here, a tick here, a tick here, a tick here, and a tick for the table, which makes it to be 5 marks. And if you find a question like this and tablet, and there is only 10 minutes or 10 seconds remaining, don't waste time. At least you can grab that 1 mark. Just draw a table like this, and then put DNA, and then put RNA. Yeah, even if you don't have anything here, then we shall come and give you a tick. At least you will score that 1 tick. So yeah, please try to use different ways in which you can get marks. Yes. Yeah. So this is the same thing we have been trying to discuss. Phosphate, you have seen them, and then M2 process, which require the strand of the nucleotides in a single strand of DNA. Sometimes people think that DNA can be a single strand. It can be a single strand. What makes this one to be DNA is because of the thymine, which is being indicated there. So thymine is in this form, while adenine is in that form. Then they're saying that how many nucleotides are represented in the diagram? Yes, let's try to answer this. How many nucleotides? A nucleotide is a phosphate, nitrogenous base, and a sugar. So this is 1 nucleotide, it's the 2nd nucleotide, it's the 3rd nucleotide, and then this is the 4th nucleotide. So the answer is going to be 4 nucleotides. And then they're saying phosphate molecule, then phosphate molecules, you just count them 1, 2, 3, 4. So the answer is also 4. And then they're saying that how many nucleotides, okay, I done with that. Sugar molecule, there are 4, But sugar, this is the phosphate, and then this is the sugar. Write down the nitrogenous base from top to bottom as indicated by the arrow as complementary base strand and this molecule. Here is just T, they're saying on DNA. Complementary. So they are not talking about convert it into RNA. Don't forget that A goes with the T, then C goes with G. So whenever there is A, T, we put A. Whenever there is C, we put G. Whenever there is A, you put T. Whenever there is C, you put G. So basically, that's how it's supposed to be answered. Name two processes that require the DNA strands of DNA molecule to separate into single strand as shown in the diagram. Which processes? Two processes. We only find this in when the DNA replication is going to take place, and also we find this when there is a transcription. Remember here you form two strands, two separate strands. Each strand is acting as a template. Well, here you form two separate strands. Only one strand will act as a template. And while here, two strands will act as a template. So these are the two processes which make DNA molecule to open. DNA profiling is also not a big deal. Technique, you have to know how to identify this. Read the question very well and then find out. Don't just answer the question. Detectives were investigating crime scene, found the blood on the broken window. Then they suspect that the blood was that of the criminal. To identify the criminal, they analyzed the DNA sample from different DNA sample from the blood and compare it to the four suspects. Now, look, this is DNA sample, and then these are the suspects. It means that these are the DNA they found on there, but you have to see where it is corresponding. This one goes with that. This one goes with that. Still, yeah. This one is that. Use a ruler to identify this. That this with this. This with this. This with this. This with this. So it means that Jenny is a suspect for that case, whose DNA profile is matching with the DNA sample obtained from the crime scene. Name the technique which was used. It's DNA profiling technique is DNA profiling, and what is being shown here is a DNA profile. So you must know the difference. DNA profile is not a technique. DNA profile is what you obtain when you analyze the DNA sample. So who was the possible criminal? We have seen it is Jenny, because the DNA profile, or the DNA profile of bars, there of sample. DNA sample profile is matching with the profile of Jenny, or the band of DNA profile sample, they are matching with that of Jenny. So state one other use of DNA technique, or state other use of the technique identified in the question above, meaning that the DNA profiling, what is it used? Can be used to identify the relatives. They have talked about the criminalism, so you don't talk about that anymore. So talk about the identification of the perfect organ donor. You can talk about lost relatives. You can talk about diagnosis of the inherited diseases. So there are so many uses you can use to when you are dealing with the DNA profiling. So it's the same thing. You see that is the same thing. I don't usually take too much time here because these questions are really open. Who is a suspect? You know how we have identified it. What diagram is DNA profile? what does the diagrams above represent? They're not saying the technique, they're asking for the diagram. Therefore, it is DNA profile, not DNA profiling, because here they said which technique, name the technique. So it's DNA profiling, but here it is DNA profile. Which suspect? Look at the bands and then you match them, give a reason. So the suspect, if you look at them, you find out that Q is most likely to be the suspect because the bands of broken glass, they are matching with that of Q. So they're saying that name 2 possible disadvantages of using this evidence in the court. It might not be accurate 100%, because number one, they can human error, number one, and also it may not be unique if a small sample is used. Also, they can a human being can manipulate them, so it may not be 100% accurate. So we have seen many questions concerning about DNA profiling. Download this and then try to do these questions. Where you get stuck, come back to me. I'm asking me questions. I'll be able to help you out. My number is always on the website 084-833-1738. Yes, and then speak to M.Saidi, who is always ready to help you out. Then another question is still the same tablet. Whenever you talk about tablet, make sure that you draw a nice table, and then so that you get that 1 mark. That's why there are 5, because they're asking for 2. They give you 2 marks there, and then 1 mark for tabulating. Describe the process that is being represented or responsible for formation of molecule B. So here, molecule B is messenger RNA. Why messenger RNA? It's because of this. So this is not, you see, this is messenger RNA, which is uracil. Therefore, and also 1 strand is being used. So that's evidence to show that this is transcription, because they're saying this is non-coding strand. So it means that only this is acting as a template. Yes. So they're saying that describe, so what are they looking for here? Describe the process that is responsible for formation of B. If you look at B, is messenger. So you describe. How do you describe here? Let me just talk about it as I told you. Talk about DNA double helix, unwind, weak hydrogen bond, break, and unzip. So when the weak hydrogen bond break, it's the same thing saying unzip. So you get 1 mark there. To form 2 separate, the word separate must be there, 2 separate strands. Only 1, you see it here, that only 1 is acting as a what? Is acting as a template. Only 1 is acting as a template. So it's being shown there that this one is acting as a template, and then using free floating messenger RNA. So you have to write messenger RNA nucleotide, yes, to form a complementary strand where adenine goes with uracil, and then guanine goes with cytosine. Adenine goes with uracil, and then guanine goes with cytosine. The complementary strand move out of the nucleus via the nucleopore to the cytoplasm on the ribosome. So basically, that's what you're supposed to explain. Please don't try to bridge it, because it's a process. 1 step leads to the other. If you start with what is on at the bottom and then you start with it, then you lose marks because the sequence is not the same. So you cannot start with the last step and then you end with the first step. You have to start with the first step and then you end with the yes. So molecule B, we said it is messenger RNA. Name the process is transcription. Explain how the protein molecule would be affected if codon GUU is changed to CGC. So if it here, it depends on 2 things. If there is a table Yes, now you compare the table and see if it's gonna lead to a different amino acid. But because now this is open, then we assume that it's gonna be a different amino acid. For example, if you say that you started with GUU, yes, and then GUC, from this to that. If this, let me explain this. If this is tryptophan, yes, codes for tryptophan, and this one also codes for tryptophan. However much this has been changed to this, is going to code for the same amino acid. Therefore, the functioning of the protein is not going to change. But if this tryptophan, and then now this one codes for example, leucine, then it means that there will be a change in the functioning of the protein, because this is tryptophan, and then this is leucine. Therefore, a different protein will be changed, will be formed, and then still the functioning of the protein will also change. So explain how the protein molecule would be affected if this is changed to this. So therefore, you tell us that if GUC is changed to GUC, GUU is changed to GUC, because they say that codon, this changed to, because this, when they say codon, it means that we have started from the messenger RNA. Then this will lead to different sequence of amino acid. Different amino acid will be brought to the molecule or to the messenger RNA, not messenger RNA, will be brought to different amino acids will be brought to ribosome, and then a different protein will be formed. So that's basically you have to understand. And then when you come here, this is translation, because it is occurring on the ribosome. So what is A? A is messenger RNA. What is B is the ribosome. And then what is C? C is the transfer RNA. What is Z? Z is the amino acid. So this is the monomers, amino acids, monomers of the protein. And then this is the transfer RNA. You have seen it. And then this is the ribosome, and then this is the messenger RNA. So basically, you need to do that. And then what is this? These are 3 bases. So therefore, these are codons. Yeah, remember, the 3 bases on the DNA, we call them codes. And then 3 bases on the messenger, we call them codons. And then 3 bases on the transfer, we call them anticodons. Organelle X, what is organelle X? We have seen it. We have seen all the questions. You see now, when you start with trying to label them, you'll be able to answer all the questions. Molecule W formed, you see, organelle Z, X, we have seen it, ribosome, monomer W, the monomer. They're saying the monomer of molecule W. Remember the building blocks, that's what we call the monomer. Therefore, it's going to be the nucleotide. Be the nucleotide. Yes. And then molecule W and Y, W and Y, we have seen it. W is messenger RNA, and then Y is the transfer RNA, and then molecule organelle X, we have seen it is the ribosome. So still the same thing. You can see some people, they call this a protein. This is not a protein. It is only a protein if it is 50 and above amino acids. So what is this then? This is a polypeptide chain. Yes, so it is a polypeptide chain. So what is X? This is the amino acid. And then what is this? This is transfer RNA. And then what is Z? We have seen them. These are anticodons, or anticodon. And then this is messenger RNA, and the 3 bases on the messenger RNA, we say we call them codons. You see, you can't fail such questions. When you come to the question, you see that they are asking you the exactly the same thing, and then asking you describe the process of transcription, which we have described. So basically, at least you have some light on DNA, and then maybe we will talk about the process of translation, and then after that we wind up with the coding and then the mutation. These are some of the short question which you need to know before you go for the exam. Number one, DNA replication. You have to know DNA replication, the process by which DNA makes the exact copy of itself. Where does it occur? We have seen it in the nucleus. When does it occur? We say that it occurs during interphase. And then we said, why does it occur? We say that to double the chromosome number. And then how does it occur? That's the process of DNA replication. And don't forget that here you have to explain that here the you form two separate strands, and each strand is acting as a template, and that is the major difference between DNA replication and transcription. So in most cases, people confuse transcription and the DNA replication. Then number two, describe the location and the structure of DNA. Here I explained it at the beginning, the structure of DNA. In most cases, give it 6 marks. And then location, we have 3 locations, that is nuclear DNA, mitochondrial DNA, and chloroplastic DNA. So if they talk about in plants, then you talk about all of the 3. But if they talk about in animals, you only talk about the 2, that is the, because we don't have a chloroplast in animals. So describe the structure of RNA. Structure of RNA, it's a little bit the same as DNA, but only few things which are not there. That is the thymine, the double strand, and then also the weak hydrogen bonds. Describe the involvement of different types of messenger RNA process in protein synthesis. Here they are talking about the functions of each. For example, you talk about transcription and then translation. So it is like you are describing the whole process of DNA of protein synthesis. Another thing you need to know here is also describe transcription in detail, and then also translation also in, yes. And then also what they can ask you is why is the mutations which occur. And I say that if you change the code, for example, from GUA to GCA, then if these two they code for the same amino acid, therefore the protein will not change. The structure of the protein will not change. But if the different amino acids are being coded for, then the structure of the protein will be changed. So let's go to meiosis. Let's go to meiosis. There are so many questions which can talk about DNA. And also the base calculation, whereby we're supposed to talk about if adenine is equal to 10%, what is the percentage of guanine, or the ratio of guanine, whereby you have to tell us that adenine goes with the thymine, and then cytosine goes with the guanine, whereby if this is 10, or definitely this is 10, and then 10 plus 10 is equal to 20. Since this is percentage, therefore I'm going to say 100 minus 20, which means 80%. And then 80%, so what happens? So it's going to give us 80% for these two, so divide by 2, and then from 40%, therefore this is 40, then this is 40. So this is just a simple recall. I hope you remember. Please don't go without knowing it, because at least those 2 marks you'll be able to answer them. And then if they are looking for the ratio, you can put them maybe adenine now. Adenine, we said it is 10, and then maybe with the guanine we said is 40. So divide by 10, and then we're gonna get 1, 2, 4. So sometimes they swap it and they say 4 to 1. And then because when now you have seen 1, and then you have seen 4, you have seen 4, you have seen 1, you just circle that. It is what? It is wrong. So when you go to meiosis, so many point where crossing over takes place. We call it chiasma. You see a chiasma. Splitting of the cytoplasm, we call it cytokinesis. Okay, we can answer them. Failure of the chromosome to separate during meiosis, the non-disjunction, the structure in the cell that forms spindle fiber, centrioles, and then the phase of meiosis where homologous chromosomes are aligned along the equator, that is Mera phase 1, because they're saying homologous chromosomes. And the point at which the two chromatids of the chromosome join, we call it centromere, and this division of the nucleus. Division of the nucleus is karyokinesis. And then they're saying that the site of meiosis in a female, because in female we find meiosis in ovary. So you're right, ovary there. Ovary. And then the exchange of genetic material between chromatids in the homologous chromosome, which is crossing over. And then they are saying the genetic disorder caused by extra chromosome on chromosome number 21, that is Down's syndrome. Yes. And then they are saying that the structure formed between the centrioles by the centrioles during the cell division with spindle fibers. Yeah, spindle fibers. And then they're saying that non-sex chromosomes in human, we call them non-sex autosomes. And then they're saying the condition in the cell where there is only one set of chromosome. How do you call that condition? We call that condition haploid. And then the structure that is responsible for formation of the spindle fiber during spindle fiber, we say that is called the centrioles. Yes, and then the A chromosome aligned along the equator, none, because here it must be Mera phase. Random arrangement of chromosomes, it must be Mera phase. So it's A, B only. The site of meiosis in plants, anthers, and ovary, both in, this is male, this is female, and then the process of meiosis produces meiosis result in 4 haploid daughter cells, 4 diploid. So this one becomes wrong. So this is the correct one. The mechanism to introduce genetic variation, crossing over, is correct. Random arrangement of chromosome is also correct. So it is both A and B. So basically, you see, yeah. So now we are starting the other part of meiosis. All right. Please don't forget to subscribe. Stay in touch. If you are in any problem, we need to smash these questions, and then we get all the marks at the end of this period. Yeah, when you come to meiosis, there are some questions we can talk about. Let's see what we can go through so that they can also benefit you in answering questions related to meiosis. I know it is a simple topic, but is a little bit challenging to some other. But let's see if we can smash these questions, and then we get a better understanding. If you don't understand the topic, let me know. I'll help you out so that at least we can have more distinctions in this year. All right. They are saying that here you are seeing diagrams. What comes into your mind is the kind of phases which this diagram is indicating. So, what is C? C is spindle fibers. What is B? This because there are 2 chromosomes of the same kind or the same size and structure, therefore is homologous chromosome. Then the joint between two, or the joint between two chromatids of the same chromosome, it is a centromere. And then diagram A, what does diagram A represent? Because of this one is starting to form, so it can represent prophase, prophase 2. Why prophase 2? Why not prophase 1? It's because now this, they are individual, they are not in pairs. And then number 2, you see that crossing over took place a long time ago. And then this one is metaphase. The chromosomes are aligned. If you turn this, and then comes this side, and then this goes that side, so that this thing is in between your chest. So you see that if this is one side of the chest, and then this is on the other side of the chest, I think now you see what I'm trying to say. So you see that they're in the middle. So why? These are the spindle fibers. Yes, if this is the spindle fiber, if this is the spindle fiber, and this is the spindle, centrio. So these ones are in the middle. That's why you say that it is under metaphase. But metaphase, which metaphase? It is metaphase 1, because these chromosomes are in the middle. Then they say identify A. We have seen it. A is centromere. B, what is B? B, we have seen it homologous chromosomes. Then C, you see, we have seen it as a spindle fibers. Identify the phase in diagram 3. Diagram 3, because they are separating. Yes. So if they are separating, then they are moving to opposite poles. You see them? They are moving to opposite poles. Therefore, it is anaphase. But which is it? Anaphase 1 or it is anaphase 2? Is it anaphase 1 or anaphase 2? Because now there are chromatids which are moving to opposite poles. Chromatids moving to opposite poles. Therefore, it's going to be anaphase 2. Anaphase 2. And then they're saying that if it was anaphase 1, then this would have been individual chromosome. This would have been a chromosome moving to the other side. Also, this one would have been a chromosome, and the centromere there, this would have been a holochromosome, and also this would have been a chromosome. So it would have been anaphase 1. But because their chromatids moving to opposite poles, therefore it is anaphase 2. Then they're saying that write down the number of the diagram shown in the sequence which the phase occurs. The number of the sequence. They're saying write down the numbers of the diagram to show the sequence in which. So basically, we have to look for meiosis 1. The chromosomes are supposed to be in homologous pair. So this one becomes, this is homologous pair. So it is 1. Therefore, it is what? It is metaphase 1. And then this one is prophase 2. And then after prophase 2, they separate, which is, so it's going to be 1, it's going to be 2, and then it's going to be 3. So when you are writing the numbers here, it's going to be diagram 2, diagram 1, and then diagram 3. So it's going to be diagram 2, diagram 1, and then diagram 3. Then one difference between metaphase 1 and metaphase 2. In metaphase 1, the chromosomes are aligned along the equator in homologous pairs, while in metaphase 2, chromosomes are aligned along the equator singly. They are individual chromosomes are the one which align along the equator. So that is the major difference. And then if we see another question, so label it. Very simple. You see, we usually bring the same kind of questions. So this is the spindle fibers. This is the centromere. This is chromosome. Non-homologous chromosome is 1 chromosome. And then this is the centrio, which is being produced by centrosome. Okay, centrio. And then what causes these differences in the chromosomes or chromatids is because of crossing over. When does crossing over takes place? It takes place during prophase 1. How does it take place? Homologous chromosomes, they align together. Chromatids of the homologous chromosome, they cross over at a point called chiasma, and then they exchange the genetic material, and then they separate to form different chromosomes which are genetically different. Basically, that is the process of crossing over. You can't go to the exam without knowing the process of crossing over. Identify the phase of meiosis in diagram 1. Diagram 1, because now, yes, the chromatids are separating, is the same thing like this one. It's the same thing. The same thing like this is exactly the same. Yes. So it's gonna be anaphase 2. And then this one is going to be metaphase 2 because the chromosomes are aligned along the equator singly. Yes, they align along the equator singly. Then they're saying that identify the phase. You are done with that. State what happens to structure D in the next phase. Where is structure D? Structure D, these are the chromosomes. So the chromatids, they move to opposite pole due to contraction of the spindle fiber or due to the shortening of the spindle fiber. Name the process during which genetic material were exchanged. What was exchanged? We say that the process is called crossing over, and then we said it crossing over, and then we said it occurs during prophase 1. State the consequence if the mentioned does not occur. If this process does not occur, it means that organisms it will reduce what you call the genetic variation. It means that more organisms will be more similar to each other. So it will reduce variation among the organisms because crossing over leads to genetic variation. Then they're saying that give the number of the chromosome present in the original parent cell in this diagram. They didn't say this is a human cell. I don't see anywhere there saying this is a human cell. So you have to know, you have to use the number which is being given. So because this is meiosis 2, and meiosis 2 leads to the diploid state being halved. So for example, you start with 1N, and then you form 2 cells, which is 1N, and then another 2 cells you divide to form 1N, 1N. So if you start with the 4 here, therefore it's going to be 2, 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 it's going to be 2. So it means that this is meiosis. Therefore, this is 1, this is 2. Therefore, the total number is 2. Please don't talk about 46 if they didn't say that this is the human cell. And then now they are saying original cell. So if it is original cell, therefore it's going to be 4. And then you see now they are saying, specifying it. Human cell in the sample diagram 2. Because diagram 2 is haploid, therefore it's going to be 20. It's going to be 23 chromosomes. So basically, that's how you're supposed to answer such questions. So you can answer these questions, at least in meiosis. What is A? A, in most cases, students confuse it with a nuclear membrane. This is not a nuclear membrane. You can't have a nuclear membrane when the cell is under a metaphase. Therefore, this is the cell membrane. This is the chromatid. This is the chromatid. This is the spindle fiber. And then this is the centriole. So this is metaphase 1. While this one is anaphase 1. Why? Because chromosomes are separating. What about this? This is the telophase. Telophase 1. Why? Because they are chromosomes. They are chromosomes which are still double-stranded. Yes. Or at the beginning of prophase 2. Yes. So you see that now once you name that, you are able to answer this question. What is phase 1? We have seen it is metaphase 1. What is 3? We have seen it is telophase 1. Then you're saying the letter only of the part contain DNA contains DNA. Give the letter of only part that contains DNA. It is B, because in this case they didn't mention any chromosome here. It's only B, which is chromatid, and then attaches the centromere of the chromosome, attaches the centromere chromosome, which is C, attaches is the centromere. Yes, and then they're saying that forms spindle fiber, which is the centriole. Where is the centriole mentioned? Is D. You see, we are able to answer these questions. Name the organs in humans, male, where meiosis occur. In humans, male, it occurs in the testes. While in female, it occurs in ovary. In plants, it occurs in anthers, that is male, and then stigma, ovary in female. So here they produce sperms, they produce ovum or ova. Here they produce pollen grain, while here they produce ovules. Yes. So basically that's a simple illustration of meiosis occurring in plants and also in animals. Describe how meiosis leads to genetic variation. So now here you are talking about only processes which talk about genetic variation. Here you talk about crossing over. So you describe crossing over. Then you talk about the random arrangement of chromosomes during metaphase. Metaphase. Yes, so basically that's what you need to explain. It can lead to genetic variation. You can also mutation can also be part of meiosis. Why? Because if you talk about Down syndrome, it occurs during the process of what? Occurs in process of meiosis. But these two, they can help you to get those marks you are looking for. Here is an example of a karyotype. You have two individuals. Identify which karyotype. Here, when you're given a karyotype, look at, is there any abnormality? Which one is the male and which one is the female? So if you look at this, no abnormality. All of them, they are in pairs. But if you look at that, one is long and then one is short. Therefore, this is X. This is X, and then this is Y. So therefore, this is a male. And then both of them are the same size. Therefore, this is a female. So now from there, you can now go and then try to answer. What term is given the chromosome number 1 to 22? So we call them, because they can ask you to describe the structure of a karyotype. You call them orosome. The orosomes. And then 23, these are gonosomes. Why? These are sex chromosomes. Yes. State the gender of P. We have seen it. P. Where is P? Male, because of this. Give one observable reason, because the chromosome number, the chromosome number, the chromosome pair number 23. One chromosome is bigger than the other. Therefore, one is X, one is Y. Hence, the person is a male. Then they're saying, give one observable. We have seen it. Each of the pairs shown is a homologous pair of chromosome. State the origin of each chromosome in the homologous pair of the chromosome. So here, where do they are two. Where are they coming from? These pairs, one is coming from the father, and then one is coming from the mother. Yeah, that's why there are two. That's why we say that when the sperm comes, is haploid, and then fuses the ovum, which is haploid. So one chromosome, another chromosome, when they combine, you form the pair. So it's the same thing. Sperm, ovum, sperm, ovum, sperm, ovum, sperm, ovum, like that, like that, like that. So one comes from the father, one comes from the mother, and then from a diploid, a zygote. Then they are saying that the diagram below represents the distribution of chromosome 21 as it appears in the gamete. So it's the same thing, guys. Yeah. So you see that you always find almost the same diagrams, so same structures. So now draw a diagram to show the cell during anaphase 2 of meiosis. Now here, when I ask you to draw a diagram, you must write a title. That is the first thing, title. You cannot draw anything without a title. Use a sharp pencil. It must be neat. Yes. Someone is going to ask you, am I neat when I'm writing? Yeah, I'm saying you must be neat. I'm not a student. All right. I'm saying that. So when you draw, they're saying this is the prophase 2. Prophase 2, why? Because they are single. So what do? You draw the cell, and then they're saying during anaphase. So it means that before it's going to be like this, and then the short one down. So this is prophase. Just drawing a structure, but you have to show the variations when you're drawing. So during anaphase, it means that one, and then one has a variation. So it's still moving to the poles, and then also then the short one is gonna be the short one, and then it's also moving. You see, but it also has the biggest variation. The biggest variation, and then this other one is completely shaded. You see, you have to show that. And then this is spindle, this is chromatid moving, and then and then this is the cell membrane. Then write the title. Yeah, at least you are able to answer such question. Yes, so that when you come, we'll maybe give you a mark for title. Yes, a mark for drawing, correct drawing, and then maybe those are 2 marks. Now, maybe 3 marks for labeling. Yes. So that's why I'm saying labeled diagram. So meiosis is also easy. If you look at most of the questions, you will see that we have answered those questions. Can you answer this? Yes. 10 seconds, 8 seconds, 7 seconds, 5 seconds, 3 seconds, 1 second. Okay, let's see. Then they're saying that this is what? Spindle fibers. What is this? Homologous chromosome. What is this? Centromere. Yes. So these are homologous chromosomes. And then this is the centriole. And then this is the cell membrane. So you can answer these questions. You see, nothing which is going to be new in your paper. Name the process diagram above. You see, it is metaphase. You see, you have to turn it so that these things are in the sides. Check. You see, they're in the sides. So that you see now clearly it is metaphase. Name the process, name the phase, represent a diagram. We have seen it. Then they are saying name part C. We have seen it also. Centromere. Then B represent homologous chromosomes. B represent what is homologous chromosome. Chromosomes which have the same structure and the same size, and then they carry genes. Genes of the same characteristics are always on the same locus. Yeah, so we call them homologous chromosome. Then they are not genetically identical, but genes of the same characteristic, they are in the same locus. Locus is a position of a gene. It's singular. Loci when they are many. Explain the appearance of chromosomes in diagram. Why are they shaded like this? Is due to crossing over, which took place in prophase 1, whereby the chromatid exchange the genetic material. That's why they are like that. They are saying that total number of DNA is the total amount of DNA is 12 viral units. Each daughter cell at the end of this cell division. How many DNA was in the parent cell? So there they are saying that the total number of DNA is 12 units in each daughter cell. Remember, you start with the 2n. You start with what? 2n. Yes. So 2n, you form two cells, 1n, 1n, then you form another two cells, you form another two cells. So 1n, 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 1n. So if you started with the, if your daughter cells are having 12 here, so it means that then if you want to get the parent, is times 2. So which is 24. So the answer is going to be 24. 24 DNA binary units. Don't forget to put the units as they are being given to you. So many questions, you'll find them like this. This is the stereokaryotype. Male or female is a female, while chromosome number 23 is having chromosomes which are of the same size and the shape. See? So describe the karyotype of person with Down syndrome. So a karyotype with Down syndrome will have an extra chromosome on chromosome number 23. Yes, so the pair chromosome, so there will be 3 instead of what? Instead of 2. So you see that these diagrams, these questions, you can answer them very well. Yes, you can answer them very, very well. All right, let's just do few questions about it. What is this? What is this? This is a centromere. What is B is a crossing over, point where crossing over takes place, therefore is chiasma. These 2 chromosomes under crossing over, we call it a bivalent or homologous chromosomes during crossing over. Then this, what is this? This is a chromatid, and then what is this? Is a chromosome. So these are sister chromosomes. So now you can answer the question. Describe the process that occurred at D. So it means that they're asking to describe the process of crossing over. I explained it before. This is where tricky things come from. They're saying the certain number species has 42 chromosomes. They're not saying it's human. So don't talk about 46. Yes, please don't talk about 46. They are saying 42. The muscle cell, in its muscle cell, remember muscle cells, those are autosomes. They're always 2n. Yes. Give the chromosome number of each of the sperm cell. So it's going to be half because sperm cell is 1n. Therefore, the answer here is going to be 21. The skin is autosomes is 2n. Therefore, it's going to be 42. And then ovum of the female of the counterpart is the same thing, is going to be 1n, which is 21. In most cases, the number of chromosomes are the one which are confusing students and they end up losing marks. So it's the same thing here. Crossing over took place. They can ask you to draw the diagram after crossing over. So members, students, please don't lose marks. Don't lose hope. You will make it. You will make it. I'm going to come back for genetics and inheritance. And then after that, I will go to human evolution. Thank you very much for watching. Don't forget to subscribe, like, and share. Don't also stop to comment so that I know where exactly I should put more effort and where to help so that you can get your distinction.
Please use the link below to get the notes, questions, and other videos. Don't forget to subscribe, like, and share to others for our daily new videos.
Yeah, welcome back, guys. Welcome back to our class. We are looking for distinctions and making sure that we pass. All of us, we must pass. We must go to the university. Nothing can refuse us from passing. This is meiosis. And the content creator today is M.Saidi, as usual. we have exciting news. We have exciting news on our channel. To our learners who are using our channel, we'll be providing vouchers along the video. So it will be airtime. It can be 10 rand, it can be 20 rand, it can be 30 rand, 50 rand, 100 rand. So in each and every video we'll be releasing from now. Sometimes the first person to comment on our video, then we will take that person and then we'll give a voucher. Maybe it's a voucher maybe for KFC, a voucher for McDonald's. Yeah, as we grow, as we continue growing, you never know that maybe we will have to have big prizes for our channel. Number two is we have also opened another YouTube channel which is going to help us in yeah, we know that this channel is designated to students who are grade 7 to grade 12. We are still building it. We are adding more videos each and every day. We have more teachers who are going to be coming to be producing content. But now we have opened another channel which is going to be shooting to the university, and also at least know how you can make money when you're still a student. Yes, you can also ask questions. It can be questions for health, any questions you feel like you wanna know, maybe, but not in the context of education only. There are some things which parents, people don't talk about. So this is the time. This channel is going to be trying to answer those questions people have. You just drop a question or WhatsApp and then we make a video concerning about the question you have asked. So guys, subscribe and then hit the notification bell, so that when you release any video in our channel, you just know that, or just click the link and then start watching the video. Then you get your airtime, get your prizes. Yeah, the voucher will be embedded in the video, and then the first person to spot that voucher, then we'll be able to use it. But that person who has got that voucher, please comment down that, yeah, I'm so and so, yeah, I've got the vote. Yes, today we are going to be looking at what you call meiosis. I know meiosis is one of the difficult topics, very simple to take, to grasp, but when it comes to answering questions, people get a lot of problems concerning about this topic. Yet is very easy to understand, but it is very complex when it comes to answering questions concerning about it. So what is meiosis? So you can visit our website, www.sandaeduc.com, and then, or if you have any challenge question, then you can also WhatsApp on that number, that is M.Saidi. So I'll be able to try to answer your questions. Let's look at meiosis. Meiosis, we said that meiosis, humans reproduce, other organisms reproduce. They multiply. What about cells? Cells, they have two kinds of reproduction they undergo. They have what called mitosis and you have what called meiosis. Mitosis can occur in other body cells, but meiosis occurs only in the reproductive cells. Where did you come from? Actually, meiosis was the basis of you living. Without meiosis, you wouldn't have existed. So let's look at meiosis in detail. So it's a type of cell division that results in 4 daughter cells, each will have number of chromosomes to that of the parent. So it means that meiosis starts with the one cell. This cell divides and then you form 4 daughter cells. These cells, they have the chromosome number to that of a parent. If you have started with the 4 chromosomes, it means that these cells, they will result in 2 chromosomes. That's why you're saying that half the chromosome number of that of the parent. If the man, the body cell is having 46 chromosomes, then the reproductive cells or the daughter cells which are resulting from that are going to be having 23. And then from the mother also 46, they are going to result into 23. So the process of reducing these cells into
have the chromosome number, that process or that reproductive process is what we call meiosis. So what does meiosis result in? So this meiosis will result in the production of gametes. Gametes, these are the sperms and the ova. And when you're talking about the humans, and then plants, you're talking about spores, the pollen grains. You can talk about the ovules. Those are some of the things which are as a result of meiosis. So in humans or in animals, it occurs in the ovary. See this colored word, in the ovary. Basically, ovary are found in female, and then these ovary, they produce what you call the ova. So the reproductive cells in the females, we call them ova. If it's one, it's called the ovum. If there are many, we call them ova. And then in male, we call it's occurring in testes. And then in testes, what we call them spermatozoa. The cells which are being produced, we call them spermatozoa. So in female, we call them ovary. Those are the ova. Ovaries, the ovaries produce the ova, and then in the testes, they produce what we call the spermatozoa. What about in plants? In plants, occurs in the ovary still, but what it produces, ovary produce what we call the ovules. Meiosis occurs in the anthers, and then what it is being produced is what you call the pollen grain. So the question can come and say that other than the ovary in animals, which other place does meiosis occur? That is testes. And what does it produce? It produces what you call the spermatozoa. And then in plants, it occurs in the ovary. It produces what you call the ovules. Basically, that's what you needed to know about this first introduction of meiosis. You know, meiosis is basically the cell divides, and the most important thing to divide is the nucleus. So we need to know the nucleus to do a recall about the nucleus, because we saw the nucleus at the beginning of DNA and also saw the nucleus in the grade 10. So let's look at the nucleus, the centrosome, and then the cytoplasm. Nucleus contains the genetic material. What about cyto means a cell, and then plasm means a fluid, the fluid of the cell. So basically, this is what you are going to look at. Let's look at them in detail. So this is a cell. It has the nucleus. Outside, you have what called the cytoplasm. Then in the cytoplasm, you have what called the centrosome. Centrosome, why do we talk about the nucleus, centrosome, and the cytoplasm? Because the DNA, you'll find it there, and then it is the centrosome which produces the centrioles, and the centrioles produce spindle fibers which hold the chromosomes in position. That's why you talk about it. And then there is a time when this one disappears, and then you remain the content of the cytoplasm. So the nucleus disappears, and then you remain only with the content of the cytoplasm. So basically, that is a nucleus, and then when it brings it in exam, it looks like this. So this is a nucleus. It's a nucleus, but the nucleus has a dense body which we call the nucleolus. Then outside the nucleus, you have the nuclear membrane which has the extension which we call the endoplasmic reticulum. But because it has the ribosome, they look rough. Then we call them the rough endoplasmic reticulum. The rough endoplasmic reticulum. So basically, that's it. It has the nuclear pore, the holes where substances go through, most especially messenger RNA in this case, and other substances. Then you have what you call the nucleoplasm, the plasma of the nucleus, meaning that the fluid of the nucleus. The nuclear envelope, that's what you call the nuclear membrane. Then the ribosome, we say that the ribosome, the sites for protein synthesis. Chromatin, this one is the one which condenses to form chromosomes. And then you have the nucleolus. Then you have the rough endoplasmic reticulum. So we have explained all that. So when you look at the nucleus, or when you look at the DNA, basically that's what you see. You have, when the chromatin network condenses, you form what you call the chromosomes. So the chromosomes, they condense. You see, they have condensed. And then after that, you form the real structure of a chromosome. It looks like this. So that's the structure of a chromosome. Now let's look at the chromosome in detail. So we have seen where the chromosome come from.
It cannot exist without DNA with its specific genes. You see, it has specific genes. So let's look at the structure of a chromosome. Why do we need to look at it? Chromosome? You resemble your father because of the chromosome, because of the genes you obtained from your parents. Your father, your mother. What about those who don't resemble the father and the mother, but they resemble their grannies? We shall see. Another question. What about why? So in this topic, we're going to see why. Why do you resemble your neighbor? Why do you resemble when you go somewhere? When you go somewhere, someone can say that, hey, when you resemble someone. Why do you resemble someone? Those are some of the questions we are going to answer. And this is the reason why we need the structure of the chromosome. So the structure of the chromosome. Chromosomes are thread-like structures present in the nucleus. They're thread-like. They're thread-like. It means that they are like strings, thread-like structures found in the nucleus. That's why we talked about the nucleus, which carry genetic material. That's why you resemble your father. That's why you resemble your mother, which carry genetic material from one generation to the next generation. So it means that your kids will try to resemble you. Yes. Then you're saying that each chromosome is made up of the DNA. We saw this, that DNA genes are almost the same thing, but on a different scale. So these DNA or these genes are tightly coiled many times around a protein called histones. So the histones, we saw this when we are talking about protein synthesis, and also when you're talking about the structure of the DNA. Yes, so they coil around to form what called the histones that support the structure of the DNA. Because the genes cannot just exist there in air, they need to have a support. So the support we call them histones. So these are the support which make the DNA strand to stand or to be where it is, and so that the chromosome can be formed when this DNA condense. All right, this is the structure of chromosome. Before, remember, we talked about during DNA replication. Does it occur? We say that it occurs before the cell division. Before the cell divide, the chromosome will look like this. Yes, they will look like this. Therefore, we call this one single-stranded chromosome. Chromosome 1, chromosome 2. Sometimes we call them unreplicated chromosomes. They have not yet done DNA replication. But the moment DNA replication occurs, this chromosome is going to change from this format to this format. That's why we say that it doubles the chromosome number. Don't say that it increases the chromosome number. It increases the chromosome number. It increases the genetic material. From what to what? The best answer is double the chromosome number. Doubles the chromosome number. So that when it was like this, then it becomes like that. So this is the centromere, the center of the chromosome. We call it centromere. The two chromosomes which look alike, it means that they share the same structure. Yes. And the position of the genes on them, then we call them homologous chromosomes. Homologous chromosomes. So one chromatid from one chromosome and another chromatid from another chromosome, because they are homologous chromosomes. Therefore, because we are talking about chromatids of homologous chromosomes, then they are going to become homologous chromatids. Why? Because we say that they are coming from homologous chromosomes, but each part is a chromatid. Therefore, they become homologous chromatids. So sometimes, as I say, that this is called unreplicated chromosomes, the replicated chromosomes, or what you call homologous chromosomes. So each of these has two names: unreplicated chromosomes or single-stranded chromosomes. And then these ones, double-stranded chromosomes or replicated chromosomes. So The number of chromosomes in each cell is a characteristic of organisms. For example, humans, we have 46 chromosomes. You'll find out that Drosophila melanogaster has 8 chromosomes. You'll find out that the sheep has over 60 chromosomes. So each organism has a specific number of chromosomes it has. And you might find out that the biggest organism has fewer number of chromosomes. So that's how life, or that's how God is. So it doesn't matter whether you are big or small, but you might have your own uniqueness. We are saying that chromosomes which are single thread or strand become double when the chromatid joined by centromere. So the center of the chromosome, we call it centromere, as a result of DNA replication. So if DNA replication occurs, as we said, now the single-stranded chromosome are going to become double-stranded. So that's why you call it unreplicated chromosome or single-stranded chromosome. The moment DNA replication, yeah. So what are some of the difference between the haploid and diploid? Di means two, ploide means you are describing a body. Yeah, so hap means it's from half the chromosome number. If a human being is supposed to have 46 chromosomes, so when you are haploid, or when the structure is haploid, it has half the chromosome number of that organism. For example, if an organism has 20, the haploid state of it is going to be 10. If the organism is having 32, the number of that organism, or haploid state of that organism is going to be 16. So haploid means half the chromosome number. Diploid means doubling. Doubling. What does it mean? Where is the double there? It means that half from the father, half from the mother, and then they double it. When they double, then they form what called the diploid or diploid. The way you pronounce it. So haploid is the quantity of the cell or organism having single set of chromosomes. They have what called a single set of chromosomes. So if you have chromosome number from 1 to 2, from chromosome number from 1 to 10, these chromosomes must be in pairs. So if you are haploid, chromosome number 1 to 10, they will be in single. They will be 1, 1, 1, 1. They won't be in pairs. So that's what you call a haploid. But if it is diploid, is when the cell or organism has a paired or two sets of chromosomes, one from each parent. It means that one from the father, one from the mother. When they combine, because there are two organisms contributing this, then you form the word di, which is 2. And then ploidy means a body. Yes, it's a state which describes organism with two sets of chromosomes, one from the father and one from the mother. That's the meaning. For sexual reproduction to take place, it means that it must include the gametes. It must include the gametes and must include the sex. Reproduction to take place, yes, what happens? A haploid male gamete, that is, if we are talking about humans, we're talking about sperms, will fuse with the haploid. It means that it will refuse. When you say a haploid male gamete, half of the chromosome number from the male, the gamete will have half of the chromosome number from the male. If the male is 46, the gamete is going to have, or the sperm is going to have 23 chromosomes. Will fuse with the haploid number from the female. Also now the female is going to produce 23. When they fuse together, you form fertilization. So when they fuse together, that process is called fertilization. Now you form what you call a diploid, because half from the father, half from the mother. Then because it's half from different people, then it becomes becomes two. They fuse together. And then when we talk fusion or fertilization, we are not talking about sex, because the fusion or fertilization can take place without sex. Meaning that maybe they used a male just give out the sperms. So when you're talking about fertilization, we're talking about sex has already occurred, and then the sperms, they swim along the female reproductive system, and then fusion will take place. This fusion can even take place. Someone had sex at night, and then you're walking on the street, fusion is taking place. Imagine, did you know when was your fertilization took place? You don't know. Even your parent doesn't know. Okay, those are the wonders of the science. All right. If you need more questions concerning about this reproduction, go and visit our YouTube channel, which you call Bright Doctor. Bright Doctor. I'll give you the link in the description below so that you can understand it better. All right. The result is a diploid zygote. The result is a diploid zygote. So when you have a haploid, you form a diploid or a diploid zygote. So it means that when the sperm fuses with the ovum, you form what you call a zygote. But this zygote has a diploid number of chromosomes. This body has a diploid number of chromosomes. It means half from the father, half from the mother. All right, haploid, half, which is indicated, haploid is indicated as 1N. And then diploid is also indicated as haploid from the mother, 1N. For example, haploid from the father, for example, is also 1N. So when you combine them, you form the diploid, which is whole. Yes. It's the whole. Now half and half, you get whole. Yes, basically half from the father, half from the mother, from the diploid. Di means two, meaning that contribution from one person and then contribution from another person. So because this resultant body is having two contributions, then we call it a diploid. So it is indicated as 2N. We shall be using this language. So you have to know when someone uses this language, you must know what it means. So a sperm, ovum, and then you form a zygote. Zygote is where a fetus develops from. We shall see in the videos we're going to be releasing soon how is the zygote form. You see the sperm swimming after sexual intercourse. You see the sperm swimming, going to the ovum, and fertilization takes place. For more detailed about this, visit what you call the Bright Doctor. Bright Doctor, just a link below. Drop your question, then we shall give you the answers on that YouTube channel. But for purpose of passing exams, we don't go far away from this. But there we can describe as much detailed as you want. So sexual cells is that is sperms and ova are haploid. So we call them haploid. They are 1N, 23 chromosomes in humans. So it means that the other organisms have different chromosomes. So we have what called the somatic cells. So it means that we have two types of cells. We have what called the sex cells. Those are the gametes. I say that meiosis can only take place in these cells. We don't find meiosis in any other cells, only in the gametes. So you have the other body cells. Soma comes from the word soma, which means a body. Yes. So these are body cells, are diploid, which means that they are 2N. If they are 46, it means that the haploid is going to be 23. But the question when it comes, it does not bring only human. You can only talk about 46 if they have specified in the question that this is a human cell, because they can bring a cell with 10 chromosomes. They can bring a cell with 4 chromosomes. If they don't specify that this is a human, please don't use 46, because the question is not asking about humans. Then you have the 23 pair of chromosomes that result in the zygote are divided as follow. How? Number 22 pairs. These are the number of chromosomes we have in the body. 22 pairs. That is from pair number 1 to pair number 22. We call them autosomes. Autosomes. It means that how many chromosomes do we have? 44, because this is a pair number 1 up to pair number 22. We call these ones autosomes. One pair of sex chromosome, that one pair, that is the 23rd pair, we call it sex chromosome, or scientifically we call them gonosomes. Scientifically we call it gonosomes. So this pair is divided into two. We have what called the XX, which is female, and then you have XY, which is male. So it means that females always, they will have XX, and males will always have XY. There is a question which is supposed to be posed here. Why, if females are XX and the males are XY, it means that it's the males to determine the sex, because female have X, also male has X, but female don't have Y, but male have Y. So it's the males to determine the sex of the human being, the sex of an individual. So males, you must stop saying that ladies are only giving you females, they're only giving you boys, they're only giving you girls. Say, I'm giving myself boys, I'm giving myself girls. It's you who is determining this. However, there are some other factors which also determine. It can be on the side of the female. For more details, visit the Bright Doctor. Drop your question there. We will be able to answer those questions. If you drop the question there, then I'll be able to answer those questions. Humans have what we call the chromosomes. We say that from chromosome number 1 to chromosome number 22. These chromosomes are arranged in a specific order, and they appear in a specific way. They are in pairs, and then they describe different characteristics in the organism. So let's look at the karyotype, the composition of the nucleus. Which kind of nucleus do you have? The chromosomes, the type. Karyo is derived from the nucleus, and the type. So which type of the nucleus do humans have? That's derive the name of karyotype. Humans cells are made up of 46 chromosomes. It means that our nucleus have 46 chromosomes. And then these 46 chromosomes are arranged in pairs. These chromosomes are arranged in pairs. It means that they cannot exist as singles. What does it mean if you're not married? All right. It means that they are arranged in pairs. Each pair is different from each other. Each pair is different from other. So these pairs are different. How do they differ? How do they differ? They differ by shape, size, and genetic composition. So pair number 1 is totally different from pair number 2 by size, shape, and the genetic composition. It means that what pair number 1 determines is not the same thing what pair number 2 determines. For example, you'll find out that chromosome number 23, pair number 23, will determine the sex. You cannot find this in other chromosomes. So they vary in shape, size, and the genetic composition. From pair number 1 to pair number 22, number 1 to 22 are called autosomes. What about 23rd pair is called gonosomes, which has XX for female and the XY for male. X, you have to know this, X is longer and bigger over Y. Why? X is longer and bigger over Y. Question, why? Why is X longer and bigger? Bright Doctor will answer that. But on the purpose of this, X carries more genes compared to Y. So a karyotype, an example looks like this. You see that chromosome number pair number 1, number 2, number 3, number 4. They vary in appearance, shape, and also they vary in the genetic composition. So from 1 up to 22, we call them all these. We call them gonosomes. We call them autosomes. And then the 22nd pair, we call it gonosomes. So basically, that's it. Revision of the process of mitosis. It's advisable before you start meiosis to do a revision concerning about the process of mitosis. So guys, our next video, as we say, that we're gonna be having prizes, airtime, meals. Yes, we send you a voucher and then you buy whatever you want to buy. It can be a school bag, t-shirts. We have a lot of gifts to give out on our YouTube channel. So please don't forget to subscribe. Don't forget to subscribe, like, share, so that others also benefit from this content. And then we're gonna be having all the subjects. So we are trying to increase this. So mitosis occurs in every living organism. In the somatic body cell to produce, those are the body cells, other cells other than sperm and ovum, to produce genetically identical cells. So you can have a hole on the mouth, on the cheek, and then you need meat. So what they can do, they can cut the bum and then they put on the mouth. So you move with the bum on your face. Basically, why do they do that? Because these cells, they undergo what called mitosis. They are genetically identical. The meat from your bums is exactly the same as the meat from your face in terms of the genetic composition. So two identical cells are reproduced. Two identical cells are produced with which develop. They're exactly the same. Mitosis has only one cell cycle, i.e., it doesn't have that mitosis 1 and mitosis 2. No, it only has one cell cycle. Only one cell cycle. It means that meiosis, when on the side of meiosis, it has 2 cell cycles, meaning that it has meiosis 1 and also has meiosis 2. So guys, let's look at the process of meiosis. Yeah, the process of meiosis was a very good introduction of meiosis. Now let's look at meiosis in detail. And this will be our next topic tomorrow. Don't miss it. Don't forget to subscribe, like, and then share the video. We will be having gifts, prizes in our next videos, starting from meiosis. Thank you very much. And also don't forget to like, subscribe, and share our new YouTube channel in the link below called The Bright Doctor. M.Saidi was giving you the lesson. Thank you.
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
They're saying is a cross which involves only one characteristic or trait. Trait is the same as the character, and it's being shown in the genetic cross. This guy Mendel put forward the law of segregation. He said segregate means that now individuals during meiosis, they are supposed to segregate. They are supposed to divide, so that the offspring must receive one from the parent male, and one from the parent, a female. So How do they form these gametes which are being formed, which are being given to the offspring? They're being formed during the process of segregation. So he says that diploid, it means that the organism has two sets of chromosome organisms. For example, how many chromosomes do you have? You have 46, but you have 23 pairs. So if there are 23 pairs, then we say that you are a diploid or diploid. Randomly, you don't have a choice which one is going, which one is not going. It's randomly to the offspring, such that the offspring receives one allele from each parent. The characteristics you have now, the parent gave you, each parent brought one allele. If you are tall, for example, it means that those genes you have, one parent gave you one allele for tallness, and another parent gave you one allele for tallness. So if both of them, they gave you a dominant allele, then it means that you are homozygous tall. If one gave you a dominant allele and another gave you a recessive allele, then it means that you are heterozygous tall. So that is the meaning, that each parent is supposed to give you one character. Let's look at how do we mark. This is how we mark in exam. And if you miss something, then it means that you won't get a mark. Number one, whenever they give you a genetic cross, this is what you're supposed to write. P1, which means parent one, phenotype. Phenotype, physical expression of an organism. We get this from the question. A man who is dark-skinned marries a lady who is light-skinned. Dark-skinned man, light-skinned lady. That's it. That's the phenotype. So you go ready. Then genotype. Now, who is this dark-skinned, light-skinned? Is that person homozygous or is heterozygous? That is the genotype. So when you read the story they have given you, you'll find out that you'll be able to scoop out what is called the genotype. So we give you a tick here, we'll give you a tick here. From there, because you have now the genotypes, you have the genes for each parent, then meiosis must take place, segregation must take place. When it takes place, yes, you form what you call gametes. And then this side or the ovum this side, then fertilization must take place. Fertilization must take place so that offspring can be formed. Then you have F1, which means first filial generation. And then now you get the phenotype of the F1. I'm going to explain this. After that, you need to get what called the ratio or the genotypic